Good morning, everyone. Now we are starting with the next session, session 4, omics. The omics deals with the technologies that measure some characteristic of a large family of cellular molecules, such as genes, proteins, or small metabolites. These type of molecular profiles can vary with cell or tissue exposure to chemicals or drugs and thus have potential use in toxicological assessments. On behalf of MIT Institute of Biotechnology, MIT University, Madhya Pradesh, Gwalior, I, Dr. Pallavi Singh Chauhan, Assistant Professor AIB, heartily welcome Chairperson of the session, Professor Dr. Raghuveer Singh Tomar, Department of Biological Science, Indian Institute of Science, Education and Research, Bhopal. Co-chairperson of the session, Dr. Geetanjali Yadav, Scientist, Computational Biology Laboratory, National Institute of Plant Genome Research, New Delhi, and to all the invitees, presenter, and attendees. Now, I would like to invite Professor Dr. Raghuveer Singh Tomar, Department of Biological Science, Indian Institute of Science, Education and Research, Bhopal, for his talk. Professor Raghuveer Singh Tomar is a professor in biological science, Indian Institute of Science, Education and Research, Bhopal. He has done PhD in zoology from Center for Advanced Study in Zoology, Banaras Hindu University, Varanasi. Postdoc from Ohio State University, Columbus, USA in 2008. Postdoctorate from the Penn State University, University Park, USA in 2006. Postdoctorate from University of Texas, Southwestern Medical Center, Dallas in 2003 to study genetic and epigenetic modification executed by cells in response to environmental toxins. I request Professor Raghuveer Singh Tomar to kindly deliver his talk on dissecting the role of epigenetic in regulation of cellular homeostatic pathway under stress condition. Please, sir.
Thank you very much, Dr. Pallavi, and thank you very much. I must thank Professor Tomar also for giving me this opportunity to share some of our research, projects today in this platform. So I'll share now my presentation and let me know whether it's visible or not. Is it visible? My slides are visible?
Yes, sir, absolutely, it is visible.
Great. So I will begin with a broad hypothesis. The topic that I decided today basically is not a very specific topic. I thinking that there are students from undergraduate programs and PhD students. So there is some message for everyone. So this is our broad hypothesis where we, as you all know, that our health is actually maintained through genetics and also epigenetics, right? And both the things can be impacted from various factors, environmental factors, via modulating gene expression. And any abnormal change in gene expression program can lead to disease and normally, otherwise maintained, basically our health is maintained. And there are several age associated disease also happens due to changes in abnormal changes in our gene expression program through modulating epigenetics and genetics. So to begin with, this is just a picture from textbooks. Students, I am sure they know chromatin, what is called chromatin, and the basic unit of chromatin is nucleosome, which is nothing but basically octamer of histone proteins. Where DNA is wrapped around this bundle of histone proteins, and you can see there, thread kind of structure, which are N-terminal and some of the C-terminal regions of the histone proteins, which can be modified by various enzymes. These modifications are responsible for this dynamic nature of chromatin, starting from free DNA to the chromosome. Okay. So the changes that happens to the chromatin, these are called epigenetic changes, which are mostly post-translational modifications within the histone proteins, which can be classified, and the changes can be classified into two major categories: chemical change, which are post-translational modifications to histone proteins, and physical change, where the bundle of nucleosome can be slided back and forth along the DNA, and therefore can have impact on the fundamental biological processes, replication, transcription, DNA repair, recombination, and so on. So so that's where epigenetics has impact on these processes. And there's the other third change, which is called basically irreversible change, where the thread kind of structures, histone proteins, can be proteolytically removed. This is a little bit understudied area within the epigenetics, and we trying to understand how the removal of histone proteins with tails specifically can which processes are regulated by this change. And these chemical changes are broadly classified in basically can be of different types: acetylation, methylation, phosphorylation. I'll show you next slide in more detail. And epigenetics has huge impact or during development several changes are known to happen within histone proteins, chemical change and physical change. And certain environmental factors, drugs, pharmaceutical, also known to impact on epigenetics. Even our diet, physical activities, our behavior is controlled by epigenetics. There are certain drugs which modify histone proteins, which are are in clinical trials. They are the drugs which utilize to some of the psychological, behavioral abnormal changes to correct those by reversing histone modification. So so epigenetics basically you can see is everywhere. Uh, this fundamental, basic unit of chromatin nucleosome was discovered, in late 1970s by Arthur Kornberg, and he did, basically some of the experiments called basically he digested this nuclei, and that you could see that pattern of DNA, multiples of 200 base pairs. So that was evidence that basic unit of chromatin is the nucleosome. Another experiment, as you can see in electron microscopy, also you can see nucleosomes, beads on a string, which is also a model given by him, beads on a string structure of nucleosome. And the third evidence came from, you can have a free DNA, free from protein, and have histone proteins pure. You can actually make nucleosome. So that's the, very three lines of evidence suggest the nucleosome is the basic unit of eukaryotic chromatin. This is there in every textbook these days. We teach students, they learn all these things, and also all the experiments they could do in laboratory. So this is the very basic fundamental aspect of eukaryotic chromatin. Uh, and I already said that there are various changes that occur along the histone tails, which you can see here in this image. Histone H2A, H3, all four different histone protein. Only four different histones are shown here. There are two copies of each. And you can see certain of these amino acids on their N-terminal tail can be modified by methylation, acetylation, phosphorylation, and some of these, modification together also act as a code, like which is termed as histone code, which also basically regulates certain specific biological processes. There is a specific histone modification, histone H3 and H4, which has been termed as code for DNA repair. So that's where repair mechanisms are activated by changing that particular amino acid by post-translational modification within the nucleosome. So so there are likewise there can be many histone codes for different things, which is yet to be discovered. And already I've said that these are the classical modifications. They are mostly chemical changes within histone proteins responsible for dynamic nature of nucleosome and therefore chromatin, and responsible for regulation of those fundamental biological processes, replication, transcription, repair, recombination, and the major one. There are many other, even apoptosis is
Dr. Neha, I'm not able to hear Professor Tomar. Is everything okay?
Uh, um, yes, ma'am. Everything is okay. Sir is maybe disconnected due to network issue.
Okay, okay.
Uh,
Let's
he
schedule for
joined.
that.
He joined.
problem from my end. Sorry about that. These days, I don't know what they're doing. Anyway, uh, did you hear my this slide?
Yeah,
Am I
yes, sir.
audible now?
Yes, sir. Clearly.
So did you hear this slide?
Yes, sir. Sir, make
This
it full
I covered?
screen.
Very good. So this I started, right?
Yes, sir.
Oh, very good. Thank you. So what I'm saying, the chromatin remodeling, change in any change in chromatin from the normal change called chromatin remodeling. There are 4 different mechanisms of chromatin remodeling. One of the mechanisms is called, sliding these histone bundle, nucleosome back and forth can also alter, or DNA can be made accessible or inaccessible for factors that they regulate these fundamental biological processes. Similarly, some of the bundles can be completely removed by some of the histone chaperones. Therefore, DNA is freely available for replication enzyme to act on DNA or transcription, enzymes to transcribe the, make the RNA or to repair DNA or recombination. Okay, and similarly, some of the bundles can be replaced by other bundles, which are called histone variants and chemical change. So these are the 4 major classical examples, not example, mechanisms are responsible for chromatin remodeling or chromatin dynamics, therefore regulating DNA-dependent biological processes. So these are the few, very fundamental information regarding epigenetics. Now I'll go to my, some of the research highlights. So already I've said, in addition to those physical changes, sliding nucleosome back and forth along DNA, therefore regulating the biological process, the histone clipping process or the removal of histone tails is not completely understood or is very poorly understood, I would say. So we're trying to understand this mechanism in our lab. So this process is known in many organisms, tetraamina, mouse, human, and chicken, yeast, and of course, some of the viral infected human samples also that being reported. And the processes, sporulation of fungus is actually regulated by clipping of histone S3 during development, aging, senescence, apoptosis, things like that. Some of the biology, so there are very, very, few studies which suggesting that these processes are regulated by removal of histone proteins, that specifically their end-terminal regions. However, this is not completely understood mechanism. Many times people say when you purify histones, there can be artifact during process of purification and histones are removed. So that is not the case now. There are examples very well studied. I'll show you some of these. So these very, very, very few old studies which has suggested, given some evidence that tails can be removed proteolytically. And this majorly the studies were conducted in tetraamina. Then now basically next question then came into scientists' mind that is it known only in tetraamina, right? Histones are present from yeast to all higher eukaryotes. Even yeast single cellular organism has almost same kind of histone protein that we have. So it cannot be so restricted only to tetraamina. Then it was then basically discovered that these proteins, histone tail proteins, can be removed in other organisms too. So there are the few papers you can see, you can count describing the process of histone tail in different aspects. Let's see, cathepsinol, which is lysosomal enzyme. It is required to remove histone S3 tail, and that correlates with stem cell differentiation. This paper came in 2009 only when I joined IISER Bhopal. This is from Rockefeller, from CDLS lab. And that's where we also started working on thinking how we can contribute this process. And then histone tail, S3 tail clipping, this is also known in yeast, which they say this is required for gene expression for spores, that's in the yeast. And similarly, in other organisms, different histone, and this is histone S3.3 is histone variant. The specific protease, they say. And in most of these cases, enzyme that is responsible for these, tails is not very well understood. So we started our work with chicken model, and you must be surprised why chicken model is basically, almost like, you go to market, you can get tissue, you can do beautiful biochemistry, and economically very important organism. Many of us take poultry products for different purposes. And it's very important to understand, physiology of chicken. So we are very lucky to find that there's a specific histone S3 tail clipping in chicken liver. The study we published in several papers in the last few years. You can see here, this is, all 4 different histone proteins which were isolated from liver, chicken liver. We have compared many tissues, liver, brain, kidney, whatever tissues we could get from chicken. And we found that this process of histone S3, the extra band you see here, this is very specific only in liver, not in other tissues. So there's a protease that we believe which regulating or responsible for histone S3 clipping in liver. And liver, all of us know, is very different tissue than other tissues, right? In the sense that it only basically it has property of regeneration. It also protects us from many toxic chemicals. So probably this kind of histone clipping, histone metabolic rate could be different in liver. Has to be because they are exposed to more extracellular or environmental toxic chemicals. Therefore, their fast removal of stones and with the new stones probably is required. So this is just also a wild hypothesis. Why is so specific only liver? Or it could be basically to regulate liver specific gene expression. Every tissue has different set of gene expression program. So this we discovered that there's an enzyme called glutamate dehydrogenase. Later on, our purification procedure led to the identification of an enzyme, which we then merged, basically mixed with pure stones. You can see only histone S3 is disappeared. Rest all stones are present. So it's very specific bond with the histone S3 of this enzyme. The question is, this enzyme in literature is never known to go into nucleus. Histones are present only in nucleus, right? This is a mitochondrial enzyme, which we say glutamate dehydrogenase. But think that this enzyme exists in different forms. So there is a specific particular form only goes into nucleus, and other this enzyme remains in mitochondria for this amino acid metabolism. So that's the thing that we have. And the next question that we're trying to understand now, what is the significance? I mean, that's the question, right? You will ask, what is the significance of this enzyme removing histone S3 from chicken liver? So we believe that some of the probably removal of histone S3 from chicken liver may be required for tissue-specific gene expression. All right, every tissue is different. So that's the one hypothesis. Yes, certain hypothesis highlights that we're trying to now explore more. It requires extensive work to Yeah, there was again network problem, probably from my end. No problem. I will quickly share my the slides. So I think I explained this slide.
Yes, sir.
Yeah, great. So what we believe that the removal of histone S3 from chicken liver, probably it is required or it is induced by a diet that we give to poultry in poultry farms, or certain chemicals mixed in the form of this antibiotics, or maybe a developmental process, right? We have to see from starting from embryo to older chicken and see at what stage it is more or less we need to do. Also, there are many studies which suggest that neutrophil extracellular traps, right? When there is a damage to tissue, certain of the histone proteins actually are released from outside cell, from inside also, right? And the histone proteins and then some other, fragments of DNA, they actually form, and other proteins form a network that is sort of a basically required for innate immunity of the organisms. This is the well-established fact. So probably some amount of cleavage of histone proteins from chicken liver, they are probably required for neutrophil extracellular trap formation, and that's where health of a chicken is maintained. And we believe that our study is more relevant in this regard, because some of the pathogens we know, influenza and others, can infect chicken, which can be transmitted to human, people who consume poultry products. So therefore, maintaining chicken health is very important in this regard. So some studies suggest that roxarsone is a compound actually which is given poultry feed, which can be metabolized into a much more toxic, chemicals, which has huge impact our health, right? So this compound has been banned in many countries, but in our country, we still use this compound in form of chicken supplement, in the feed supplement. And I believe probably roxasone kind of such chemicals may induce clipping of H1 S3 that we need to study further. And or else, not only that, even this metabolized product of roxasone can actually have impact on our health if we are consuming faulty products. And so that's something that the questions that we're trying to. So our hypothesis here is that histone peptides may be essential to modulate innate immune response in the form of neutrophil extracellular trap. That is the already well-known fact. Or histone peptides may also act as antimicrobials, that we need to discover. And this study further supports our hypothesis, which, as you can see, it says extracellular histones are major mediator of death in sepsis. That's where I also believe that even during COVID-19, when this virus, we know that it actually causes damage to many of our tissues, right? And in tissue damage condition, it is well known from many studies that histones are released outside, and that which can lead to the multi-organ failure. So that's our hypothesis, which one can test further, and let's see how whether or not we are correct. So there are several questions into this end to understand the importance of histone S3 clipping in chicken. We believe that certain stress factors, temperature, humidity, antibiotics, for example, roxasone. Roxasone is arsenic-containing compound, and there are some studies also have suggested that human individuals who are exposed to arsenic from water or soil, in their tissues, histone S3 clipping has been reported. So we believe there is some common, stimuli here in the form of roxasone, which also contain arsenic, or pure arsenic, which is available basically in the soil and water, can lead to, through some mechanism, of course, we don't know what is the mechanism, can trigger removal of histone S3 tail, which can lead to, all these problems. So what is the biological significance? We don't know yet. We believe could be a tissue-specific gene expression in program maybe is required, or there are some proteins. So they are certain of our objectives. I'm not to tell you now. So students probably they will be lost otherwise. So this is our broad project to understand how histone S3 peptide or histone S3 clipped products are required for poultry health. That's that's our biggest goal here. That's we trying to understand. And then next question, we also try to understand certain biological processes. For example, how genes are regulated in terms of basically change within the cytoplasm or change in environment, how genes are regulated by change within the histone proteins. We take example of yeast, which is a single eukaryotic cell, which is very good organism to understand certain of these biological processes. So there we have studied some of the molecules which are basically synthetic or natural. For example, cantharidin is a one molecule which is proposed through many studies. This is anti-cancer molecule. However, mechanism of this molecule, how does it act as anti-cancer molecule is not known. And we, through our study from yeast, we believe that this chemical actually modifies GPA anchor, and therefore it can suppress some of these protein modifications, and then can act as anti-cancer molecule, which needs to be further studied. That's where we have published some of the articles on this. And while working in yeast, we also found that yeast cells, as you know, that fungus can be pathogenic, and fungus is also used to produce some of the useful biomolecules, right? Useful drugs and all. And in alcohol industry, we use fermentation technology, we use fungus. So while working with yeast, we also found that some of the yeast mutants, or some of the even histone mutants, have tendency to form aggregate. And that's very good phenotype for alcohol producing fuel industry for. Okay, so this is something we're trying to understand this mechanism, how each cells actually form flocks, and this kind of mechanism, if we can somehow control or we can have a switch, then it's very good for this brewers, okay, the alcohol producing factories. So that's something we're trying to project, how we can study these things. So we believe that there is a switch which we are based on our study. There is a switch which can allow us to control cells to flock or not flock. That's something this is other highlight of our study, which we are now trying to pursue. But hopefully we will collaborate with some of the companies and to make use of this, to commercialize this aspect of our study with yeast. So with this, I have basically I want to now summarize my study, my talk. Basically, I talked about poultry, wherein we identified proteolytic clipping of histone S3 in chicken liver, and probably that is required for chicken health, and also it probably regulates gene expression, which needs to be studied further. And with yeast, we have utilized yeast to understand
Okay, so all the participants, if you have any question, you can just put it in the question answer box. Yes, sir.
I'm very sorry. There is some problem from my end today. Sorry about that. So, did you? I was here. This slide, did you hear?
So you
No.
were on the next slide,
Acknowledgement.
slide 18th. Yes, sir. Exactly.
Oh, great. So, yeah. So, several of the students who have graduated from my lab, they all gone for postdocs. Some already faculty, some universities, and their work actually. My idea was just to do this or that. This may not lead to the good results. This may be bad. So, otherwise, these are the students who actually extensively contributed all the progress that which I presented today. And some of the postdocs and the other BSMS students who also work in our lab, and some are trainees. And this is our current team, where you see there are PhD students and one BSMS students and one postdoc which has joined recently. My collaborators are mainly from chemistry, and funding support is from different funding agencies. Of course, our institute generously gave us our fund to support our research. So with this, I must thank you all for paying attention to my talk, and sorry about many times my network was broken in between. Thank you very much. I'll be happy to take questions from if you have any questions. Yeah, thank you very much.
Thank you very much, sir. I think there is one question from Alka Rajput, Dr. Alka Rajput.
Okay.
She is actually raising her hand.
Oh, let me see. Okay. What is the question? Can you read that for me, please? I unable to see that question somehow. Let's see.
So actually, she was raising her hand, but now
Okay,
it is not visible to me.
can you please speak out, Alka, so that I can?
I think she has got disconnected.
Oh, okay.
Okay.
Did
So
is-
any of the participant, if you have any question, you can put your question in the question answer box.
Correct. Yeah.
Okay. Dr. Alka, are you able to speak up? Unmute yourself. Okay, sir, can we just continue with the next?
Okay, please introduce. Can you please do
Yeah,
for
sure,
me?
sir.
Can you please
First of all, sir, thank you so much for providing your
problem.
insight on various aspects of regulation of cellular homeostasis. Thank you very much, sir.
Thank you very much.
Now, I would like to invite Dr. Geetanjali Yadav, Scientist, Computational Biology Laboratory, National Institute of Plant Genome Research and IPGR, New Delhi, for her talk. Dr. Geetanjali Yadav is a specialist in genomics and structural bioinformatics with specific applications in food security and ecosystem conservation. She has done PhD in National Institute of Immunology and NIL, JNU, New Delhi. She has wide experience of research in the field of plant genome, interference of botany, and chemistry. She has published several research papers in SCI journal with good citation indices. 20 major research project. I request Dr. Geetanjali Yadav to kindly deliver her talk on spatiotemporal gene regulatory network of algal biophysical CCM for food security. Please, ma'am.
Thank you, Dr. Pallavi, for the kind introduction, and good morning, everybody. I hope you can hear me.
Yes, ma'am.
Fantastic. So first of all, I would like to take this opportunity to thank Dr. Tomar and Amity, of course, for inviting me to the second Int Bio Nano meeting. This is an opportunity for us to be able to present some of our recent work. And I am, as you all know clearly, I am not in front with you physically, but we are all meeting online. However, I do recall and do hope that some locked out currently, I suppose, are able to return to full-time real. The things about college and university time is our social interactions, and I think all of you, it's quite tragic that all of you have, that's what is basically lost out while we are all part of the pandemic. My own memories from my days in college and my days as a student was not which marks I got in which subject. I think what I remember most is which classroom, how the classrooms looked, and how what in the college to spend time, our friends, our favorite teachers. But all of that somehow the pandemic has taken away. And I think it's almost tragic to think that our memories, I don't know your generation, what memories you will carry when history is written and of this phase in our history of the world. Much has changed in the past few years, not just for me or you. As everyone is aware, it has changed globally, and we are still coming to change, I would say, coming to terms with the change that we're seeing around us. So I'm Geetanjali Yadav. I'm a plant scientist. Very kindly, Pallavi has just introduced me. I will today talk about the work we have done for green carbon in food security and how it is even more important now for us to be doing this work. But I was very happy to hear the talk that finished just now by Dr. Tomar. The fact that you've been working on histones, one of my PhD students during the pandemic, he got his PhD awarded. Amish has done his work on histones, core histones, and he has compared them with plant histones. So you were talking so much about animal histones. Interestingly, in plants, the histone fold itself has evolved into DNA binding mode. So a lot of transcription factors in plants have the histone fold. And what Amish found was that the protein binding residues that are required for the dimerization of histone monomers have somehow become less, there's been less selection for these, and there is more on DNA binding. So the residues, there's been some evolution towards DNA recognition and less protein recognition, so that the dimerization still happens, but the octamerization doesn't happen. And these dimerized proteins, dimerized histone-like proteins, are therefore many of the NFY and several other transcription in several other transcription factor families in plants. So it's quite interesting to see how evolution folds about in different lineages in the tree of life. Right, so I will be talking anyway about food security and the work that we've done at the interface of India and Cambridge University, where I've just completed a 5-year deputation as a group leader in the Department of Plant Sciences. And I'll also focus on, as Pallavi said, on the computational techniques that we use to build spatiotemporal hypernetworks that we created for enhancement of food security. The current COVID pandemic, bringing it back into the picture, makes us realize the value of the great outdoors, bringing home the necessity of working together for a better future, for preserving Earth's natural resources and addressing, global challenges. And when you close your mind, what do you think, when you close your eyes, what do you think are the global challenges that we have? Urbanization, deforestation, finding ways to feed an increasing population, fuel security, food security, and above all, climate change, right? So with climate change, as many of you may be aware, much of climate change is driven by the rising levels of atmospheric carbon dioxide. And these levels currently at roughly 440 ppm volume are levels that are much higher than what the Earth has seen in the last 800,000 years. And as we emerge slowly from this pandemic or from the COVID-related lockdowns in different states all across the country, all across the world, I do hope that some of us are able to reflect upon, how important this carbon footprint is and how it has taken tens of thousands of years, and it will take tens of thousands of years for all of that emitted carbon that we create to make its way back into the Earth's crust. So how can we make things better? And leaving aside the causes of this increase in atmospheric carbon, yes, we could do loads and loads of research on that. I think we need to find ways to bring it down. And once again, as I have emphasized in so many of my other talks, if you've heard them on YouTube, this is where plants come to our rescue. And this is where I hope I'm going to start sharing my slides now so that you can see the work that we are going to be talking about today. All righty, can everyone see my slides? Oldest question on the planet right now. I think you can. Right, Pallavi?
Yes, ma'am, it is visible.
Okay, and as happened with Dr. Tomar, I think it's very important for you to tell me or remind me when I get glitched out, because the one thing with an online mode of delivering talks or having an interaction or online teaching is that the internet is our limiting factor. Everything depends upon the internet, and the moment it glitches, I'm quite sure it might glitch, or if the electricity goes and my machine does not have a UPS, I'm going to shut down and you're going to have to remind me through my phone. So thank you very
Definitely,
much.
ma'am. Definitely.
Get on. So back to plants. Even though most of us are aware that plants are the natural air filters of the earth, we still fail to appreciate the true value of the natural capital of plants, and more importantly, of photosynthesis. The photosynthesis, those of you who remember kindergarten science, photosynthesis is the process by which plants are able to A, draw down atmospheric carbon, B, fix inorganic carbon into organic carbon, right? The carbon that all of us, all life forms are made up of, otherwise known as food. And C, release pure oxygen back into the air, okay? So as researchers, the questions that we ask are which plants are most efficient at photosynthesis? So that's the kind of question that we are asking. And where do these most efficient carbon sequesters live? And more importantly, can we understand them somehow to improve the less efficient plants all around us? And so why am I going on saying efficiency of carbon sequestration? Let me come to that also. Surprisingly, the answer, first of all, the answer to the first question, which are the most efficient plants at photosynthesis? Can someone tell me that? And if you can, please put it in the chat box. Before I tell you the answer is that some of the most efficient people at photosynthesis are belonging to this family of Chlorophyceae. So one tiny unicellular green alga that lives all around us, the one that we are working with, mostly in freshwater habitats, but also soil environments, the oceans, you name it, it's all around you. And ocean phytoplankton, in fact, ocean phytoplankton and marine organisms make up less than 1% of the total plant biomass, but they are responsible for nearly half the net photosynthesis that is happening on Earth, okay? And research has actually also shown that green algae are able to fix carbon up to 40 times as well as higher plants. And to understand how they're able to do this, I think you've got to first understand the issue of efficiency in photosynthesis. I had told you earlier, there's this thing about efficiency that you need to really understand. The key is the manner in which these green plants have managed to rectify a flaw that is at the heart of photosynthesis. And what is that flaw in photosynthesis? The flaw is rubisco. How many of you have heard the word rubisco or know what it does or what it is? Rubisco is the global green, carbon fixing enzyme, the guy in chlorophyll, the guy that stands at the interface of living and non-living and performs the critical task of converting inorganic carbon to organic carbon, as I told you earlier. The thing with Rubisco is that it performs a little less well. It doesn't perform very efficiently in most plants. And why is that? That's because Rubisco is not able to distinguish atmospheric carbon dioxide from atmospheric oxygen. Okay, and plants usually respond to this situation of the wrong number dialing by making huge amounts of Rubisco. And so much so that even by default, even if half the time there's a wrong number being dialed, sometimes the right number will be dialed and the correct substrate will bind to Rubisco. So much so is this huge amount of production of Rubisco that Rubisco is the most abundant, arguably the most abundant protein on the planet. And yet, being this much, it is still not enough. How could it be enough? What is the matter with the wrong number? Oxygen, obviously oxygen levels in the air at 21% are definitely higher than carbon dioxide levels. However bad it is still less than oxygen. And so therefore, back to the question, how are green algae fixing this flaw? So I'm going to tell you how green algae fix this flaw by going on to my next slide, where I introduce you to the model unicellular alga that we are working with, and that fixes carbon very well. This little guy is called Chlamydomonas reinhardtii, and I'm going to call it Clammy. This is the tiny little plant form that we work with in the lab to understand the business of the super efficient removal or fixation of carbon from the atmosphere. So how does Clammy do that? The ingenious trick that is being used by this little beast is that Clammy has evolved a mechanism to physically separate Rubisco from oxygen. Now, other plants have also done that. And I want some of you students who remember their kindergarten biochemistry, can you write in the chat box and tell us what are the other mechanisms that plants have come up with, or maybe cyanobacteria, to improve the efficiency of Rubisco by separating oxygen from Rubisco. So if oxygen does not come in contact with Rubisco at all, obviously it will not make the mistake of dialing the wrong number. So it will only see carbon dioxide. So that's what Clammy has done. It has physically separated Rubisco from oxygen, resulting in a tremendous increase in photosynthetic efficiency. And Clammy does this by packaging. So how Clammy does this is what I'm going to tell you. What you are going to tell me is how other plants do it and what are those mechanisms called. Okay, so Clammy does this by packaging Rubisco in a tiny little micro compartment called the pyrenoid. All right, and pyrenoid is what you see in this picture. Do you see this guy in the center, the new right below over here, the purple shade? I don't know if you can see my my mouse moving about, and so you should be able to see this little guy here, the purple guy. That's the pyrenoid, and that's the guy that contains Rubisco. It's chock full of Rubisco. That's the little, I would say it's a microorganelle, and we call it a microcompartment actually. And the pyrenoid, as I said, is chock full of Rubisco, and it is designed in such a unique way that while carbon can enter the pyrenoid, and it's full of Rubisco, so Rubisco sees carbon, oxygen cannot enter it, okay? And thus by proxy, Rubisco sees only the correct substrate. Even though the atmosphere is full of oxygen and carbon dioxide, Rubisco gets to see only the right substrate, which is carbon dioxide. And such mechanisms that physically separate Rubisco from oxygen by concentrating the carbon dioxide around Rubisco, around the physical vicinity of Rubisco, they're called carbon concentrating mechanisms, okay? And these have evolved, as I said already, not just in Clammy, but also in blue-green alga. And the other things, I'm looking at the chat box, I don't see many of you have responded, but the way they do it is, the way higher plants do it is called C4 mechanisms or CAM plants. What we are trying to do with Clammy is that we are trying to identify key components of the algal biophysical CCM in the hope of using it to improve crops or higher plants, okay? And this is what brings me, in this kind of a preamble or a background, it brings me to the work that we are doing with my collaborator and colleague, Professor Howard Griffiths, in the Department of Plant Sciences at Cambridge. This drawing of Clammy has also been made by one of the students in the department. And our research is therefore to interrogate very large genomic data sets and to find molecular fingerprints, all of which, could give us some patterns in the DNA, maybe RNA, maybe proteins, that help us identify the details of this process. In our case, it's algal carbon sequestration. In your case, it could be any other process that you're interested in. You could use very similar methods as we've been using. And so what we've done is to collate over 50 important genes for several, using several decades of published literature in CLAMI. And then we took these core set of important genes and then we divided them into time segments. So before that, let me just tell you what we've done with the genes is a separate story. But what I want to talk about is how you could have a favorite organism of interest, what you should pay attention to if you want to ask questions that have not been answered, are about what kind of data is available. For instance, in CLAMI, we have, as you can see on the slide, we have the nuclear genome sequence, we have the organellar genome sequence, both of them annotated, fully assembled. We also have nuclear time-resolved transcriptomes and carbon-deprived transcriptomes, which allow us to see what happens when under carbon deprivation and under normal circumstances, where normal carbon would be, I would say, 0.04% in the ambient solution, right? But when it becomes, when there's carbon deprivation, how does this kick in and do even better at sequestering carbon from the atmosphere? And then we have the organellar transcriptomes, we have the spatial interactome, which means the interactions between different proteins. At what locations do these interactions happen? Protein-protein interactions can be transient and they can be long-term, and they can also very often be spatial. So where is that interaction happening? In other places, it may not be happening. So we do have the spatial interactomes characterized for CLAMI, which is a great thing, lucky for us, because we're in a good position to then extract out the key components of algal carbon sequestration. Then we have individual microcompartment proteomes, such as you can see on the slide, centriolar, flagellar, chloroplast, the CCM repressed phosphoproteome is also there. And we have the regulome, which means we also know who's regulating whom. So the list of transcription factors in this genome, their targets for this genome. And when you look at the regulome, essentially these days, in addition to the transcriptome, in addition to the genome, of course, antiquity-oriented genome, we also look at the regulome, which means we want to know which part of the genome is regulating which other part of it, under which circumstances. And we have several related genomes, which are pyrenoid positive and pyrenoid negative, which allow us to compare CLAMI with these cousins and brothers and sisters in order to understand how algal carbon sequestration or how the CCM works. And on the right side of this slide, you basically have the method that we've used, that we begin with the core carbon concentration components of chlamydomonas, which we call the CCCC5C. And this is a list of genes that we find important, which I'm trying to speak in terms of students who are listening, who may have their own questions. Come up with a set of genes that you're interested in, whether you know their roles or not, whether, based on review of literature, which is where all of your research should always begin, however short, however long. And then you get their gene attributes. Then you try to perform different kinds of network analyses. What we've done is we've built so many different networks for these, and then we've performed visualization and superimposition of these networks. And I will be showing you how how using all of these different networks allowed us to gain insights into the process that we were interested in, namely the mechanism of the algal biophysical CCM. And then we use this to identify new or key components, and certain components which we found were very, very important. We started looking for them in higher plants and other algae to be able to see whether, especially the components that are helping rubisco get pushed and how should I say, two-sword inside the pyrenoid. So that's what we were doing. And so here are the core set of important genes, which I call the 5C, and we divided them into time segments, as you can see over here. And we did because we do know the cell cycle, and we know which ones of these get expressed at what time. As I told you, we have the transcriptome. We also have the time-resolved transcriptome, which means we know which genes get expressed at what time, right? So during the day-night cycle of growth, we know out of our genes of interest, which ones are getting expressed at what time. So in this picture, what you see here is a heat map, and the green colors are basically showing you genes that light up or get activated at various time points in the clammy day-night cycle. And now for each of the time segments, you see 5 time segments over here, right? For each of these 5 time segments, which you're seeing in this heat map, basically the G1S to G0 timing, we call it, starting from there, we call it the division, resting, pre-dawn, and these 3 are basically in the dark, which happen at night. And then during light, you have the active CCM and so on. So for each of these time segments, we extracted the genes that are switched on, or in other words, bright green, that you see in that vertical box. Each time segment is a vertical box, and in each vertical box, you see a set of bright green lights on. So we pull out these bright green lights because each row is a gene, and then we built gene correlation networks in order to understand how the genes in that time segment are associated with the remaining genes of clammy. How many genes does clammy have compared to plants, which have more than, often more than 30,000 genes, and humans somewhere about 2022. Clammy has 17,000 genes, and we wanted to understand how these genes in CCM are related to the rest of it. So our work pipeline was to begin over here, and I call this to a large extent supervised analysis, because we begin with a review of literature where we identify some genes of interest to ourselves, but quite often regulatory novel factors, and more recently, as we are advancing with data science and statistics and machine learning, a lot of unsupervised machine learning methods have also been developed, which allow you to identify regulators of critical processes, which we have also then used. So we use supervised ML and unsupervised ML to generate gene regulatory networks, and then superimpose them with the protein-protein interaction networks for the same 5C genes. And then we identified hubs in these genes, and we consider those hubs to be important. But more important than that, or the second step or the final step, was where we partitioned the networks into clusters. And then we identified which are those partitions which are more important. Okay. So most of this work has been done by my student C2. And this is our pipeline. I'm sorry, I was just trying to explain to you the pipeline without showing it how it works. So the spatiotemporal analysis has been done both at the level of the locations of these genes and the knowledge that we've had, and the temporal knowledge that we have of which time these genes are getting expressed, and so on. So we have a knowledge-based supervised analysis, and we also have the unsupervised clustering that we perform, which is also very well known as the weighted gene co-expression network analysis, of the WGCNA. We have applied it here, and we pulled it together to get regulatory inferences using common hubs. And as I said, this work has been done by C2. So in this slide, you can basically visualize, as in front of you, the clammy CCM gene correlation networks in various time segments. So you have the dark on one side, and you have the light over here on the right side. And at various time segments, C2 has built these gene correlation networks, or GCNs. And what we observed, and what you can see in this figure, is that there are critical regulatory subnetworks that are active at different times, at different time points. Most importantly, let me see clearly what we've got on this slide. Yeah, right. So most importantly, as you can see in this figure, do you see there are critical regulatory subnetworks that are active at different time points, especially in the pre-dawn picture? If you can look at the pre-dawn picture, you have this little orange set of the little orange glob over here, and so this is the second network, and we call it the orange globs, or the orange nodes, or the orange circles, are genes that are active only at that time, and the gray guys are the ones that are that may have been active but at another time not here. So all the subnetworks, all the temporal subnetworks, are basically superimposed, and the subnetworks that get switched on are orange, the ones that are off are in gray. And this allowed us to to identify novel regulators or novel clusters that light up only at certain time points. So this, in the pre-dawn network here, for example, each dot, please remember, is a gene, and the orange dots are the ones that get switched on only at this pre-dawn time segment. Okay, and since this activity takes place a few hours before dawn, which is the onset of light, we are assuming that this is extremely important for CCM because active CCM happens in full on light. Of course, that's where photosynthesis is kicking off. And so we are now in a position to explore these little details, these little pieces of information in more detail and identify missing links in the regulation of the clammy CCM. Okay, so overall, in the long term, what I'm hoping is, based on the work that we have done, based on the networks that we've created and the temporal separation that we've been able to achieve in these networks, we hope that some of the work from our molecular research can be translated to the field, as you can see over here, even to crops. And much of this work is funded by this program at the India-UK interface, which I think is a transformative, unique kind of effort, especially in terms of its ability to perform, for us to do interdisciplinary work, which I'm realizing over the past few years is just the right way to do research. So within Tigris, we are transforming India's green revolution by research and empowerment for sustainable food supplies. But what we've been able to do it is in collaboration with the Punjab Agriculture University and ICRISAT, we've been, and some of our collaborators in Princeton and York University, we have been able to successfully get some elements of the biophysical CCM into higher plants like tobacco. And for me personally, this has been, quite a learning experience because we have been able to work together at the intersection of multiple fields and technologies. We have 6 multidisciplinary flagship programs in one grant, over 30 organizations, both in the UK and in India, right? And all partners have joined hands. There's so much we do for putting together. In the olden days, I used to come and meet y'all also, and I used to be traveling a lot to meet children. But a lot more has been about, being able to, especially during the pandemic, to be able to teach things. So either way, what we've been able to do in this program is come together with people we would not otherwise have met, meet students from places that I would not otherwise have been able to meet, and researchers who do completely different things. So everybody speaks about interdisciplinarity, but I think true interdisciplinarity requires you to bring down methodological rigor, people who work in areas which are totally different from yours, right? So we've worked with supply chain modelers, agricultural economists, farmers, women's collectives, robots, geography experts, mathematical simulators, all kinds of data analytics is what we've done to address the global challenge of feeding an ever-increasing population, okay? And of course, we teach and train rural youth, urban youth to contribute to capacity building. The trainings that I perform, you're more than welcome to attend these. You can join me on Twitter. My Twitter ID is GILIENV, GILIENV. But the thing is that more often than not, these trainings allow you to learn, especially over weekends. I hope you get back to college and I hope you learn from your actual teachers in real time. But if you don't have that kind of freedom, then on weekends, I usually take these courses. This course I did, this one I started just last month. I mean, I did it last year. It started last month also. The next one is going to be in, right now we're having the March, April 22 wala course, sorry for 21 on this slide. And then the next one is starting next month. We also did something really interesting during this with women. All aspects of digital, the gender divide is worse for women, right? So we decided to teach women how to do AI. And we got teachers, professors, even vice chancellors, lady vice chancellors from many universities who came together for 4 days to do this course with us. And I think that was real fun. I'm doing the next version of the Vividya course is it in April this year, April 2022. You're more than welcome. Again, all information is, you don't have to email me. You can find the information on Twitter. Our partners will also be sharing the same things. The Twitter world is where you get onto. And I think the best part during the pandemic for me has been most incredibly fulfilling is this internships that we have done with our students online between India and Cambridge that we continue to do. And we have mentored students during the pandemic where we began, initially we were just doing, we used to do internships every year, you remember, before the pandemic, but then everything got locked down and we felt that students have extra time, but they have nowhere to go. And so as bioinformaticians, as computational biologists, we decided to, move on with students and continue to teach them during our lockdown. And that's what we did by joining hands with Indian academies, as well as the Department of Science and Tech. Of course, Cambridge and NIPG are involved, the FAST SF. You're more than welcome to join this initiative if you want to. And these are some of the courses that I'm going to be teaching in 2022. Forget about 21 and all of these. Everything is going to happen in 2022, and all the updates and additional information on posting about other courses is going to happen on Twitter. With that, I thank you for this opportunity to be able to interact with you, and I'm happy to take questions if you have any. Thank you, Pallavi.
Thank you so much, ma'am. I think one person is raising her hand. This is Muskan Bhatia. Muskan, do you have any question?
Should I
You can
stop?
put your question in the chat box, Muskan.
Ma'am, there is one question about the C3 and C4 cycle. Someone is writing in chat box, Shan, Shan. Can you elaborate this question? What you
Oh,
are
I don't think that was a question. I think that was an answer to my question, because I was initially trying to get students to talk about what are the mechanisms by which plants are able to overcome this inefficiency with Rubisco. And you correctly wrote C4, but C3 is not right. C3 is where you have the whole inefficiency, and C4 plants are the ones that are able to improve efficiency correctly. So C4 is right. CAM is also right, but CAM nobody has written here, but I did tell you about it. So that was an answer. Are there any questions? I'm happy to take those. Otherwise, join me online at you can always ask. Today is the International Day for Girls and Women, and so that's another good thing. If we have girls here, I think you have to be able to ask yourself questions. Why are you in science? What is it that you want to solve? And don't say poverty in the world. Have a question which is clear, which you think from your literature analysis, from your knowledge, has not yet been answered, that you can pick up, right?
okay? Thank you so much, ma'am. There is no question in chat box.
No worries. Thank you so much. Glad to have been here.
Thank you very much, ma'am, for providing your deep insight on the genomics and structural bioinformatics with specific applications in food security and ecosystem conservation. Thank you so much.
Pallavi, 1 minute. Gitanjali ma'am, there is one question by Anukampa. Can you explain heat map little? She is asking about heat map.
Oh, heat maps. Heat maps are basically correlation plots. So when you have data in rows and columns, and you want to understand whether there's a correspondence between the variables in your rows and the variables in your columns, you make heat maps out of them. Most often you would have seen these in case of gene expression data sets, where all of in your table, you have thousands of rows, tens of thousands of rows, each row is a gene, and your columns are basically gene expression values under different experiments, controls, something that you experimented with, or time series experiments. So your columns are the samples and your rows are the genes, and you want to know if certain genes have certain correlation with certain samples, such as in the experiment. Does something happen uniquely in the experiment that does not happen in control? And which are the genes which are doing that? So you would have a correlation matrix built out of these normalized gene expression values. And to view the correlation matrix, you could either look at an Excel sheet with zillions of numbers, or you just put colors for high, low, and medium values. And so when you put these colors and you see a box full of colors, and those colors have shades which tell you have some meaning, then that's called a heat map. And we call it heat map because red is hot, and in the olden days you used to have green and red color, but that was before the world realized the value of color blindedness and being able to make pictures or figures that did not impact colorblind people, or they were colorblind friendly. And so we don't use red and green so much more these days, but the word heat map has still stayed, because red would mean really hot and really high value of transcription. We use it for generating correlation plots between gene expression values. And the techniques, there are many techniques. We work everything in ours. So the courses that I teach also are about data science. And as a student, I think it's very important for you to pick up whichever subject you're studying: medicine, zoology, geography, English. Learn data science, my dear, even if it is social media and not just biology, but learn data science. In data science, there are many different platforms, I would suggest pick up a free platform so you don't have to worry about licenses. And I would say pick up the easiest one, that is R. There's a huge social community. All of our work, all of C2's work is been done in R, and you can also use R. So that's the answer to your tools. May I know, there's another question. Convention effect. I didn't get that question, Vijay.
Vijay Singh,
Effect.
can you rewrite the question? It is not clear.
Yeah, it's slightly not clear.
Effect in DNA over heat transform at the high temperature.
No, I'm not clear about your question, and I think it is not connected to my talk at all. Are you talking about quenching, maybe? DNA quenching. That was not my talk, Vijay. Did you hear my talk at all? So that question, I think it is not related to CCM, so I won't be taking it in this session. Are there any questions related to the talk that I gave?
No, ma'am. Thank you so much, ma'am. There is no more questions. Thank you, ma'am.
Thank you so much, ma'am.
It's a pleasure to
Now,
have been here, and I hope to meet you again. Bye.
Now, I would like to invite Dr. Sameer Srivastav, Department of Biotechnology, Motilal Nehru National Institute of Technology, Allahabad, Uttar Pradesh for his talk. Dr. Sameer Srivastav is Assistant Professor, Department of Biotechnology, Motilal Nehru National Institute of Technology, Allahabad, Prayagraj. He has expertise in regulation of gene expression, epigenetics, colorectal cancer biology. He is member of several research bodies. He has published 3 books, 6 book chapters, and several research papers in various national and international journal of repute. I request Dr. Sameer Srivastav to kindly deliver his talk on transcriptomic profiling of early onset CRCs. Please, sir.
Yeah. So am I audible to all of you?
Yes, sir. You're audible.
Yeah. Thank you, Dr. Paloli, for a nice introduction. At the very outset only, I want to thank Professor R.S. Tomar, sir, convener of the
There is some network issue.
Yes, sir.
audible or not? Hello?
Yeah,
Hello?
there is a small break.
Am I
So
audible?
now, yeah, now we can hear you. Yeah, please go ahead.
Sir, I request you to kindly share your slide. Sir, is there any network issue?
Okay. Thank you, sir. Thank you, sir. So
Yeah, we are unable to hear, and neither we able to see slides. Pallavi ma'am, maybe you ask him to join, restart again from his end.
Yes, sir.
Sometime.
Actually, he is trying to reconnect, but
Oh, okay,
there is
okay.
some network issue, so we have to
Yeah.
wait for a few minutes.
Yeah, no problem.
Yes, sir. ये जो है इनका net
Yes. Am I audible to everyone now?
Yes, sir, you are audible as well as visible.
Okay.
I
And
request you to kindly share your slides.
yeah, I'm sharing my slides. So I'm sharing my screen actually. So
Okay, sir.
I'm sharing the whole screen, entire screen. Okay. So is my screen visible to everyone now?
Not
Okay.
yet, sir.
So
Maybe it will take few seconds. Let's wait, sir.
so yeah. So thank you for giving me this opportunity. And
Sorry,
it's
sir. Voice is very cracking and slide is still not visible.
a wonderful conference, I believe, having experts from all over.
Samir sir, sorry for interrupting. Sir, your voice is cracking and slides are still not coming.
Is this visible now?
Not yet, sir.
Sir, I think the slides may be visible later on, but till then you can give the introductory part of the presentation.
Yeah. Okay. So I'll be talking about transcriptomic profiling in these type of cancers at a very alarming rate. So in these cancers, CRCs basically they the onset is at a very early age, but preferably before the age of 50. Okay, so the age at diagnosis is
I request you, Samir sir, can you please change the internet connectivity? Because we are unable to hear you clearly. The voice is cracking very much. Raghavi sir, can we just wait for a few minutes?
You asking me?
Yes, sir, I'm asking it to you. So can we
Yeah,
just
no
wait for a
problem.
few minutes?
I'm just saying that
please.
let them try different options.
Yes, sir, definitely. Thank you for your patience, sir.
No problem. I'm fine.
UCRCs. Okay.
Lisa, your screen is not visible. Pallavi ma'am,
Yes, sir.
I think some problem from the some visa and so
Okay, sir. Can we just take the next invited talk?
Okay, now again it is gone. So I'll share.
those are is not visible right now. So I think you should check your connection and then rejoin again. Ma'am, meanwhile you go ahead with the next candidate.
Okay, sir. Thank you so much.
Okay, okay.
Just a minute, sir. Now I would like to invite Dr. Gopalji Gopal, CJ Bhakta Institute of Biotechnology, Oka Tarsadi University, Bardoli, Gujarat, for his talk. Dr. Gopalji Gopal received his PhD degree from Special Center for Molecular Medicine, Jawaharlal Nehru University, New Delhi in 2012. Currently, he is assistant professor at CJ Bhakta Institute of Biotechnology, Oka Tarsadia University, Bardoli, Gujarat, and also worked as assistant professor at Department of Biochemistry, Faculty of Science, Maharaja Sayaji Rao University, Baroda. Also, from 18th May 2015 to 5th August 2015, he served as a scientist under Fast Track Scheme of Young Scientist, and also he has about 10 years of teaching experience. He has a specialization in the area of recombinant DNA technology and molecular biology, and research interests include the plastic degrading recombinant enzymes, type 4 secretion system of Bacillus anthracis, and Heliobacter pylori. I request Dr. Gopalji Gopal to kindly deliver his talk on role of recombinant DNA technology in the management of disease. Please, sir.
Thank you, madam, and thank you to all the management of MET and conference committee for giving me opportunity, and you have given me earlier than my time, scheduled time. So it's good. I was waiting for this so that I can complete before time and I can go to my institute. I am delivering from my home, and I have connected with broadband. See how my internet is working. So now I'm showing my presentation. Fine. Is it visible?
Yes, sir, yes, visible.
Visible, and I'm audible also.
Yes, sir, definitely.
So madam has given a nice introduction, but I think that is old CB or something. I delivered 17 batches of recombinant DNA technology. Means I have conducted 17 batches of 30 hours course in recombinant DNA technology. I'm not very good researcher, but yes, I teach. I love to teach. So my topic is not very concentrated on my that data based or result based. This is common topic so that research scholar and a student may get benefit, and they can utilize that potential of recombinant DNA technology in future scientific endeavor. That is my purpose. So what is biotechnology? So using living organism or material for commercial, industrial or health purpose, overall welfare of human. And biotechnology is the use of biological process. So there are different definitions. So in every definition, we use the biological system for betterment of human life. And gene manipulation technology or recombinant DNA technology, gene cloning has revolutionized the biotechnology. Then only after first recombinant product, so every country have started biotechnology division, Department of Biotechnology. Means recombinant DNA technology is a fuel of biotechnology. And we all know that gene is the basic functional unit of heredity, and genes are DNA segments. Means all gene are DNA, but not all DNA are genes. And every organism means आदमी के बच्चे आदमी, गधे के बच्चे गधे, due to transfer of that gene, means their offsprings are similar to their parents. Means gene directly or indirectly is responsible for every character of organism, living organism. And why this gene manipulation technology become feasible, possible? Due to this universal decoding pathway, almost universal. In all organism, this decoding pathway is almost universal. Gene to mRNA and mRNA to protein. This central dogma is almost universally present from bacteria to the higher organism. And codons are also universal. Due to this two universality, gene manipulation technology become feasible and possible and fruitful. So you all know that this is basic gene cloning steps. Depending upon source available and knowledge available, we either isolate the mRNA, then convert it into cDNA, and then we produce the fragment which we wish to clone. Either we can go for directly PCR-based isolation of the gene, or we can sear the genomic DNA, and then we clone that fragment. Or if that PCR method is not available, genomic DNA is not available, then we can go for synthetic DNA. So these are the ways to isolate the gene of interest to be cloned. So we modify it, digest it, and then separately we digest the vector, means isolate the vector and digest it with compatible restriction enzyme, modify it, and then we mix this vector and the gene of interest or DNA of interest, and then we ligate. So we can transfer that ligated product if it is plasmid, and if it is a bacteriophage vector, then we have to package it into bacteriophage coat protein, and then we infect it into E. coli. In this way, we can get transformed E. coli. And so we select the positive clone, means we select the transformed colony, and then we select the positive clone. And from that positive clone, either we multiply that gene of interest, means we culture that DNA and that bacteria containing or harboring our recombinant DNA. So we can get multiple copy of that DNA, or we can harvest the protein coded by that gene. This is overall process of production of either protein coded by that gene or multiple copy of that gene. So now if we discuss pathogenic disease, so normal strategy to tackle that pathogenic disease is, if it's not known, then we either give them analgesic, antipyretic, or other drugs. If that region of cause of disease is not known, so कोई भी disease है उसमें पहले pain होगा, उसमें पहले heat होगा, fever आएगा, so we give. But after diagnosis, if we know that this disease is due to some bacteria, then we can administer antibiotics. And sometimes if that antibiotic doesn't work, so we can prevent entry of that pathogen inside the host, so that it will not enter and it will not cause disease. Another strategy may be, if it enters into the body, then we try to prevent its multiplication, so that whatever number infected, whatever viral or bacterial load is, starting load, it will not multiply inside. Or suppose we are not able to control at that point, so we try to control the secretion of the pathogenic protein, toxic protein. So how can we stop that? So we neutralize that toxic protein by adding inhibitors, either a small molecule or antipeptide we can add, or we can administer antibody against that toxins. And if we are not able to add antibody or anti-toxic things which can neutralize the toxin, so we try to reduce the production of that toxic protein. So how? So by using antisense RNA technology, we add the toxin coding. I mean, we add antisense RNA of that toxin coding mRNA, so that it will not participate in translation. In this way, production of that toxic protein will reduce. And if that pathogenic disease is like a pandemic, then we produce a vaccine. We try to produce a vaccine so that we can save the population from infection. So this is a preventive method. So we produce the vaccine. This is overall general strategy to tackle the pathogenic disease. Not all diseases are pathogenic, caused by virus or bacteria. Some diseases are genetic disease. Some diseases are due to malnutrition. So we will see how recombinant DNA technology playing a role in reducing the burden of the disease. Where is the where recombinant DNA technology playing a role in reducing the overall burden of the disease? Fine. So now you can see we will discuss this five strategy. So as you all know that if we provide nutritious food, then disease burden due to malnutrition will reduce. I will show you that role of recombinant DNA technology in providing nutritious food in third world country, in underdeveloped country. Well, most of the disease are due to malnutrition. If people are not getting sufficient food, or at least micronutrients deficiency is a major cause. Then we will discuss prevention of pathogenic disease, how recombinant DNA technology playing a role in prevention of the pathogenic disease, and then how recombinant DNA technology playing a role in diagnosis of disease, and then treatment of disease. So under 5 subheadings, we will discuss role of recombinant DNA technology in managing the disease. Now, one by one. So we all know that malnutrition is major cause of disease in developing and underdeveloped country like India, like other South African country. So recombinant DNA technology have produced several high nutritious food. Like you all know, golden rice having high vitamin content. So if people will consume this golden rice, they don't have to take vitamin A pills, because through genetic engineering, this carotene producing gene has been added, so that rice have, in addition to their normal content, it has more amount of vitamin A precursor, that is carotene. High iron containing transgenic plant. So this transgenic banana have high iron in normal, in comparison to normal banana, it has high this iron. And you all know that most of the pregnant women, they take these iron pills, folic acid and other things. So if she will consume this banana, they don't have to take extra things, iron. There are several other food items having extra nutrition, vitamins, or you can say mineral containing through that transgenic or you can say recombinant DNA technology. Using tools of recombinant DNA technology, scientists have produced nutritious food item having extra vitamins, extra mineral, so that if someone will consume, they don't have to take extra that nutrition. In this way, if we consume this nutritious food, so burden of malnutrition related disease will reduce. This is a role of recombinant DNA technology. People are working, so many successful stories known, but people are working on other food items or crops. And you all know, so second is recombinant DNA technology plays a vital role in prevention of disease, means production of vaccine. And you all know that prevention is better than cure. So if we are able to prevent this pathogenic disease, so disease burden will reduce. Nowadays, we all know that coronavirus, you can imagine if there would not be vaccine, then what would be the impact of this pandemic. Am I audible? Whenever we get disconnected, just let me know.
Yes, sir, you are clearly audible.
Okay, good. So production of different categories of vaccines. So in almost all types of vaccines, there is role of recombinant DNA technology, gene manipulation technology. In all types, there are different types of vaccines, there are different modes of vaccines. So vaccine is the administration of antigenic material to stimulate an individual's immune system to develop immunity against that pathogen. So we use either entire pathogen or their subunit, their proteins. I will show you that. So different vaccines are available against the different, you can say, pathogenic virus and bacteria. You all know that this, see, I have prepared this. So there are different modes, attenuated vaccine. So we can attenuate the bacteria or virus by removing their virulence factor, their toxic things. So it will not cause disease, but it can elicit immune system. So for removal of that pathogenic disease, again, we apply the recombinant DNA technology. Inactivated vaccine, you all know, either we heat kill or we chemically kill the bacteria. So in that case, we don't use recombinant DNA technology because sometimes we use heat killing or chemical methods of killing. Then we don't use the recombinant DNA technology. But for attenuated vaccine production, for subunit vaccine production also. So we use recombinant DNA technology method to clone this coding gene, the gene which codes this subunit, success exposed protein. Whose mic is on? Okay. So for subunit vaccine also, we apply recombinant DNA technology. Plasmid vaccine, DNA plasmid vaccine, DNA vaccine. So we can use cloned gene in the plasmid as a source of vaccine, or we can directly use that DNA which code for that antigenic protein. We can use lipid nanoparticle encapsulated mRNA. So in different modes of vaccine, we use recombinant DNA technology. Without using recombinant DNA technology, we cannot produce vaccines. And you all know the role of vaccine in prevention of disease. So you can see this data I have taken from somewhere. So diphtheria in 1950, 5,796 cases, but after vaccination, 0 cases. Means there is reduction, 100% reduction after vaccination. Population may have increased threefold from 1950 to 2009. But you can see there is 100% reduction. There is no cases of diphtheria. Tetanus, there is 96% reduction. Pertussis, 86% reduction. Means due to vaccination, there is huge reduction of pathogenic diseases, cases of pathogenic diseases. Here you can see, I have taken this from WHO. There are different vaccines are available, successful vaccines are available for administration into the population. So you can imagine if that vaccine not available for normal population, then what could be the impact? Now, third, so recombinant DNA technology in diagnosis of disease. So you all know that in this case, in corona cases, everybody, even non-scientific people were also suggesting that diagnosis ज़्यादा होना चाहिए, ज़्यादा से ज़्यादा लोगों को diagnose करना चाहिए. So molecular basis of diagnosis, it's more sensitive and you can say more authentic also. So we have different ways of diagnosis. PCR based diagnosis of disease, PCR, RT-PCR, real-time PCR, use multiplex PCR, all these things are used to diagnose the disease. So presence of bacteria or virus or their respective gene can be identified. Antibody-based diagnosis of disease. So ELISA, immunocytochemistry, immunohistochemistry, Western blotting-based, radioimmuno-based, immunofluorescence, immunoelectron. So in all these things, we need the antibody for diagnosis. Now one can say that antibody, where is the use of recombinant DNA technology in production of antibody? Because it's produced by animal cell or their cell line if it is monoclonal antibody. So as you all know that for production of antibody, we have to inject the antigen. So we can purify that antigenic protein from native source also, but most of the time we don't prefer because amount or proportion of that protein is very less in native sources. So we go for again recombinant DNA technology method, means we clone that gene which codes for antigenic protein, and then we purify that protein using different tags. So we are dependent on recombinant DNA technology for production of that antigenic protein, which can be injected into the animal so that we can produce either monoclonal antibody or polyclonal antibody. And we are using this antibody, you can see here, for different methods of diagnosis of the disease. DNA hybridization based diagnosis. So you all know the southern blotting, northern blotting, FIS, DNA microarray. All these techniques are based on hybridization method. So if we have that probe, labeled probe, we can use this probe for identification of our desired presence of our desired DNA among the millions of the DNA. Among the mixtures of millions of DNA, we can identify presence of our DNA. So DNA sequencing based diagnosis. So single nucleotide polymorphism, as you all know that we all are different though 99.9% genome sequences are similar to all human, but 0.1% is different. Due to that difference, someone is susceptible to some disease, someone is more prone to the disease, someone is resistant to disease. So we can identify single nucleotide polymorphism either by sequencing, or there are different PCR-based methods also available, ARMS PCR and other methods. Gene sequencing can also be carried out, finding triplet codons. So a few of the neurogenetic disease where there is more number of CAG repeat, which cause the disease. So we can identify that presence of that repeat either by sequencing or through PCR also. So now we will discuss briefly about all this. So you all know that PCR. So it's in vitro amplification of known DNA sequence. So for PCR, your sequence must be known, then only we can identify. So for that knowing, we have to go either for sequencing, genome sequencing, or for different methods of Sanger sequencing. But if we know this sequence, then we can do that PCR. So there are two characteristic feature of PCR which revolutionize the molecular biology. So you can see here, increase the quantity to millions to billions times. So in 20 to 30 cycle, you can get million to billion times amplification of given DNA. So PCR is the method to increase the quantity of that DNA up to the level of identification, because you cannot identify presence of 2 or 100 copy, 200 copy of DNA. For that, you have to amplify it into million copies so that at least you have nanogram, picogram level. Second is amplify only a specific sequence, means for a screening. So PCR is an alternate method of gene cloning, and PCR is also an alternate method, or you can say substitute of southern blotting and northern blotting. If in most of the cases we can substitute northern and southern blotting with PCR. In most of the cases we can substitute gene amplification cloning method with the PCR. So this is very versatile technique. If we have antibody, we can identify presence of any protein either on cell surface, or if we have some extract, cell extract, we can identify presence of that protein. And that protein may be specific to your pathogen, your virus, your bacteria. We can identify through antibody. So you have seen that recombinant DNA technology playing a very good role in diagnosis of the disease. Treatment of disease. So we can produce therapeutic recombinant protein using recombinant DNA technology. And you all know that insulin and several therapeutic, hundreds of therapeutic proteins are available in market. So nowadays people are not suffering due to deficiency of that proteins, either insulin deficiency or growth hormone or clotting factor. There are several recombinant proteins are available, which are used as a therapeutics. Therapeutic nucleic acid. So you all know for gene therapy, we need that more amount of that DNA. So addition or deletion of gene which cause the disease or mask the expression of disease causing gene by antisense technology. So we can mask the expression of non-desired gene, or we can replace the non-functional gene with the functional gene. So for all this gene therapy, we need more amount of that DNA. So we apply recombinant DNA technology to produce that DNA or RNA. Therapeutic. So therapeutic antibodies. So antibodies conjugated with toxins kills cancer cells, tissues, and for other wherever we wish to neutralize the non-desired protein, toxic protein, we use monoclonal antibodies. And there are different forms of monoclonal antibodies are available. So again, either we for production of antibody, you all know that I have discussed that we use recombinant DNA technology to produce antigenic protein. Even if we are not using recombinant DNA technology for production of toxic this antigenic protein, suppose you have purified that protein from natural sources, not used. But nowadays, people are using recombinant antibodies also. If you know the heavy chain sequence, if you know the light chain sequence, so we can produce recombinant antibody in E. coli also, or in other host also. Kitna minute bacha hai, madam? Okay. So,
Sir, you can summarize within 5 minutes, sir.
okay. ठीक है, हमने अपने टाइम से ही किया है. Okay.
Yes, sir.
So, antibodies conjugated. So we use, there are several applications of monoclonal antibody. So we are dependent on recombinant DNA technology. Antibiotics. So again, you all know use of antibiotics. So used to treat microbial disease. Antibiotics are secreted by microorganism. So one can say that where is used. So to increase the quantity of natural production of antibiotic, again we use metabolic engineering technique to increase the production of antibiotic by same host, same bacteria. Biosynthesis and production of natural drug. So again we use same host, same plant, same animal for production of you can say natural drugs, but we apply recombinant DNA technology to increase their production. Like people are working on increasing the production of vinblastine and vincristine from that plant. Means in treatment of disease also we use that recombinant DNA technology. So these are list of, this is old, but these are list of recombinant therapeutic proteins available in the market. You all know gene therapy we can use for replacing the functional gene, for antisense gene, for killing these cells. So there are different modes of gene therapy. This is a list of therapeutic monoclonal antibody for different disease condition. We use this monoclonal antibody. Again, we apply recombinant DNA technology. How we produce, you can see. In you all know that impact of discovery of antibiotics. So this data is only for penicillin. So it is estimated that penicillin has saved at least 200 million lives since its first use as medicine. You can see this is a serendipity, but it has saved 200 million, means 20 crore people. Increased drug biosynthesis and production by metabolic engineering. So removal of rate limiting, how we do metabolic engineering. So removal of rate limiting transcriptional and allosteric controls, kinetic enhancement of rate limiting enzymes, genetic inhibition of competing pathway, enhanced carbon commitment to the primary metabolic pathway from central metabolism, modification of secondary metabolic pathway. These are the strategy to increase the metabolites which may be of medicinal use. In this way, we can, this is you can say strategy for metabolic engineering. In this way, we can increase the production of therapeutic compound in the same host without changing the host. This is very important to understand the cause and mechanism of disease. So transgenic animals as a model human disease. So to understand the new disease or efficacy of new drug, we must have that model organism. So we create disease mice, rabbit by deletion or addition of gene to understand the disease. Creation of model of Alzheimer's disease by overexpression of amyloid precursor protein. Creation of mouse model by knockout for hypoxanthine guanine. Gene targeting has been widely used to model human cancer caused by the inactivation of tumor suppressor gene, such as TP53. Finding of functional role of unknown gene. As you all know that due to presence of nowadays this next generation sequencing platform, it's easy to sequence whole genome, but it's not easy to determine role of all the genes present in the genome. Hardly we know 2 to 3% gene and their roles, but we have known sequences of trillions of bases or more than trillions of bases from different organisms. But again, we have to apply recombinant DNA technology method to determine the role of the gene, either by gene knockout, gene knockdown, overexpression, production of recombinant protein. In pharmacogenomics, so role of genome in drug response, it opens the field of personalized medicine. Means to understand the new disease or efficacy and efficiency of new drugs, we have to apply recombinant DNA technology. So means in all the aspect of management of disease, we apply recombinant DNA technology. So this is summary. So for production of nutritious disease, prevention, cure, diagnosis, to understand in all the aspect of reducing the burden of the disease, we use recombinant DNA technology tools and techniques. This is very important technique. Fine. So I think I have summarized within time. क्या बोल रहे हैं नेहा मैडम?
Yes, sir.
So, but I prefer question answer. If there is any question, I welcome participants, and I'm also available on LinkedIn, Twitter, Facebook. So participants, mostly a student, can join and ask question in future also. But now it's open.
So there is no question in question and answer box and in chat. If they have, sir, they contact you.
Okay.
Thank you so much.
Thank you.
Thank you very much, sir, for providing your deep insight on the role of recombinant DNA technology in the management of disease. Thank you so much.
Thank you, madam.
Now, I would like to request chairperson of the session, Professor Dr. Raghuveer Singh Tomar, to kindly sum up the session. I request chairperson of the session, Professor Dr. Raghuveer Singh Tomar, to kindly sum up the session.
सर का भी इंटरनेट नहीं चल रहा लग रहा.
Yes, there is a lot of problem.
Dr. Pallavi,
yes, sir.
This is Pankaj from NIB Hyderabad.
Yes, sir.
I was supposed to talk in this session or
sir,
some other session
in the
somewhere?
next session, sir. In the next session,
Huh?
I'm just going to just close this session.
Okay, I thought. Okay.
Yes, sir.
So,
okay.
Sir, are रहे हैं?
No, sir. Gitanjali, our chairperson, co-chairperson, both are not connected. So we are continuing with our next session.
Okay. Now I can leave, na?
Yes, sir. Thank you so much, sir.
Thank you, madam. Thank you.
Dr. Pallavi?
Yes, sir.
Yeah.
So on behalf of AIB AUMP, I would like to thank all the eminent persons who added glory to this session. Now this is the time to close this session. And as per the schedule, we will be continuing with the next session. This is Animal and Medical Biotechnology. Thank you very much.
Yes, you are audible.
Dr. Pallavi, Dr. Pallavi, am I audible?
Yes, sir, you are audible.
Yeah. So Dr. Ramkumar Mishra sir has joined.
Yes, sir.
Okay.
Yes, I'm very much here.
Sir, I welcome
Good
you
morning, everyone.
on behalf of MIT Institute of Biotechnology, MIT University, Gwalior.
Vikas sir, Dr. Pankaj Suman sir is also with us. Both chairperson of next session.
Okay, Dr. Pankaj Suman, I heartily welcome to you all.
Thank you very much.
I think that my camera is not working. Yeah. So, Dr. Pallavi, we can start.
Yes, sir, definitely. Okay. So good morning, everyone. The animal biotechnology include creating transgenic animals with one or more genes introduced by human interventions, using gene knockout technology to make animals with a specific inactivated gene and producing nearly identical animals by somatic cell nuclear transfer or cloning. Whereas medical biotechnology uses living cells and cell material to research and then produce pharmaceutical and diagnosing products. So on behalf of MIT Institute of Biotechnology, MIT University, Madhya Pradesh, Gwalior, I, Dr. Pallavi Singh Chauhan, Assistant Professor AIB, heartily welcome Chairperson of the session, Dr. Ram Kumar Mishra, Department of Biological Science, Indian Institute of Science, Education and Research, Bhopal. Chairperson of the session, Dr. Pankaj Suman, National Institute of Animal Biotechnology, Hyderabad, Telangana, and to all the invitees, presenters, and attendees. Now, I would like to invite Dr. Ram Kumar Mishra, Department of Biological Science, Indian Institute of Science, Education and Research, Bhopal, for his talk. Dr. Ram Kumar Mishra is an associate professor, Indian Institute of Science, Education and Research, Bhopal, India. Dr. Mishra had done his PhD in biology from Department of Biological Science, DBS, Tata Institute of Fundamental Research, TIFR, Mumbai. Postdoc from Sensory Cell Regeneration and Development Section, Laboratory of Molecular Biology, National Institute of Deafness and Other Communication Disorders, that is NIDCD. National Institute of Health, NIH, Rockville, MD, USA. His research interest is focused on molecular and cell biology of nuclear pore and their dynamic during cell division, mitotic event during cell cycle, microtubule dynamics in mitosis and developmental biology. He has several research publications in various national and international journals of repute. I request Dr. Ram Kumar Mishra to kindly deliver his talk on getting insight on cancer, ex vivo laboratory approaches to delineate the menace. Please, sir.
Thank you. Thank you, Pallavi, for introduction, kind introduction. And thank you, Dr. Tomar, Dr. Vikas, and Dr. Raghavendra for giving this opportunity so that I can discuss some work which is related to what we do as well as what in general people do in worldwide to understand the cancer. So I hope my screen, I'm sharing it, is visible. Is it visible, ma'am?
Yes, sir, it is visible.
Okay.
It is visible.
And my slides are visible?
Yes, sir.
Yeah. So good morning again, and welcome to this happening event at Amity Gwalior. And I actually was going through the initial sessions as well, where Dr. Kaushik was introducing that how cancer is a big burden and what we need to do at different level to ensure that this thing is cured and we get a solution of it. So what I'm going to discuss today is the how different laboratory practices are in place, which are helping us delineate this menace, which we call cancer. And I'm sure everybody has heard of it, or at least everybody has heard that their Keith and Kins or somebody in distant relation has suffered through this disease, right? And when we think about cancer, what we, is it not moving? Yeah, it's moving. So when we think about cancer, what comes to our mind is that there's something which is uncontrolled as a growth, and that is leading to tumor, and that may lead to further poor differentiation and metastasis. But actually, cancer is not just about the uncontrolled growth. It could actually mean a lot of problems which are associated. What you are seeing in this background image is a cell which is trying to divide, and this structure which is present here is called spindle. The entire chromatin is assembled in the center in some structure called metaphase, and these microtubule structure, which are protein, which help ensure equal division of the chromatin into two daughter cells. So cancer may occur because of problems which may happen during this process of division, and which may lead to the uncontrolled growth. So actually, tumor or cancer is not just uncontrolled growth of cells, but it also reflects in many other ways what we understand, that in the morphology of the cell also changes. It's not just the number. While it is dividing rapidly, uncontrolled manner, the morphology of the cell also changes. Like here you see that nuclei is much bigger. And quite often it has been noticed that the nuclei, there are multinucleated cells. And what we have been learning, our students know that every cell has a single nucleus. That's actually the normal scenario. But in cancer, the number of nuclei can increase. And many of the dividing cells which are in the cancer, they are disorganized. So every tissue which we happen to have in our system is very well coordinated and organized in terms of what cells are there, what proteins are there, how extracellular matrix is arranged. And there's a very good communication between cell and extracellular matrix to ensure that the tissue functions normally. That particular aspect can also go wrong in cancer, as well as the chromosomal abnormalities come in. I was showing you in the previous slide how that spindle-like structure ensures, with the help of microtubules, that two daughter cells receive equal chromatin content without any problem. But in many cases of cancer, chromosomal abnormalities arise. We have heard about aneuploidy, polyploidy, and so on. So aneuploidy is something which is very common in cancer cells. And at the same time, what is another important feature is that they lose the shape. The differentiation of the tissue becomes very poor. So when some of the cancer tissue is used for biopsy purpose, we realize that the cell has lost its differentiation. That is also a mark for progressive stages of cancer. The poor differentiation is one very hallmark feature. And because of that and certain other features which we have mentioned earlier, one key feature happens which is called epithelial to mesenchymal transition, which leads to the metastasis, where cancer or tumor moves from one place to another place, which is much more detrimental. So to gain an understanding or to even to develop a cure, potential cure, we need to understand this particular thing in greater detail. So how we can understand what is going on wrong in cancer and what tools we can develop accordingly. So if you think about it, the cancer can be addressed at multiple levels, and which is most prevalent in clinician setup is that patient samples are derived, they are sectioned, they are stained to look for these morphological changes which I mentioned in the previous slide, as well as people look for presence of certain biomarkers which can be associated with the cancer. What has been coming up in recent time is something called organoids, where cells which are stem cell nature, they can be tuned to differentiate into an organ-like structure, that is organoid. And these organoid-like structures are very helpful in terms of maintaining the extracellular environment, tumor microenvironment, and thus they are more close to the patient sample. Then one can also assemble the three-dimensional assembly of the tumor cells, which are grown in laboratory conditions. And these tumor assemblies, the cells which are assembling into tumor, they give you a appearance of a sphere or spheroid, and they can also be used for different types of study. So when we are going from patient sample further down to the item number 5, which is cell culture, we are moving from a macroscopic view to a microscopic view. But eventually what is being most commonly used in different laboratories which are trying to get insights into the molecular details, the changes which are happening at the cellular level, the most preferred one is the cell culture models. I'll take you through each one of them one by one. So when we discuss about the patient sample, what can happen is that some patient sample can be used for immunohistochemistry analysis, where tissue can be sectioned, they can be stained by a certain dye. Some of the very common dyes are hematoxylin and leucine, and as well as the presence of certain RNA can be assessed by in situ hybridization. Right? So just to see if there are certain features which are changing at the protein level or some defect or aberrant expression of some of these RNA molecules are happening. And thus one can derive more information directly from the tissue. And this has been the practice in many labs, whether it is research lab or the direct hospital setting where clinicians work on this. So what you see in this table on the left side is that there are different types of tumors and how a particular molecule has been detected in terms of its intensity, where M stands for moderate, S stands for strong, and W stands for weak. So by using these immunohistochemistry analysis as well as in situ hybridization analysis, you can actually say whether the tissue is having normal expression level or very strong reactivity. And thus you can associate certain cancer with a biomarker molecule that every time when there is a cancer, this molecule will be present in moderate to strong levels. Similarly, one can also look for using these methods, using patient sample and immunohistochemistry, one can look for the presence of certain proteins as biomarker, I said, and one can associate. So what you saw in the previous slide in the table, it was the quantitation, and this particular image which is here is giving you a visual appeal of the same thing, where a protein which is shown to be playing important role in gastric cancer called cell cycle and apoptosis regulator 1 is tested in different tissues. So in normal tissues, there's a very poor reactivity, or you can say this is the normal level of this protein in the tissue. But when you start getting into the tumor fractions, whether it is tumor negative, you may feel that there is some elicitation of the signal right here in tumor negative. But as tumor is becoming more aggressive from weak to strong, you can see this brown color appearing, which is actually detecting the protein in the system. So by this, you can associate a biomarker to a cancer type. As well as using these tissues, what we have also shown that some proteins, they are expressed in the higher concentration. While upper image is of immunohistochemistry, this is your Western blotting images showing that under normal circumstance, the level is very poor. But in case of tumor, the level of these proteins increase in different samples. So same has been quantitated here to suggest that given molecule, or if there is an oncogene or protein, which is oncoprotein, that can be increased, the level can increase in case of cancers. So clinicians do prefer this macro level view to suggest that what exactly morphologically is going wrong, what kind of molecules are present, if a given molecule is present in higher concentration or the lower concentration, right? It gives you that information. But obviously there are some limitations because the pace is very slow. It is dependent on the availability of the tissue, and it does not give you core detail in what is going wrong inside the cell, how you can develop. It can give you, it can tell you about the biomarker, and diagnostic tools can be, prognostic markers can be defined, but diagnostic tools may not be developed just based on the biomarker defined here. So one needs to understand the cancer in a more detailed way, in a more facile manner, something which is more giving you the answers much faster. And there are many different ways by which you can achieve this. So one of the things which I told earlier was organoids, and as the name suggests, that organoids are the three-dimensional structure. They are formed by differentiation of the progenitor or the stem cells. So like here you see in this picture on the left, the organoids which have been formed, and you can feel the presence of different types of cell and their relative orientation. This gives you a feel how cells are arranged in tissue. Because when you talk about a tissue, it's a complex assembly of different types of cell. Students who have been learning animal biotechnology or animal physiology, they have studied that the location of each and every cell type in a tissue is very precise. And something goes wrong with that can lead to the cancer. So organoids help you give rise to these structure which mimic the way cells will look like inside the original tissue. And what can be done, that since you are using organ progenitor cells or stem cell, you can actually pick a stem cell from a given organ, and you can make it behave like, or you can generate the organoids from there. So like here shown, from different organs, organoids can be used, and this can be studied directly into the system. And they are more, they are coming into fashion now. This is very recent development where people are trying to establish organoids to gain more insights into that. But what it is useful is that it gives you the similarity with the regular tissue, and you can actually generate organoid of a given organ for which you want to study, rather than using a plain cells and so. However, this again is little cumbersome process. A lot of customization needs to happen. A lot of optimization need to happen to ensure that organoids are forming. And they give you three-dimensional appearance, but they may not give you the exact morphology of the organ. So there are still some limitations people are trying to understand, deal more in detail. But definitely this is going to be the future, the way cancer will be studied in a more holistic tissue level setting. The next level of understanding, when we are going from macro level to the micro level, is a spheroid. As the name would suggest, some cells come together and they form a spherical structure. And these cells are multicellular assemblies. I would like to tell you that they do not form a very strong connection which will give you appearance of a tissue. Rather, these are multicellular assembly, like a colony. So spheroids are like a colony where you can understand many different aspects. So the image shown here is like a spheroid here. Multiple cells have come together. They are not in a tissue-like arrangement, but they are held together still relatively more tightly than individual cells. One can visualize them, one can understand more details of that. So this is again your immunohistochemistry showing that how these different cells are expressing different kind of molecule, as well as you can also test some of the markers inside these spheroids to suggest whether the cells are empty, some process is activated, and so on. On the similar line, these MCTs can be used as a in vitro setup where the drug efficacy, cytotoxicity growth, or other cell-related aspects can be studied, but a pseudo feel of a 3-dimension associated with that. Like this study which we have performed, we show that your spheroids can be formed and you can actually test the effect of a drug which should affect their survival. So what you notice up here is that untreated spheroids have a given morphology. A low concentration of this drug is not affecting the spheroid as such, but when you look internally and look for the effect on the cells, you realize this red signal coming up, which is indication of cell death. And as the cell death proceeds at higher concentration of this drug, the spheroids disintegrates into much smaller mass. So for such studies, these spheroids can be used effectively. So in some sense, they are a better system as compared to simple 2-dimensional cell culture, but they are nowhere close to 3-dimensional assemblies, and they may not be very useful or have a very diverse application in understanding the cancer. The something which is more closer to handling the cells individually, which can give you more insight on what is going on wrong at the cellular level, whether it is on the surface of the cell, whether it is in the cytoplasm, or something really going wrong into the nuclear organization, chromatin distribution, and so on, where cells are grown in a matrix. Why it is important? Because I have been emphasizing that cells inside a tissue, they are not lying on a flat surface, which is hard, right? There is a definite softness associated with the substratum in which cell is growing. So that softness or that environment, tumor microenvironment or extracellular environment, is provided by some of these complex structures which are called matrigel. And this will be more closer to a system where a cell you are trying to understand at individual level, but you are giving an environment which is more like the organ environment. So like here, a study shows very clearly that when you grow these cells on a substratum which is like absorbable sponge, which may give you feel that it is soft enough and the surface undulations are there, the cell is growing, or you can provide certain extracellular components, so some calcium derivative and some lipid derivative to see how it affects. But when you provide matrigel, which is going to have a gel, has a lot of proteins like collagen, BSA, and all that, when that is present, the same cells, you see how nicely they grow and differentiate. Why? Because this arrangement is more closer to the tissue arrangement or where the organ is functioning. And same has been quantified here on the left side, where when cells are grown on matrigel, you see more living cells as compared to other two cases. So this may still be sufficient to perform many of the studies, but most relevant information probably you will get when you have these matrigel associated over there. Right? And these matrigel based systems are used to understand how cells are moving on a surface. If required, how cells will go deeper into the surface and perform the invasion. This becomes very important when somebody is trying to understand the metastatic nature of the cells. So cells move from one place, go through the extracellular matrix and find a new location. So matrigel assays are very useful in carrying out those kind of studies. The final thing which I'll talk about is the cell culture system, which is regularly 2-dimensional hard Petri plate. Cells are grown and they can be used for many diverse purposes. As a researcher in the laboratory, Many people prefer this because of its ease as well as the diversity for which it can be used. It can be used to understand the biochemical processes which are going on inside the cell biological processes, understand more about the growth and inhibition of the cell. This can also be used for expressing or knocking down a particular protein and understand their role in the cellular physiology. And on top of that, they can be used for understanding the interacting partner. I was listening to Dr. Geetanjali Yadav and how she was saying that different interactomes can be understood and the method they have developed. So these cells can definitely be used that for such establishing such interaction paradigm. So what you see here in the coming slide is the image here, where you can ask the protein which you are interested in to see how they are located inside the cell. Because I told you that in the very second slide that in cancer, a lot of changes happen in the cell morphology. And thus the localization of key important molecule can also change. So what you see here is that one protein which probably can be important regulator in cancer progression. And when you map it inside the cell, what you realize that it is very nicely present on these structures which are called nuclear membrane. So same things can be used to understand protein, also provide you temporal information about the interaction. So what you notice here is that this protein, a part of this protein, which is here NUP88, it's a nuclear pore protein, and trying to understand how it binds to the other nucleoporin, which is NUP62, a pull down experiment performed. So what you're doing is that you're trying to see if this protein 62 can interact with 88, and this protein is present in the cells. What is noticed here is that the cells can be left of their own, grow normally without any without subjecting them to any treatment. At the same time, you can subject the cells such that they are arrested in a phase of cell division, cell growth, and a phase which is where the division is happening. But what you'll notice here is that irrespective of the treatment given to the cell, in all three cases, this interaction is happening. So such temporal information can be derived from a protein-protein interaction analysis performed using these 2D cell culture assays. Further, trying to understand how cells migrate, especially this is a method which tells you that if cells are able to migrate and form a new identity or a new place or retain the identity. So it is also called a wound healing assay because same thing happens in the surface wound. When there is a cut happens, cells migrate to the area of the wound and heal them. So what we do inside the cell is that inside the 2D cell culture system, that you grow the cells, then allow them to grow to a higher confluence and then create a wound. It can be just a simple scratch in the layer of cell and then ask how things are changing. So this study, which has been reported by others here, shows very clearly that if you have cell and you create a wound, as the time progresses, because of the migration, this wound is healed. So area between these dotted line is the wound area. So you can see that in control, this wound is healing. But if you knock down certain molecule, you want to understand about function of certain molecule, like here CCAR1, I told in the very beginning, which was important for gastric cancer. So when you knock down that molecule, this wound is not healing. So by that, you can assess whether this protein is important for migration properties, which may be contributing to the cancerous property to these cells. Similarly, you can also form, if the cells have lost any contact inhibition, which is a hallmark of cancer cells, that normally cells, when they grow, they touch each other, they don't, they stop growing beyond that. But cancer cells actually go beyond that. So what can you do is that you can grow these cells and ask if these spheroid-like structures can form, which will be an indication of contact inhibition loss. The same thing performed, one can see that more spheroids are forming when you overexpress a protein like this. Similarly, you can also ask if these cells are capable of forming colony, right? And same study suggests that you have more colony forming in one case versus the other. And the staining which is here also suggests that cells are viable. So some of these studies which will be giving you direct input at the molecular and cellular level about what happens, what goes wrong in cancer can be derived from here. This is a small video which I would like to play to show that how we can get more information from the cells, that what is going on wrong. So these are live images which shows you that cells can be subjected to certain treatment and see what is going on. Here what you're noticing is some of the cytoskeletal proteins, when they are subjected to a treatment, they move from a filament structure to a more of a granular structure. This is a sign of degradation of that filament. But what you notice here is that you see much more granules forming here, right? So what kind of information you can get is that if there is a protein which is present in normal scenario versus a mutation scenario, what changes can go on. So if you notice here the T58 here, that's a 58 minute, and this is also starts with T58. So what you can obviously notice that here the system is not disintegrating at this time point, but here it has started to disintegrate very strongly. And what is the change here is that one protein expressed here as a wild type and a mutant protein. So this kind of information you can derive by studying the cell culture in a two-dimensional approach. And I'm sure when you go to the matrigel assay or spheroid assay, you may not be able to get these information very clearly. So what I would like to summarize that for a laboratory researcher, the use of 2D cells is imperative, because we derive a lot of critical information about whatever is going on at the cellular and molecular level. And this will help. This process is rapid and helps in the diagnostic and cure development. A small comparison here shows that the cell lines which are normally used in the research laboratory, the advantages obvious are reproducible and rapid growth. The cost is also relatively low, as well as high throughput analysis can be performed. But disadvantage is obvious. It is oversimplified because it is a 2D monolayer culture, whereas in patient 3D samples, you have more cellular environment, more tumor, extracellular tumor environment present, which is not the case in 2D cells. While 3D cultures have definite advantages, that they allow more similarity to the tissue or the patient sample, and they retain the microenvironment which is present in the tumor. But as I mentioned earlier, that the process is little slow, it may not be helpful in defining or doing these tests, which will give you more cellular perspective, as well as the performing these experiments is costly, because you remember organoids, which I told in the very beginning, you have to differentiate the stem cells, and that itself is a very costly time-taking event. So as a whole, if I say a couple of points, the whole tissue-based analysis which clinicians do and many researchers do, that can never be substituted, because that gives you very different aspect about the cancer. But 2D culture is something which has been the backbone for cancer analysis in laboratories, and it has yielded many more critical information which have been taken further up to develop diagnostic tools. 3D culture in forms of organoids is definitely the future, where cancer can be studied in greater detail. With this, I would like to thank, and I'll also like to thank the Institute and other places which I have received funding. So I would stop here and would like to take questions if they are there.
Thank you very much, sir. If someone has any question, they can put their question in the question and answer box.
No, Pallavi, there is a question.
Okay.
Thank you.
Thank you very much, sir, for providing your deep insight on cancer, ex vivo laboratory approaches to delineate the MNEC. Now, I would like to invite Dr. Pankaj Suman, National Institute of Animal Biotechnology, Hyderabad, Telangana, for his talk. Dr. Pankaj Suman is a scientist and principal investigator at the National Institute of Animal Biotechnology, Hyderabad. He did his PhD from the National Institute of Immunology, New Delhi. He also worked as visiting scholar at the Friedrich Schiller University, Jena, Germany. His research experience spans through various disciplines of science pertaining to immunology, reproductive biology, veterinary, and clinical medicine. Till now, he has published many research articles in various reputed journals. He is the recipient of the several prestigious fellowships and awards from agencies like Council of Scientific and Industrial Research India, Department of Science and Technology India, United States Department of Agriculture USA. He is also the recipient of prestigious GP Talwar Young Scientist Award for outstanding contribution in the reproductive health. He has attended several national and international conferences and delivered invited talks. I request Dr. Pankaj Suman to kindly deliver his talk on field applicable APTA sensors for detection of residual antibiotics in animal food product. Please, sir.
Uh, Thank you, Dr. Pallavi, for a nice introduction, and it's my pleasure to be here. And I also thank Dr. Tomar, Dr. Anurag for inviting me and giving me opportunity to present my work over here. So I will go ahead and I will share the presentation. Dr. Pallavi, is it visible?
Yes, sir, it is clearly visible.
Okay, great. So our laboratory works on various aspects to develop diagnostics, and aptamer is one of the focus area where we work. And today for this talk, I have selected detection of antibiotics because it's a like nowadays lots of problems we are facing, and a big problem we are anticipating that antibiotic residual antibiotics in food products may create a big havoc for antimicrobial resistance. So let's begin with the what residual antibiotics, how it can it's posing a threat to the human and animal health. So India is the biggest consumer of milk in the world, consuming around 77 million metric tons of milk that is coming from cow, buffalo, and sheep and goat. And antibiotics are widely used in livestock industry for antimicrobial properties against various diseases, and it's a long time consumption of such antibiotics through milk may create serious health complications like development of antimicrobial resistance. Apart from that, some health issues also. Most of the tests show which kind of milk that you are drinking. The milk is the milk which you are consuming every day, and specifically the kids. Do they have antibiotics or not? We don't have any kind of kit that can be used for testing at the doorstep or at the level of consumer.
Hello.
The reason is most of the kits are imported, and this kits costs also that in Indian rupees its cost is very high, and more than 150-200 rupees per test. And because of misuse of antibiotics, more than 500,000 people are dying every year around across the globe. So to reduce the cost without compromising the sensitivity of antibiotic detection, we have developed an aptamer-based lateral flow assay for oxytetracycline detection as a proof of concept. And we are now working on with the different other antibiotics that is being commonly used in veterinary practices. So I will show you some data for how we developed this aptamer-based technology for affordable diagnostic for antibiotic detection. Because right, so for that, these aptamers are very small nucleotides that can bind with varying level of targets, bigger molecules to a smaller target molecules, and a smaller target molecules where detection of generation of antibody is difficult. You can generate an aptamer. So it's being preferred because of low cost, thermostability, chemical synthesis, and the pointer. So chemical synthesis, less time of production, and low immunogenicity if we are going for the therapeutic application. So this aptamers are preferred because of all these reasons. And I will take you through the process through which we can develop an aptamer sensor for various molecules. And here we are talking about antibiotics as an example. So So these aptamers are nucleic acids that are obtained through in vitro Darwinian evolution process that is called systemic evolution of ligands by exponential enrichment. So what we do is we take a pool of different kind of oligonucleotides, put into a reaction, and where it will bind with the specific target, and nucleotide which is specific for that target is used for diagnosis. So this is the process that happens over here is a very intricate and laborious process that takes several days. And for that, we have a DNA library of varying length. So you can see here, the DNA library that we are using is having two. These two are the constant regions, and the central one, N, is the variable region. So the variable region's length can vary from 20 to 60 nucleotides depending on the size of the target. But here we are working with the 20 nucleotides because we are dealing with the antibiotics. And if you go with the, if you incubate this library with your target molecule, then this is suppose this is the target, this is the DNA, then you incubate it, and then you keep on selecting the specific ones and discarding the unbound ones. So ultimately you will come with the aptamer. So in our laboratory, what we do is we have what we do. This is the, suppose this is the DNA library. The library is first immobilized on iron nanoparticles. This is the iron nanoparticle where library is immobilized. And then we add a specific target, any antibiotic or any small molecules over here. And then it will bind with the, when this small molecule will bind with the aptamer, then it will come into the supernatant, and then you collect the supernatant, the aptamers bound with the target, amplify it, and then again generate the secondary library. Here from double-stranded DNA, you have to separate it out so that you just generate the one single-stranded DNA, and this is the increased pool of oligonucleotides from here to here. Because here we anticipate that whatever sequences we have is a specific for the target. So this process will keep on going, and for this, how your enrichment is going on, we do the real-time PCR. Suppose the CT value in cycle 1 is like high, it will keep on reducing, and if the cycle has become constant, then you have to do counter selection. Upon counter selection with some buffer or proteins of serum and milk, then again there will be a low yield of the DNA, and again with the selection it will keep on improving. So ultimately you will get such kind of thing. And so in antibiotic based aptamer selection, when there is a constant level of DNA recovery in this consecutive 3-4 cycles, then we go for counter selection. Once this you get the pool of nucleotides, then how to get the idea about the unique nucleotide. For that, we clone them and do the restriction diarization, and then we check that a specific unique clones, and then we sequence it. Once you sequence all this, so after sequencing, you will get more than 100, 200 sequences. Then how to come down to few of them? For that, we have devised a newer strategy for aptamer selection, screening of these aptamers. In this, what we do is, this is a lateral flow based assay where we have different tubes where you add first the aptamers, then you label these aptamers with gold nanoparticles. Once these gold nanoparticles labeled aptamers are there, then suppose, see, this is the aptamer and this is the gold labeled aptamer. Then this is the lateral flow strip where we put antibiotic. Suppose here we are working with the oxytetracycline, so you put over an antibiotic, and as and when this antibiotic, this aptamer is put over here, it will go and bind to antibiotic, and you will get a red band. If you get this kind of band, then your aptamer is very unique. So if you have 1 to 5 aptamers with you, within 1 or 2 days, you will be able to screen that which aptamer sequence is very unique to you. And then you can go for development of any kind of lateral flow based assays. So for designing the apta sensor, what we did is this. This is one of the aptamers that we chosen for developing apta sensor. So the extra portion of the aptamer was chopped off to enhance the specificity and sensitivity of the aptamer. And we checked it with the microscale thermophoresis. NST is the instrument that has been used, where we thermophoretic movement of the ligand and target was calculated, measured, and then after measuring the binding affinity, you can see the binding affinity has improved upon truncation of this aptamer extranucleotides. And then we also checked with the CDS spectroscopy that how this secondary structure is formed upon binding with the oxytetracycline. And we observed that this is the characteristic lower this negative and positive peak that it corresponds to 280 and 290. It corresponds to G quadruplex structure. So it confirmed that the oxytetracycline binds to the G quadruplex region and it intercalates. By docking, we have confirmed that it intercalates. We require conjugation of first is making conjugate of gold with the aptamer. So thiol labeled aptamers were conjugated with gold, and you can see on the agarogel the gold nanoparticle. If you run on the agarogel, it will not move; it will remain in the well. Once it gets conjugated, it will move across the gel. So the successful conjugation has been confirmed through this. Apart from that, when you do the TEM analysis of gold nanoparticles, then the size of particle was 11 nanometer, and there is no halo. But in this case, oxytetracycline with a carrier protein. Because oxytetracycline can't be immobilized on nitrocellulose membrane, so we have conjugated with a protein for easy immobilization. And once all the conjugates are prepared, then we connect sample pad, conjugate pad, nitrocellulose membrane, and absorbent pad to develop LFA. On conjugate pad, we put the gold nanoparticle aptamer conjugate, and on nitrocellulose membrane, we have put the oxytetracycline carrier protein conjugate, and on line B, the complementary sequence to aptamer. So if there is no, if there is presence of aptamer OTC, the sample, then we will get faint band at the first line. Second line will be bright. If there is no oxytetracycline, then first line will be bright. You can see here in the real strip will look like if there is no oxytetracycline in the sample, you will get only one line. But if there is a oxytetracycline, then you will get two lines. And we could achieve the specificity also, a higher specificity with the oxytetracycline, and there was no binding with other homologous molecules of the oxytetracycline. We could achieve LOD of 5 nanogram per ml with this. So what we achieved is, if you have a milk sample at home, you can test it, put onto the strip, and you will get this very in less than 10 minutes, you will get the signal that whether the milk is having oxytetracycline or not. So what we have achieved with this is it's a user-friendly and having long shelf life. Within 5 to 10 minutes, you can detect it, and cost-effective. Limit of detection is also within FSSAI limits, and estimated cost is only rupees 25 to 30 rupees as compared to others commercially available antibody-based tests that cost more than 100 and 200 rupees. So with this advantage will be that anyone at home or at the farm level or at the processing plant level, they can test the antibiotic presence of antibiotic in the milk. So in future, we are working for other antibiotics as well, and we will see developing a multiplex sensor for all the animals. So I would like to acknowledge the support provided by our exciting team of students and funding support from Department of Biotechnology, NIAB, and BioRac for supporting our research activities in developing affordable diagnostics. Thank you very much, and now I can take up few questions if there are. Thank you very much.
Thank you very much, sir. If someone has any question, they can put their question in the question answer box. So now we are moving for the next invited talk. I would like to invite Dr. Sameer Srivastava, Department of Biotechnology, Motilal Nehru National Institute of Technology, Allahabad, Uttar Pradesh, for his talk. Dr. Sameer Srivastava is Assistant Professor, Department of Biotechnology, Motilal Nehru National Institute of Technology, Allahabad, Prayagraj. He has expertise in regulation of gene expression, epigenetics, colorectal cancer biology. He is member of several research bodies. He has published 3 books, 6 book chapters, and several research papers in various national and international journal of repute. I request Dr. Samir Srivastava to kindly deliver his talk on transcriptomic profiling of early onset CRCs. Please, sir.
Yeah, so am I audible now?
Yes, sir, you are audible.
Yeah, thank God. Means I wasted lots and lots of time of all
No,
of
sir,
you
no issues, sir.
at that time because of some network issue. That is fine. Never late than better late than never. Okay, so I talk about uh, transcription profiling of early onset CRCs. So they are basically a type of colorectal cancer which basically starts at an early age. Okay, so so you can say that they are diagnosed at an early age. Okay, so these are the contents of my presentation here. So I will briefly talk about the etiology of CRCs, colorectal cancers as a whole, the trends of incidences and deaths worldwide in Indian context, about EOCRCs also, and then their etiology, which is largely unknown at present time, and then the trends of incidences and of EOCRCs across the world and Indian context as well. Then what we did in our this piece of research work to explore the genes and pathways which are generally or may be implicated in EOCRCs, and the and the results which we obtained from them, from that study. Okay, so colorectal cancers basically develops with a small benign polyp, and which progresses to become malignant to form cancer. So in the colon or the rectum region of the intestine, large intestine. Okay, and in terms of most commonly diagnosed cancer, so this stands at third position in both men and women across the world. In Indian context, generally it is considered to be a disease of developed countries or Western world, but nowadays we see that there is an alarming increase in the rate of incidences of colorectal cancers also during these past 2 decades or so. Okay, so out of all colorectal cancers, about 90% CRCs are sporadic in nature. So sporadic means they don't have any means like any inherited disease. Like say there is no cancer, any kind of cancer being diagnosed in any of the family members. Okay, so nothing to do with the genetic predisposition in 90% of the CRC samples, CRC patients, and the genetic factor basically contributes for only about 10% of the total CRC incidences across the world. Okay, and these okay, so now this we can see here the world cancer statistics. So you can see in these two big pie charts where colorectal cancer stands at approximately the third number in terms of incidences and in terms of deaths as well. In fact, second in terms of deaths as well. So 10.2% overall in case of incidences of all cancers, and approximately 9.2% in terms of death for all cancers. Okay, so here in the Indian context, if you see, so this graph represents the here in this left panel you can see, so this color coded map of India is for the year 1990, and this is for the year 2016. So and you can see in this legend also that the darker color shade represents the higher number of incidents per lakh of population. Okay, so you can see that since 1990 to 2016 there is a drastic increase in the rate of incidents per lakh of population in all the states of India. So it is increasing at an alarming rate. Now in terms of combined overall cancers, so in India in both sexes it is approximately at sixth position, and in case of female it is at fourth, and in male it is at sixth position. Okay, age wise distribution also you can see. So majority of cases basically are above 70 years of age. Here you can see in this slide, sorry, in this lower panel, and with the number of years also, as we are progressing, and if you see the trends for the last decades, you can see a very rising trend in India for these for all types of cancer here. Okay. Now, these are just to give you an insight about how the colorectal cancer develops. So it starts with a very small adenoma kind thing in the inner layer of the mucosa in the colon or rectum. Okay, and so that is the stage 0. Then at stage 1, it starts to invade the underlying tissue, the submucosa and mucosa part of the intestines. Okay. So at this stage and at stage 2 also, basically it grows only in a at a means it attains a bigger volume, and it invades the muscularis mucosa and submucosa to a greater depth. Okay, but till stage 2 there is no invasion towards any lymph node, neighboring lymph node. So the lymph nodes are not invaded at this stage. But in stage 3 and stage 4 cancers, these tumors, basically tumor cells, start to migrate from that from the adenoma part towards the lymph nodes, so that they can reach the blood vessels and reach other parts of the body. So they starts to metastasize. Okay, so that is how it progresses, and this is basically the TNM classification of staging of colorectal cancers. So there are different molecular events. In fact, CRC is a very heterogeneous disease. So any tumor in a patient doesn't harbors one single type of cells. So it is basically a clonal propagation of a cell which gets an advantage over the other set of cells which are there in that particular tumor. Okay, so that way the whole tumor as such is a very heterogeneous nature. Heterogeneous in terms of molecular features. Some cells would have a different molecular feature, some cells would have a different molecular feature, and they all are combined in a one tumor sample. In fact, a patient having 2 different tumors at 2 different places may also have different types of tumor heterogeneity within itself. Okay, so because of this heterogeneity, there are certain types of pathways which have basically identified in the etiology of colorectal cancers. Like the first one is the SIN pathway. It is basically the chromosomal instability pathway. So these tumors are basically characterized by widespread alterations in chromosomal numbers, chromosomal deletions, chromosomal gains. Okay, and generally these chromosomal deletions gains are in the portions of chromosome 5q region, 18q region, and 17p region. So these 3 regions are very predominant in case of all SIN cancers, which are generally believed to be developed by SIN molecular pathway. Okay, so the majority of cancers, CRC cases, basically approximately 80 to 85% cases are basically evolved from these SIN pathways only. Okay, and if you are wondering that what kind of genes are there on these 5q and 18q chromosomes, so we can see here in my next slide. So basically very important gene APC, which is again a tumor suppressor gene, then DCC, MDH2, MDH4 at 18q, T50, TP53 at 17p. So these are very important genes for maintaining the normal state of cells in the intestinal epithelium. Okay, so there is seen to be loss of these regions only, which generally triggers the conversion or transformation of normal epithelium to a tumorous epithelium cells. Okay, and the other pathways are SIMP and MSI. So SIMP is again CpG island methylated phenotype and microsatellite instability pathway. So they contribute to approximately 15-20% of all cancer types, and this SIMP cancers, they display pattern of gene hypermethylation. So gene promoter hypermethylation. So there is a set of genes which gets hypermethylated. The promoter of those genes gets hypermethylated, and the expression of those genes goes down. So if this happens in a set of, say, tumor suppressor genes, so it could lead to inactivation of those particular genes, leading to formation of or transformation of normal cells to the cancer cells. Okay. Similarly, so these are called SIMP pathway. The tumors which are developed from gene hypermethylation mostly are called SIMP pathways. Okay, and then MSI. So when there is a change in or alteration in any mismatch repair genes, like say MLH1, MSH2, okay, these kind of genes which are generally involved in repair mechanism. So which it results in basically repairing any alteration of the DNA which happens during replication. So what happens because of the altered function of these type of genes, the microsatellites basically which are there present in the genome, they basically get uh, uh, like wrongly replicated. So their numbers can be increased or decreased, okay, because of mismatch repairing mechanism of the particular cell. So it results in microsatellite instability. So they show the microsatellite region unstable when compared to the normal cells actually. Okay, so these MSIs are characterized by the chromosomes are basically intact. There are no what you call changes in chromosomal structure is basically seen, but they have defects in DNA mismatch repair system. Okay, and because of that, the microsatellite regions of those genome are unstable in nature. Okay, and some class of these tumors are inherited also. Like say, if there is an inheritance of a non-functional mismatch repair gene, okay, so that could lead to cause of tumors which generally mimics the MSI types of tumor. Okay, but MSI types of tumors can be also non-inheritable, means they also can be non-familial also. Okay, so both type occurs in MSI tumors, and approximately 5% of total CRC they are generally inherited in nature, and those inheritance is basically in the form of mutations in the DNA repair genes actually. Okay, so this is what the molecular you can say that how a molecular changes are happening in the epithelium of colon or rectum to transform the normal epithelium into a tumorous in nature. Okay, so at different stages of the progression of tumor, different types of genes or set of genes are basically getting deregulated or altered. Okay, so like say here you can see these genes basically are reported to be hypermethylated at different stages of CRCs. Okay, or alterated at different stages of CRC. Now similarly here also so generally these APC and beta catenin and CDK, these are the first set of genes which are reported to be mutated or altered, and these only basically triggers the formation or transformation of that normal epithelium to a tumorous epithelium, okay, or tumor cells. So and during progression, the cells of these tumor basically again attains different types of advantages because of mutations or alterations of means lower expression or any kind of alteration, epigenetic alteration or genetic alteration in different set of genes results in again enlarging of that particular tumor, or ultimately leading to metastasis of the cells in the nearby lymph nodes. Okay, so at different stages, different types of genes have been identified, having the tumors having different molecular features when compared to the original stage 0 tumors. Okay, so just to highlight that what kind of molecular features or difference in the molecular heterogeneity can occur in tumors. Okay, so this whole tumor represents a accumulated alterations in various kind of genes to reach here. Okay, and these alterations can, and these lists are not limited to only these lists. There can be many other genes which are involved in the alterations. Okay, so the accumulation of these alterations would lead to cancer and then metastasis of CRCs. Now the early onset, so CRC is typically viewed as a disease of an older individual, like say above 60 years or over 70 years. People like you can expect that something of this kind can happen in your colon and tumor rectum. Okay, but EOCRC is a recently, maybe post 2000, you can say that it has been recently identified a type of tumor which is only defined as early onset. Like say the diagnosis of the patients are diagnosed at a younger age, mostly less than 50 years of age. Okay, so that is the only common criteria for defining early onset colorectal cancer. Now they have been also shown to have unique clinical, pathological, genetic and epigenetic characteristic when compared to the late onset colorectal cancer, which is, we can say that a normal colorectal cancer which occurs at a higher age, above 60 or 70 years of age. Okay, now these are generally more of left-sided colon. So this is also very interesting to see that generally they are considered, they are found in the left side of the colon and rectal cancers. Okay, and the biggest disadvantage of EOCRC is that the patients are diagnosed at a very advanced stage, because no one is expecting that a 50-year, less than 50-year-old will have such type of tumors or cancers in their intestine. So no one bothers to check for, because the symptoms are very overlapping with many other types of pathological conditions which can happen during that time. Okay, so no one bothers to do an endoscopy or colonoscopy to check that whether there are any type of tumors or not at an early age of that particular patient. Okay, so because of this, they are diagnosed at a very advanced stage. Okay, so the tumor might be developing, started to develop at the age of 40, but the patient was identified at the age of say 48 or 49. Okay, so that is a big time which can change the whole scenario in the overall survival of that particular patient. So if it would have been identified at an earlier stage, like say immediately, so possibly the patient would have been treated more effectively. Okay, so these early CRCs, so they have been also been identified to have a greater mutational load when compared to the normal CRC patients. Okay, and also they are like say this early CRC, early onset CRC is generally also identified with a patient with familial origin. Like say if the cancer was in the family, so possibly the patient was predisposed to having cancer at some time of their lifetime. Okay, so that type of cancers of familial origin basically have high mutational load. So these EOCRC patients also have tumors have a high mutational load when compared to the cancers which are diagnosed at a later stage. Okay, so identified risk factors, these are all speculations only, no one knows exactly, but speculated risk factors are familial, of course, like most of the familial CRC, they generally have at early onset, they have an early onset. Okay, obesity, dietary change, people are more shifting toward more type of processed food. Okay, so there is a big dietary change happened during the last 3-4 decades in India as well, and across the world is happening for past 50 years possibly. And high sulfur diets, okay, like say processed meats, they have generally high sulfur content. So those are been so these type of associations have been done, and these risk factors have been identified for early onset colorectal cancer. Now Bailey et al. In 15 estimated that there is a rise in of about 124% and 90% colon cancer for patient which will be under 35 years of age by 2030. Okay, so this is one estimation which was done in 2015. You can see here. So 2010, 5% colon cancer between the age of 20 to 34, and 11% at the in the year 2030 for this is approximately 90% increase for early onset. Okay, and plus this 27% also if you consider 50 years of age as a criteria for an early onset. Okay, and here also you can see for rectal cancers it is more than 150% for 50 years of age combined. Okay, so this is a US data, and you can see the cancer incidences after age of 50. So they are declining. They are generally declining. This is for colon and this is for rectal cancer. So above age of 50 years, the cancers are late onset cancer CRCs are declining, and early onset CRCs are, you can see from approximately 1995 or something, we have a rising trend in colon cancer also and in rectal cancer also. So maybe because of change in food habits and many other environmental conditions, these things are being shown up now. In Indian context, again, the situation is alarming. So like say you can see here, in case of normal CRC, means it was reported that the cancer CRC cases of total what you call CRCs were approximately 18% in 2004, and it increased to 27% in 2012-14. So in approximately 10 to 12 years, they have increased approximately 50%. Okay, but this trend is declining in Western countries because you can see here in the previous slide, age, the normal CRC which happens at an age of 50 and or above. So it is declining. So this declining is universally present in all countries actually, all Western countries. But in India, both the things are rising. Like say the early onset CRC is also rising, and the late onset CRC is also in the rising trend. So both are rising, but we aim to identify important genes and pathways implicated in EOCRC etiology and explore their biomarker potential. So this was our aim of this study, and so we'll show that how we proceeded in this for this study. So for this study, we performed an integrated bioinformatic analysis. We collected some tumor samples from RGCI New Delhi, and then these samples were so the demographic details of these patients were also collected by RGCI, and we performed genetic vital isolation, RNA, DNA. Then we performed certain mutation analysis, which was generally known to be implicated in normal CRC patients. Okay, just to know that what kind of heterogeneity these tumors have. Okay, and then we chose 4 different samples for NGS analysis. So we performed transcriptomic analysis for 4 ERC samples and 1 normal sample, and then we performed the whole bioinformatic pipeline, identified certain genes and pathways which were having which might be playing a role in etiology of EOCRCs, and possibly could be used as a biomarker, prognostic or diagnostic biomarker in future as well. Okay, and then we validated all our studies using this methodology. So this is the cohort. So we are calling this as a discovery cohort because this cohort or these 4 samples were sent for sequencing.
Excuse me, sorry to interrupt. I request you to kindly summarize.
Okay.
The talk.
Okay. So these were the 4 samples which basically we sent for sequencing. This is the bioinformatics pipeline which was followed, and this is the NGS statistics. Approximately 6 to 7 GB data was obtained for this transcriptome analysis for every sample, and then approximately 16,000 genes were identified in total in all the samples, with 4 to 5,000 genes up and down regulated in each samples, and approximately 9 to 10,000 genes were neutral in expression as well. Okay, identified as neutral in expression as well. So before proceeding for analysis, we tested the top upregulated and top downregulated genes which were present in the list, to check that the bioinformatic data which we have obtained for expression analysis is correct or not. So we found that the upregulated genes were upregulated generally, and the downregulated genes were downregulated overall. Okay, except few exceptions like TCL1 and AM129C. In all these 4 samples. So this is the discovery cohort only, the 4 samples which we sent for sequencing. Then for all those genes, the overexpressed and underexpressed genes, we plot a graph, the volcano plot. So you can see that whatever here in red, every dot is a gene which is plotted on this scale, log2 fold scale, and we considered -2 log2 fold -2 and +2 to be overexpressed and underexpressed. So anything which was above +2 was considered overexpressed. Anything which was below -2 was considered underexpressed. So we did it for all the 4 samples. Then the number which we got here basically was tested for any common genes which were there present in all the samples. So we found 95 genes were common in all these 4 samples. Okay, so further we proceeded with these 95 genes. So out of these 95 genes, we plotted plotted a heat map for the expression of these genes. So you can see that these are the top 20 genes. So these are the more purple color represent the overexpressed, and the green color represents the underexpressed genes. Okay, so the whole 95 genes were used to construct PPI network on a string database. So in PPI network, we identified that there were pro-inflammatory pathways, Wnt gene pathway, MAP kinase pathway, many different kinase pathways which are generally associated in cancer tumorigenesis or carcinogenesis were identified in this PPI network. Then this PPI network was used to create a co-expression network to identify pathways which were generally identified as, like say, mostly involved in pro-inflammatory mechanism. So generally it is also thought that these early onset CRCs are result of some inflammatory response within the individual. Okay, so because of this, possibly we were seeing mostly the pathways which were enriched were of pro-inflammatory nature, like say beta B cell receptor signaling pathway, activation of immune response, cytokine biosynthetic processes, okay, interleukin biosynthetic process. So then the whole 95 gene set of genes which were common to all, they were used, and the string network file was used in Cytoscape using CytoHubba plugin to identify the hub genes. So the hub genes are basically the genes which are having the highest number of networks. Okay, so and these are color coded. Is the more red basically is the highest ranked gene. Okay, and we did it with 2 algorithms. One was density of maximum neighborhood component, and the other was maximal clique centrality. So these 2 algorithms were used in the CytoHubba plugin, using the CytoHubba plugin to generate 2 networks of hub genes. So in both the hub genes, we took the top 20 ranked hub genes, and we found that 17 hub genes were common in both the algorithms. So we took these 17 and we tried to perform some pathway analysis to see that what kind of pathways are generally involved in these genes. So you can see here, so mostly the enriched pathways where cytokine receptor interaction, MAP signaling pathway, PI3K, KT signaling pathway, RAS signaling pathways. These are very important in terms of cancer causing, and this represents the pro-inflammatory response or pro-inflammatory conditions prevalent during the tumor formation. Okay, so these were the enriched pathways which for those 17 genes, and then later after this study we tried to explore the biomarker potential of all these 17 genes. So we tested this for the overall survival on the GPA server, and we did it for all 17 genes, and we found that out of 17, 5 had a significant correlation with the expression of these genes in the CRC samples. Okay, so you can see here, so this CXCL3 met IL-1B, TNS1 and IL-1A. So they were the most significantly associated with the expression of these genes in the tumor and normal samples. Okay, so based on this, we further try to identify the expression pattern on the already existing database on the tumor samples and normal samples. So we did it for stage-wise analysis, and we found that 2 of them MET and TNS1. So they showed a significant association with stage-wise expression, differential expression of these genes when compared to the normal. Okay, so for us, out of 17, these 2 genes were the most significant significantly associated with the survival analysis, overall survival, and the differential expression in stage-wise in different stages of the tumors. Okay, so these 2 can be used for you can say diagnosis or early diagnosis, or say prognostic value to estimate overall survival in EOCRC tumor samples or EOCRC patients. So we tested these 2 for genes for their expression in the discovery cohort, which was sent for sequencing, and the other samples which we had for EOCRC samples. We had total 9 samples. Actually, we had total 15 samples for EOCRC, but out of 15, 6 were having some inherited means they could be they had some kind of cancer in their family. So we didn't use those samples. We used only those samples where no history of cancer was there in the family and with the patient. Okay, so total we had 9 samples. Out of those 9, 4 were tested and by NGS analysis, and the results were tested on this validation cohort of 5 EOCRC samples, and we found the same, that they were overexpressed as well. Okay, so these 2 genes, so we generally are proposing that these 2 genes could be a potential biomarker for EOCRC screening and diagnosis, but their clinical utility is required to be validated on a larger cohort of EOCRC samples. So unless we identify these genes on a larger cohort, we won't be able to comment anything on about its use as a prognostic or diagnostic marker in future. Okay, so these are the references, not limited to this page, but I have just shown only the important ones, and these are the acknowledgements. So this study was funded by DBT India. Some bioinformatics pipeline was basically done at the Biotechnology. Tumor collection was done at Rajiv Gandhi Cancer Center, and I also acknowledge all my students who were part of this project. So thank you for your attention. So I'm ready to take questions, and before that, I again want to thank Professor Tomar for giving me a chance to present my work, and all the session chairperson, Professor Ramkumar Mishra from ISER Bhopal, and Dr. Pankaj Suman from NIB Nabi Hyderabad. Thank you.
Thank you very much, sir. Thank you very much for providing your insight on regulation of gene expression, epigenetics, and colorectal cancer biology. Thank you so much.
Thank you.
Now, I would like to request chairperson of the session, Dr. Ramkumar Mishra, to kindly sum up the session.
Thank you, Dr. Pallavi. Am I audible?
Yes, sir, you're audible.
So thank you all the speakers, Dr. Samir for insightful things on the colorectal cancer, and Dr. Pankaj, he gave very nice idea about how aptamers could be used for antibiotic detection in livestock and milk products and so on, what he discussed. And it was really interesting to have these presentation from all the speakers. I really thank all of them. And especially, I would like to thank Pallavi also for keeping up time and introducing all of us. And I'm sure next couple of sessions which are remaining for the today will be a good boost for everybody who is attending the session. So with these words, I would like to conclude this session. Thank you, Pallavi.
Thank you, Dr. Mishra, for concluding the session.
Thank you,
Glad enough that you have taken so much time and you really spared the time for us. And we are really indebted to you. And also, I'm thankful to all the ITs, presenters, Dr. Samir. You have tried twice, I think, in first session you could not.
Yeah, sorry about that.
Shows your zeal and enthusiasm, that how much you are dedicated for this particular work. That you came again and you presented it. Really so nice, sir. And others also in this particular session that was chaired by Dr. Ramkumar Mishra and co-chaired by Dr. Pankaj Sumanji. Sumanji, thank you very much. And the lectures by Gopalji also in this particular session. So thank you very much. Thank you all. Namaskar.
Thank you, sir. Thank you very much.
Thank you, sir.
Thank you very much. On behalf of AIB AUMP, I would like to thank all the eminent persons who added glory to this session. Now, this is the time to close this session, and as per the schedule, we will continue with the next session, that is toxicology, with the same link. So should I start?
Yeah, go ahead, please. Welcome, Dr. Nalini Srivastava ma'am and Dr. Ravikant Singh ji for this session.
Yes, sir. Good morning. Thank you, sir, for
Very good morning to you, sir.
Me. Yeah, I'm here.
It's good afternoon.
Good afternoon.
Okay, so Pallavi, proceed with the proceedings of this particular session on toxicology.
Thank you, sir. The toxicology is a scientific discipline overlapping with biology, chemistry, pharmacology, and medicine that involves study of the adverse effect of chemical substances on the living organisms, and the practice of diagnosing and treating exposure to toxins and toxicants. The relationship between dose and its effect on the exposed organism is of high significance in the toxicology. On behalf of MIT Institute of Biotechnology, MIT University, Madhya Pradesh, Gwalior, I, Dr. Pallavi Singh Chauhan, Assistant Professor AIB, heartily welcome Chairperson of this session, Professor Dr. Nalini Srivastava, Professor, School of Studies in Biochemistry, Jivaji University, Gwalior, Co-chairperson of the session, Professor Dr. Rajesh Singh Tomar, Director, MIT Institute of Biotechnology, Dean Life Sciences, MIT University, Gwalior, and to all the invitees, presenters, and attendees. Now, I would like to invite Professor Dr. Nalini Srivastava, Professor, School of Studies in Biochemistry, Jivaji University, for her talk. Professor Nalini Srivastava did her PhD in 1985 from Kanpur University, Kanpur, Uttar Pradesh. Professor Srivastava has published several papers in Journal of National and International Repute. She has completed projects with DST, UGC, and MPCST. She has supervised a number of PhD and MPhil students. Currently, a number of students are working under her able guidance. I request Professor Dr. Nalini Srivastava to kindly deliver her talk on organophosphate pesticides from neurotoxicity to cell death. Please, ma'am.
And please set the angle of your camera. It is a little bit up. Yeah, now it's okay. Welcome, ma'am. Good afternoon.
Okay. So first of all, I would like to extend my gratefulness to the authorities of MIT University, particularly Professor Tomar, for inviting me for this talk and giving me the chance to interact with this eminent scientists who have delivered their talk. Few of them are alumni from our department only, like Professor R.S. Tomar from ICER Bhopal. he used to, he's the alumni of our department. Okay. So I'm very proud of these star students. Okay, now I'll start my talk, which is on organophosphate pesticides from neurotoxicity to cell death. This work was carried out by PhD students of my laboratory in
please make
School
it
of
full screen.
It is full screen.
Hello.
It's not visible to us as full. No problem.
Yes,
Wait.
yeah, you need not introduce
Please
like
carry
that.
on.
Yeah, go ahead.
Is it not visible?
Yes.
Just a minute. Let me. Yeah, actually, there's a block in the side. It is coming. How to get rid of this block? I don't know. Anyway, so the pesticides are nowadays a very burning problem. This the state is that now we cannot leave them and nor we can continue with them, because the green revolution is possible because of the use of pesticides. Because besides this, there are many more applications and benefits of pesticides, which we have seen in the recent past, that is in about last 70-80 years. Okay. These are the toxic chemicals, but have been added purposefully in the environment for controlling the pest in the agriculture sector, in the health sector, in veterinary practices, even in the homes and offices also nowadays they are being used.
मनोज या महेंद्र है क्या? भेज दीजिए किसी को डॉक्यूमेंट ले जाने के लिए.
The green revolution, as well as the eradication of malaria, which is a big health related milestone, which was achieved only by the use of certain pesticides. But the problem is that the pesticides are not selective. They kill the pest, but they harm the non-pest, like beneficial organisms as well as the human beings also. Okay. So if we just assess the benefits and the risk of these pesticides, they are both for agriculture sector, health sector, and for the control of vector-borne diseases. They are being used in food preservation, horticulture, as well as in domestic applications as termiticides, etc. But the risks are even more worse, like human poisoning, toxic to the ecosystem, contaminate soil, water, etc. They bring about cattle poisoning and kill the beneficial insects also. In this way, harming the whole ecosystem. And the problem with the pesticide is that they are persistent and remain in the soil even after the application, and on the food grains and fruits, etc., even after the application. And many of them are having the tendency to bioaccumulate and biomagnify, and therefore become more hazardous and more dangerous for the, more toxic for the organisms, including the human beings who consume these contaminated food or water or air they inhale, etc. Okay. But this industry is hello, yes.
I'm just interrupting you. Good afternoon, ma'am. First of all,
Good afternoon.
ma'am, your slide is not moving actually.
It's not. Should I share again?
No, ma'am. Just click on that particular icon, which is just parallel to the 60%. Just click on this icon, ma'am. In your screen, there is a 60%. Just with this 60%, there is an icon of the computer like. Just click on it, and after that, it will be full screen.
But I am not able to see it. There is no 60% icon. I just have it. New share live. I am sharing once again. Stop. Okay,
Stop sharing and then reshare it, ma'am.
Okay.
Maybe problem will be solved.
Is it okay now? Full screen, the screen is visible?
Now it's okay, ma'am. If you just browse the next slide, then it will be clear. Ma'am, can you go to next slide, please?
Ma'am, there is a short key F5. Can you press on it? Function key F5.
F5. Okay. Yes. You ho gaya, but nothing is happening here.
No problem, ma'am. You continue with this, ma'am. No, not a problem.
Okay. Despite their beneficial effect, the toxic effects have been gradually identified, and in some countries and in some states, the most toxic chemicals have been banned or came in restricted use. But still, the production and the use is increasing just to feed the ever-growing population worldwide, and therefore this pesticide industry, it is being dominated by dominated by 9 multinational countries from USA to Western Europe, and it is having a 50 billion pounds pesticide which are conventionally being used in areas of agricultural land. So India is one of the most dynamic generic pesticide manufacturer, which produces about 60 technical grade pesticides being manufactured here, and there are about 120. And I think now it is more clear. Okay, slide ज़्यादा अच्छी दिख रही है. More than 500 pesticide formulators are spread all over the country, where Indian pesticide industry is ranked second in Asia after and 12th globally. China is the most, the first in using the pesticide and producing the pesticide also. These are the list of countries which are using the pesticide, although the India uses very less amount of pesticide in comparison to the developed countries like China and USA, but still the problem of the pesticide toxicity and the pesticide poisoning are more serious here because of the ignorance of the farmers, and they are not being well trained. Okay. This is the consumption of worldwide consumption of pesticides, production of pesticides worldwide, and the Indian pesticide market was 214 billion in 2019 and expected to reach to 314 billion rupees in the coming financial year 2024. Okay. The increase of 8.1% compound growth rate is expected due to several factors like growing farmers' awareness, farmers' prosperity, inclining demands for organic food, increased focus on R&D, etc. Okay. This is Indian crop protection market split. Insecticides are being highest. They constitute highest percentage of pesticide which are being produced and used in India, followed by fungicides and herbicides. These are the few industries which are mainly involved in manufacture of different type of pesticides. The major classes, organochlorines, were the first and the foremost developed important pesticides and developed first, but they tend to be very hazardous because they are persistent and tend to bioaccumulate in environment as well as in the food samples, as well as they being lipid soluble. They have been found in the fat tissues of the human beings also. Okay. So looking at the problems and the toxicities of organochlorine pesticide, the next organophosphates are being developed, which have been actually developed by Germany. For the first time, they were developed by Germany as a toxic agent in World War, organophosphate compounds like sarin, soman, etc. But later, less toxic compounds, they have also been developed by derivatization or by changing the structure of these organophosphate compounds, and they were used as a pesticides. Okay. So organophosphate pesticides have replaced now most of the organochlorines, And since they are being heavily used, the poisoning of pesticide poisoning cases are most of them are due to organophosphates. The other two classes are carbamide pesticides and pyrethroids. So our work is mainly concentrated on organophosphorus pesticides, which are derivatives of phosphoric acid. They constitute a very important class of compounds with diverse activities, including pesticide, warfare agent, metabolic poisons, etc. The basic and the most foremost mechanism of their action is they are neurotoxic in nature, and they exert their neurotoxicity by inhibiting cholinesterases, particularly acetylcholinesterase at neuromuscular junctions and then neural tissues. They degrade easily, and thus they don't have any problem of bioaccumulation or persistence. Okay. The symptoms of poisoning are muscarinic effect, nicotinic effect, and CNS effect. They cause, they inhibit irreversibly acetylcholinesterase, and this cause hyperstimulation of neurons, and therefore they lead to seizures, coma, etc. And finally, respiratory failure, and death is due to respiratory arrest. Okay. Primary mechanism of action is they are anticholinesterase, any substance that inhibits enzyme cholinesterase, which is responsible for the breakdown of the neurotransmitter. Acetylcholine at the nerve synapses is anticholinesterase compound. This includes drugs, nerve gases, and insecticides. This causes a buildup of acetylcholine, means accumulation and pileup of acetyl neurotransmitter within the synapses, leading to disruption of the nerve and muscle function. In vertebrates, these agents often cause death by paralyzing the respiratory muscles. Okay. This is the pictorial representation of mechanism of action of these pesticides and how acetylcholine works. Acetylcholinesterase enzyme, this hydrolyzes the neurotransmitter after it has performed its function to acetyl and choline is recycled for the fresh synthesis of acetylcholine. Okay. If this enzyme is inhibited, the neurotransmitter is accumulated in the synapse and cause hyperstimulation, finally respiratory arrest. Okay. Besides being anticholinesterase compounds, organophosphate compounds, they also bring about disbalance or imbalance in the oxidant and antioxidant of the body and cause oxidative stress. The oxidative stress, there are many reasons of the oxidative stress. The chemical exposure or the exposure with xenobiotics, pollutants, including pesticide, is a very common one. The formation of reactive oxygen species, including superoxide radical, hydroxy radical, H2O2, etc. This is increased. Besides this, reactive, the concentration of reactive nitrogen species is also increased. Although the reactive oxygen species are quite beneficial for the body, they are involved in signaling, phagocytosis, etc. But when they are excessively produced, they start damaging the vital macromolecules of the cell and lead to severe diseases and disorders. Okay. There is a strong defense system against combating the toxic effects of these reactive oxygen species, composed of or made up of enzymatic and non-enzymatic antioxidant machinery, which works very potently. But when the balance between the production and scavenging is lost, oxidative stress is the result. Okay. This is causing oxidative stress. When it is called, it causes diseases of almost all organs, from heart, skin, kidney, joints, lungs, brains, etc. And they start damaging the vital macromolecules, which starts accumulating and finally leading to the dysfunction and different type of effects. Okay. This is the main molecule which is affected by these pesticides or by reactive oxygen species produced by pesticide exposure is DNA, although lipid and proteins are equally susceptible. But when the damage of the DNA is less, there is elaborate repair systems also available in the cell for repairing this. But again, when the damage exceeds repair capacity, then the damaged DNA starts accumulating. So this unrepaired or the damaged DNA starts accumulating. So cells accumulated with with accumulated DNA damage products, they are either and goes to senescence or apoptotic cell, or they may go to development of the cancer also. So very serious health consequences on accumulation of DNA damage product. This is how the pesticides damage DNA. They cause all type of possible damages like a scission of single strands or double strand breaks. They cause base modification as well as the DNA crosslinks, DNA protein crosslinks, etc. And there are a number of reasons of these type of damages. Okay. Causes by they are carcinogenic due to pollutants and pesticides, causing carcinogenesis. There is elaborate repair mechanism, and most commonly, when there is a base damage, the base excision repair is the most common mechanism, which depends mainly on the different type of specific glycosylases, which remove the damaged base and replace them with the fresh, correct base. Okay. So these pesticides, organophosphate pesticides, different type of pesticide we have studied. They are responsible for generation of oxidative stress. They are found both in vivo and in vitro systems using animal model or cell culture. Studies carried out in our department on different OP pesticides, including commonly used pesticides all over the globe or in this region particularly, chlorpyrifos, malathion, parathion, monocrotophos, etc. Quinolphos, they have shown these pesticides cause increase in the levels of reactive oxygen species and reactive nitrogen species, both in vivo and in vitro systems, which has been checked by looking at the levels of hydrogen peroxide, increased levels of hydrogen peroxide or superoxide anion. There was increase in the nitrate and nitrite levels also. Besides this, they cause imbalance in the redox status by decreasing the level of GSH, NADPH, etc., and increasing the oxidized product, that is GSSG and NADP or NAD. Okay. So the ratio is, it leans towards the oxidized form, that is the imbalance or the redox imbalance is caused. Besides this, these pesticides have been shown to cause the inhibition of enzymatic and antioxidant defense machinery also by inhibiting the main enzymes of antioxidant defense, that is catalase, superoxide dismutase, glutathione peroxidase, etc. Okay. These increased reactive oxygen species bring about an increase in the lipid peroxidation, that is peroxidative damage of membrane lipids, particularly the plasma membrane as well as the mitochondrial membrane. Besides this, oxidation of proteins, which includes important enzymes, transporters, carriers, and different type of signaling molecules, which lost their function on their oxidation and have different type of consequences. The total antioxidant capacity is also decreased on exposure with these pesticides, and the resultant is oxidative stress. Okay. Besides this, these pesticides on DNA cause single strand breaks, double strand breaks, damage to purine and pyrimidine bases, as well as they are inducing protein DNA crosslinks. Okay. They are, besides this, these pesticides, they cause chromosomal damage, as increase in the sister chromatase exchange, chromosomal aberration, micronuclei formation, etc. This all have been studied in vivo and in vitro system in our laboratory. Okay. So this causes OP pesticide exposure caused inhibition of enzymes of base excision repair and down regulation of these enzymes also. increased DNA damage, increased DNA damage and decreased DNA damage repair capacity. Okay. These are the levels of enzymes in liver and brain, and this is the level of their expression. Both have been downregulated on exposure with these pesticides. Okay. This is another enzyme. These are the different glycosylases involved in the repair of damaged DNA. These all are activities decreased as well as their synthesis is also decreased, that is, they are downregulated. Okay. So this cause increase in micronuclei in PCE showed clastogenic and enugenic potential also. This is the exposure cause change in number, aneuploidy, change in size of peripheral blood lymphocytes. And the most important deleterious effect of these pesticides on the mitochondrial membrane, which causes the mitochondria is mitochondrial dysfunction. It is mainly responsible for generation of reactive oxygen species and vice versa. Excessive reactive oxygen species also cause mitochondrial dysfunction, and pesticide, the main source of reactive oxygen species in mitochondria are inhibition of mitochondrial complexes. Okay. Mitochondrial dysfunction also causes endoplasmic reticulum stress. This endoplasmic reticulum stress is a very important thing and have a grave consequences, because endoplasmic reticulum are performing very important functions of protein folding, protein glycosylation, protein modification, etc. As well as they are having the property of storing the calcium ion, segregating the calcium ion, which are required for muscle contraction. Okay. So when there is an endoplasmic reticulum stress, which leads to mitochondrial stress, and mitochondrial stress causes depolarization of mitochondrial membrane. And again, when the membrane is depolarized, the energy generation is disturbed. That is, there is a reduced level of ATP on mitochondrial depolarization, mitochondrial membrane depolarization. This also causes a release of cytochrome C, and there are two factors like B cell lymphocyte 2, BCL2, and BCL2 associated X factor, that is BAX. These are the main mitochondrial, main regulator of mitochondrial integrity. Both are very important, but are having opposite functions. Like BCL2, it is suppressor of apoptosis, while BAX is promoter of apoptosis. These are involved in cell death, but have their opposite functions. Okay. These proteins, they influence mitochondrial integrity and cytochrome C release, which is an important signal for apoptosis. And we have observed that cytochrome C release, mitochondrial dysfunction are observed in rat peripheral blood lymphocytes on exposure with these organophosphate pesticides. Okay. This in vitro exposure of quinol, phos and chloropyrifos caused loss of membrane, membrane asymmetry. It caused increased activities of caspase 9 and caspase 3, which are the apoptotic caspases responsible for induction of apoptosis. And these caspase activated DNAs cause DNA fragmentation. So characteristic DNA fragmentation or the lateral pattern is observed in lymphocytes on exposure with these pesticides. Besides this increased generation of ROS, reactive oxygen species, which is observed by flow cytometry, they correlates with these damages. Means when this level of ROS is increased, the damages are the changes which has been shown above. They have been more significant. Okay. This is DNA fragmentation pattern at different doses and at different time exposure of different time intervals. So this is how the apoptosis is caused. This mitochondria when the integrity of mitochondrial membrane is damaged, then mitochondrial membrane potential is damaged, which causes release in cytochrome C, which is an indicator of apoptosis, and responsible for that. Sorry. This brings about induction of caspase 9, and caspase 9 activity is increased, which this is a initiator caspase, and this activates the executioner caspase, which is caspase 3 in this case, and finally results in apoptosis. Okay. Besides this, the role of BCL2 and BAX, which we have just now discussed, this is also there in the mitochondrial function, mitochondrial damage theory of apoptosis. So pesticides disturb all these network and finally lead to cell death by apoptosis. Okay. So on the basis of the results obtained, we can say that oxidative stress is the major toxicity, mutagenicity, carcinogenicity of these pesticides is mainly due to oxidative stress. Excessive reactive oxygen cause mitochondrial damage by altering membrane potential, cytochrome C release, activation of caspases, and caspases induce thing is that we have to find out some strategies to reduce this stress, mitochondrial stress or oxidative stress, to combat the toxicity of these pesticides. So new strategies have to be designed for safe and planned use of these hazardous chemicals. And since it is oxidative stress, the role of antioxidant is very prominent, and we have observed that the antioxidant vitamin supplementation as well as the natural antioxidant flavonoids, etc., they have beneficial and anti-toxicity potential in this case. Okay. So this is all about the work which was carried out in our lab using different type of systems and different pesticides. And these are the research students who are carried out their work. They have been awarded their PhDs on pesticide toxicity only, using different type of pesticide and taking different aspects. The work was supported by financial assistance from different funding agencies like DST, FIS grant, individual projects, UGC, MP Council of Science and Technology, Ayush projects, etc. So with this, I thank you for patiently hearing and ready for any query if you have.
Thank you very much, ma'am. If someone has any query, then they can put their queries in the question and answer box.
So if there's no query, then thank you very much.
Thank you very much, ma'am, for providing your deep insight on the involvement of oxidative stress and toxicity of organophosphate pesticides. Thank you so much. Now,
Thank you, ma'am, and as you requested, if there is any urgent task, I think I can take care.
I can be disconnected for some time and then we rejoin.
Okay, ma'am. Thank you so much. Thank you.
Now, I would like to invite Professor Dr. Ravi Kant Singh, Amity Institute of Biotechnology, Amity University, Chhattisgarh, Raipur, for his talk. Professor Dr. Ravi Kant Singh is Dean and Director of Amity University, Raipur. He has done his PhD from Indian Institute of Technology, Roorkee, and MTech from IET, Lucknow. His research interest areas are nanobiotechnology, bioinformatics, animal biotechnology, enzyme engineering, and environmental biotechnology. He has several research publications in various national and international journal of repute and ongoing 3 projects. I request Professor Dr. Ravi Kant Singh to kindly deliver his talk on DNA damage as a biomarker for exposure, the COMET assay perspective. Please, sir.
Yeah, okay. Thank you, ma'am, for inviting me. First of all, I would like to thank Professor R.S. Tomarji for inviting me and giving me an opportunity to deliver a talk. in a MET family. I am at MET Raipur, you are in MET Gwalior, we all are in a family, in the same family. It's a pleasure to deliver a talk in a family, sir. And now I am going to start sharing my screen. I think we'll go for the complete screen sharing. And, my screen is visible. Full screen.
Yes,
Yes, sir. Yes, sir. It's visible.
Yes, sir.
It's visible.
Now, I heard the previous lecture in this session toxicology about the pesticide, how the pesticide will affect the neurotoxicity and how from the cell death also. Right now, we are on the farmers who are dealing with the pesticides. The sperm level, sperm count of that farmer will also going to be reduced. And our PhD scholars are working on it. And will not talk about in my lecture today about how the sperm count reduces to the farmers who are dealing with the chemical pesticide. That's why government is also promoting the biopesticides also. Today, I am going to deliver my talk on the DNA damage as a biomarker for any kind of exposure, any kind of mutations which can cause gene mutation. And this gene mutation can be early detected by the comet assay. Comet assay is the most reliable, most sensitive technique for determining the determining the DNA damage. And with the help of DNA damage, we can find out the level of disease problem, to what level this disease is now, at the early stage or at the final stage. Because in the cancer problem, the main problem with the cancer is early detection is not possible. The beautiful thing with this comet assay technique is we can identify the level of DNA damage at the early stage, so that we can say that after 10 years, this kind of people may cause this type of genetic disease, and this kind of cancer may arise. Okay, so this is the beauty of this comet assay. I will not talk about the advanced technique and tool, because every kind of students as well as faculties are there. They are not here to listen my expertise in that particular area, but we'll find out, we'll explain how the comet assay is working. Now, about the DNA, any kind of disease, when the disease came, for the disease, the physical and chemical factors are there. Due to that, chemical, physical, and sometimes biological factors are also involved, which cause disease. And disease can be identified or monitored with the help of the DNA damage. So the sensitive detection of the DNA damage as well as the repair can be done by this comet assay tool. You can identify the aging, clinical medicine, molecular epidemiology, toxicology, and genetic toxicology, all kind of, with the help of these works, we can easily detect it out. The thing is that about the DNA damage, the DNA can be damaged at the single strand or in the double strand also. With the help of comet assay, we can identify the single strand DNA damage also, because most of the techniques are identify the double strand DNA damage. But with the help of the comet, the single strand DNA damage also, and what will be the protocol we'll discuss about later. And everybody know about the DNA, how the DNA damage, what kind of factors are there, what kind of agents are there which are responsible for that. And that kind of DNA damage can be assessed, can be assessed by a lot of techniques are there: Ames test, chromosomal aberration, micronuclei, fluorescence in situ hybridization phase, and a lot of other techniques are there. But in this talk, we'll basically talk about the comet assay only. And the important thing is that when we are talking about the any kind of disease, in disease we are focusing on the genetic diseases also. And in the genetic disease, what level of DNA damage is there, we'll talk about. The single gene disorders are also there, polygene disorders are also there, X-linked recessive disorders and chromosomal disorders are also there. All kind of disorders can be assessed, can be find out, analyzed with the help of this comet assay tool and technique. The important thing is that the apoptosis, apoptosis or level of the apoptosis can also be measured with the help of the DNA damage. Sometimes the human occupational problems are also there. This kind of disease we are facing due to my lifestyle. So the lifestyle, how much lifestyle affects the DNA damage, that kind of assessment can also be done with the help of this technique and tool. Sometimes some peoples are vegetarians, some are non-vegetarians also. How the vegetarians, what level of DNA damage occurs in the vegetarians as well as the non-vegetarians, this kind of studies can also be done with the help of the commensura. The risk assessments are there, genetic disorders and genetic toxicology. Lot of diseases arises, but the major important thing is that how we can find out the toxicant, we can find out the level of toxicity, and this level of toxicity is very much important. That problem is at the primary stage or the final stage. So here this risk assessment is also required. Without assessing the risk, we cannot protect ourselves with this thing. So this chemical, this pollutant is responsible for the DNA damage, we find out. So that kind of risk assessment is very much important. And nowadays, the important existing tests and techniques and tools are available, or nowadays Comet assay is frequently using for the assessment of DNA damage. The important thing is this is very cost effective and less time consuming also. Sensitive, reliable also. So these are the beauty of this technique. That's why I am here to talk and discuss about this thing. The Comet assay can be assessed in vitro level study as well as in vivo level study also. So I worked in ITRC in year of 2001 and 2002, long back, under the supervision of Dr. Alok Dhawan. Right now, he's a director CVMR SGPGI. I always guided by Dr. Dhawan sir, and I worked. Then after, initially I started supervising my students. We can do with the help of this Comet assay, we can identify the sperm's count level, quality of sperm, how with the help of Comet assay we can assess. We are maintaining and preparing the protocol for that. After completing and finding some findings in the next, I think in third international conference at this same platform, we'll talk about how with the help of Comet assay, we can assess pesticides, how the pesticides can reduce the sperm level, sperm count. This biomonitoring, I think everybody understands the biomonitoring is very much important for the public health and all. And if we are talking about the this thing, biomarker, the biomarkers having three types: dose biomarker, effect biomarker, and susceptibility biomarkers. And all kind of biomarkers are nowadays using in this Comet assay. This Comet assay is well accepted biomonitoring tool to examine the effect of dietary, lifestyle, environmental, occupational exposure on the level of DNA damage in the human cell. As well as some other animals studies, we can find out the dose effect. So, and human cell, if we are talking about the human cell, all kind of, in plant also, the Comet assay is nowadays using. In animal, in bacteria also, the Comet assay can be used for assessing the DNA level of damage. Now, this Comet assay, I talked a lot of things about this. The Comet assay is a rapid visual. The good thing is that the visual level of DNA damage, we can see that this is DNA. And how much percentage of DNA is damaged, we can see, we can identify, we can quantify. The quantification is important, the visualization is important. But in a test tube, if we are taking, saying that this point level of DNA damage is found by this titration and this kind of experimental method. But here in this Comet assay, the level of DNA damage, we can visualize it also. So due to visual characteristics of the DNA Comet assay, it becomes more sensitive. And the qualitative and quantitative assessment of DNA damage can be possible only and only by this Comet assay method. And sometimes everybody talking about if the DNA is going to be damaged, the repair mechanism is there. but everybody's have not a similar kind of repair mechanism. So that repair mechanism can also be assessed and can also be visualized with the help of this Comet assay tool. It is useful as a screen for a genotoxicity and anti-genotoxic activity of any kind of chemicals in the vitro level as well as the vivo level. And it's a very inexpensive method. Not big instruments are required, fluorescent microscopes, one computer with software. And in next slide, we'll talk about how much cost is involved. But cost I not estimated, but this is not so costly in nature. It's rapid also. Within 3 to 4 days, we can find out the level of DNA damage. Within 1 day also, we can do it. Many cell types from different tissues can also be assessed for the DNA damage. And good thing is that the human studies are possible, monitoring environmental exposure or change in sensitivity at the DNA damage. But the thing is that if a good and beautiful techniques came in and everybody is working on it, but first time who invented this technique? First time in the year in 1984, one of scientists, Ostling and Johnson in 1984 identified this technique, but the final single cell gel electrophoresis technique, this is SCGE, Comet assay, was invented by Dr. N.P. Singh. He was a professor in University of Washington Medical Science. So I met with N.P. Singh, is a very gentleman. When he came in Dr. Dhawan sir ki lab mein aaye the, then I visited. We spent 1-2 days with N.P. Singh. Diana Anderson, I met with Diana Anderson also. He was also working. But the first man who invented this tool technique is Dr. N.P. Singh. And what was the philosophy? The philosophy was DNA is a super coiled structure on a strand break, could be stressed up by electrophoresis. And if DNA can be stressed by electrophoresis, because DNA negatively charged is moved towards the cathode, positively charged part in the electrophoresis, when the DNA is damaged, the damaged part of DNA will move. And the movement level, tail length, percentage DNA, and olive tail movement, these are the parameters by which we can assess the DNA damage. So the good thing is that he given information about the neutral as well as the alkaline assay. In neutral assay is used for measuring double strand break, while alkaline measures both double and single strand break. In 1988, he developed this tool technique, and everybody is completely accepted. No modifications came after Dr. N.P. Singh. So he's a great man, great scientist, very good human being also. And it's my pleasure to meet at that time due to Dr. Dhawan sir's when I was working in that lab. When we are talking about the protocol, because a lot of people after this talk wants to pursue their work in study research in Comet assay. When I discuss about the research, I found this thing, I found that thing, that will be not much more beneficial for everyone. But the protocol understanding is very much important. And now we are developing a protocol for this farmers sperm count also, with the help of Comet assay. After development in the next we'll talk about. Comet assay protocol, the first thing is step 1, from blood or other isolated cell to the cell embedded in gel and deposit on slide. We are developing a sandwich. We are taking slide, then we take blood, and then low melting point agarose, normal melting point agarose. We'll discuss in later. We're making a sandwich. Sandwich to the first level contains agarose, upper level also contains agarose, and the middle level contains blood and agarose. And this will be protected with that, and all the different steps. After with electrophoresis, we can see that the second step is first the formation of the slide, first, second, and third layer. Second is alkaline lysis. Then the lysis came at neutral pH. Lysis came, then the electrophorese will start, because double strand DNA must be converted into single strand DNA. And at the single strand level, we can identify the level of DNA damage. The DNA denaturation, unbinding, and migration will come in this electrophoresis. Alkaline assays, membrane and soluble component removal will be done in this step 2. Then step 4 is neutralization, fixation, and DNA staining. Ethidium bromide will be used for DNA staining. And when DNA staining done, then we'll put this in the fluorescence microscope, which is connected with the CCD camera in a computer, और वो हम आपको एक अच्छी सी comet वहाँ पर दिखाई देती है. Everybody know comet. आसमान में भी comet को हमने देखा है. So how the level of DNA damage can be assessed with the help of the comet level, we can analyze with the image analysis. And for that KOMET comet, we worked on comet 3.1. But nowadays the software comet 7 is there. KOMET, there is a spelling change. KOMET, this is the name of the software 7 nowadays using for the image analysis. I think, yeah. First agarose pro-coated slide. This is normal melting point agarose, NMA. Then after will contain the second layer blood plus low melting point agarose, and third layer without blood filled low melting point agarose. This is then after this, the slide becomes prepared. Slide prepared in 3 layers. For the production of blood, we maintain first layer, क्योंकि यदि हम slide लेते हैं तो some oil contents are there. First of all, you must remove the oil. Ethanol में dip करके उसको burn करेंगे. Then after we maintain a low melting point agarose level, और उसको एक बेड बना दिया. फिर उसके बाद हमने DNA रखा with LMP, low melting point agarose. And then after उसको protect करने के लिए, then after without blood will layered third low melting point agarose. This is now this step gives the prepared slide. And when the nucleus and cells are there, the lysis came at the high salt concentration, and the lysed cell are now at the alkaline condition. If pH is greater than 13, the single strand DNA will come in the picture. And if the pH is greater than 9, then double strand DNA will be there in this. So that's why we are talking about single cell gel electroporation, which were developed by Dr. N.P. Singh sir. So after that, electrophoresis come, and that electrophoresis should be carried out at 4 degrees Celsius temperature. उसको हम फ्रीज के अंदर रखते थे, रेफ्रिजरेटर के अंदर रखते थे, नॉर्मल 4 डिग्री टेम्परेचर पे, and 20 to 30 minute electrophoresis will be carried out inside the refrigerator. Because during the electrophoresis, some energy can release, and that energy can damage the DNA also. So we cannot assess now, because we are minutely monitoring the DNA damage. So that any kind of protocol in any kind of step during the process, the DNA should not be damaged. So that's why all the electrophoresis should be carried out inside the refrigerator. And then this part is damaged DNA. जितना ज़्यादा लंबी टेल होगी उतना ज़्यादा DNA damage है. और जो नहीं move किया, it means this is a strong nuclear protected, and that DNA is not moving towards the cathode. Neutralizing trace buffer will be used for hairpin loops, and then staining will be carried out with the ethidium bromide. And due to this ethidium bromide staining, we can visualize in the software. We worked in 3.1 also, we worked in 5.0 also. Nowadays, the software 5.7.0 is also there for the image processing of the DNA damage. So my purpose of today's talk is everybody must understand how in COMET the procedure is going on, and what kind of research we carried, and what kind of research other professors, other scientists carrying will also quoting. Preparation of reagents are required. Phosphate buffer saline. Low melting point agarose, if we are preparing low melting point agarose, will took 50 microliter of 10 microliter of blood. 5 microliter we can also take. So 5 microliter blood is required to identify the DNA damage, and plus same amount of PBS, phosphate buffer saline, plus double amount of 1% low melting point agarose. This is the combination. So these are the steps we'll standardize. At that moment, we standardize that protocol with me, Dr. Alok Pandey, Dr. Mahima Bajpayee. We all work together. And at that time, we developed that protocol under the able guidance of Dr. Alok Dhawan, and we start working on it. After that, I came in administration and in teaching, and Dr. Alok Pandey continuously working with Dr. Alok Dhawan. Right now, he's a principal scientist at ITRC. So these are the preparation reasons are there. We'll not talk about more and more. Less time is there. Lunch break. Everybody is waiting for the lunch also. तो ये सब सारे stains कैसे बनाएंगे? If we are preparing staining solution, how staining solution will be prepared? This alkaline electrophoresis solution में क्या होगा? EDTA होगा, क्या होगा? All the things, and every solution must be prepared in distilled water. We all know about it. Methodology, I already talked about how methodology work and how the DNA level of DNA damage. This, I think, electrophoresis, the temperature is not mentioned here. This electrophoresis must be carried out at 4 degrees Celsius. And molten low melting point agarose at 37 degrees Celsius. This right preparation is there. After that, this kind of image will come in our screen, which fluorescence microscope is connected with us. This one thing, I think, camera and microscope connected with computer, and this kind of image will come. Here, this contains head DNA. So that software all measures. कुछ नहीं करना है slide में. All things will be done by the software, and the statistical approach will come and find out the all kind of results. So the head percent, head DNA, tail DNA, as well as tail length in middle से लेंगे हम, वो अपने आप measure करता है. Tail length, and this olive tail movement is also there in that. Olive tail movement. Olive tail movement is, I think, this olive tail movement is tail DNA into tail length. This olive tail movement is the most sensitive way for measuring level of DNA damages. This thing is very much important. We'll talk about. This is the advantages. We'll already take. It's an invasive technique, non-invasive technique. It's a cost-effective also. It's a flexible also. Easy applications are there. Increased sensitivity are there. A small amount of cells are needed for measuring the DNA damage. Less time consuming, less expensive. A lot of things are giving a positive thing for the comet assay. The applications of comet assay in plant tissue culture also, in bacteria also, the apoptosis, level of apoptosis can also be determined. But the beautiful and the important thing with the cancer treatment, during the chemotherapy, most of the patients facing a lot of difficulty. And nowadays, palliative care is also everybody is focusing. Because if the chemotherapy came, a lot of pain will come. So for the family support will also help in DNA repair mechanism. So that kind of things are there. This level of repair can also be assessed by the comet assay. The comet assay will help in early detection of any kind of genetic disease, as well as during the treatment also, if that treatment is going in the positive direction or in the negative direction. The thing is that when any genetic disease come in the third level, it is very difficult to cure. And if for the cure the chemotherapy or radiotherapy comes, it's very painful. For that, the palliative care is also important. And that palliative care will help at the DNA damage. At that kind of palliative care effect on DNA damage can also be assessed in comet assay. उसके लिए हमें क्वेश्चनर बनाने पड़ेंगे, बहुत सारी चीजें. Any kind of level of study you can do, but for the research, my today's talk will help more and more listeners can do in this comet assay. detection, DNA repair, genetic toxicology, and everything will help for this method. So these are the now after the application will work in these studies at that moment in 2001, 2002, and after that human monitoring, fish, plants, earth bomb, CHO cell line we worked on, human lymphocytes we worked. That kind of studies and good publication came at that time also, and I was also acknowledged at that moment. Then at that moment, what kind of suggestions given by the studies is non-vegetarian and vegetarians. We worked smoker, non-smokers. In far village areas, the women are working on the biomass fuel. Now government, Honorable Prime Minister has provided LPG to all the farmers, all the villagers, so that protect their life level of DNA damage. We studied how the biofuel, biomass cooking is dangerous. Women with exposure to the biomass fuel show greater damage than those using LPG. So these things are there. Rickshaw puller, petrol pump pe jo workers petrol fill karte hain, that can also affect. Anybody, if anyone with pesticide, at that moment we assessed DNA damage. And at the early stage also, by studying occupational behavior, if anybody is a smoker, if anybody is non-smoker, how the smoking affect, male, female, males में DNA damage ज़्यादा होता है कि females में ज़्यादा होता है. That kind of studies can also be assessed and worked also in this. बड़ा interesting है, उस समय बड़ा अच्छा लगा था, मैं काम भी किया था. तो ये Comet ऐसे softwares हैं, that software gives this information. And वही मैं बोल रहा था कि these are the parameter, olive tail movement, OTP having arbitrary unit, tail length. This OTM is tail length into the intensity of the cell. तो यदि आप OTM measure करते हैं, olive tail movement, if you are going to measure, that contains tail length also, and that also contains the intensity of the cell. The most sensitive parameter of the DNA damage assessment is OTM. तो software तो सब दे देगा, head DNA दे देगा, tail DNA दे देगा. Tail length will also give in micrometer. So tail length or head and tail DNA is in percentage also, and OTM can be measured by this method. मैंने कंपनी का नाम भी बता दिया. This is Kinetic Imaging, Liverpool, UK. This software was there at that moment. Right now, Comet 7 was also manufactured by the same company, and we used fluorescence microscope. And fluorescent microscope, तो यदि अच्छी कंपनी का होगा तो it gives good results, and that is connected with the charge coupled device CCD camera. तो already हमने इसमें आपको बात कर लिया. Then magnification may be set at x400. Everything is important. We are talking about advanced things, but the basic fundamentals, if you do not know, we cannot initiate your research in that particular area. This is very much important. You can establish research lab which contains Comet as a facility and will work. I think if any establishment is there at Gwalior or at Raipur, at Noida, so nowadays we are sending the sample to ITRC and the results can come from this, but we can establish in our labs also. So that kind of olive tail movement and all the concentration and that kind of images we have taken, and that we captured, that kind of images have not copied from anywhere else. Apoptosis, if any kind of apoptosis is there, then this kind of image will come. तो इस तरह की image यदि आती है तो ऐसा सर ने तो बताया था, कॉमेट बड़ा सुंदर सुंदर आता है, तो ऐसा image क्यों आ रहा है? तो that indicates the apoptosis size. तो वो एक चीजों को समझने की जरूरत है. यदि हमारे पास ये सब सारी चीजें नहीं हैं तो we can also identify visual scoring also. Will not talk about more and more because time is now over. Silver staining is also there. I already submitted the abstract which contains the summary, and in this summary I talked about we worked with the villagers, we worked with the petrol pump workers, we worked studies for the smokers, non-smokers, vegetarians, non-vegetarians, male and female, and nowadays we are working on the with the farmers who are dealing with the pesticides. So these are the things, these are the main references I have collected from this. And thanks for inviting me again. And it's a pleasure to give a talk in a family, and we are in a same family. Thank you, Tomar sir, for inviting me. Thanks a lot.
Thank you very much, sir. Thank you very much for providing your deep insight on DNA damage as a biomarker for the exposure. Thank you so much.
Thank you.
Now,
any question? I am ready for reply. Any kind of.
Yes, sir. Actually, there is no question in the comment and question and answer box. So
time भी हो गया है, सबको भूख लगी होगी. ठीक, कोई बात नहीं.
Doctor, thank you so much for such a nice presentation by you.
Thank you, sir.
I was attending to you continuously, and I have seen that it's such a nice approach you have taken with regard to this particular problem that is prevailing there in the regard to DNA damage and repairing, especially. I would like to thank you for this particular topic that we have chosen, DNA damage as a biomarker for exposure, because there are so many exposures which are taking place. Occupational health hazards are also there, and also the kind of study which you have done with regard to the comparison for the vegetarian and non-vegetarian, smokers and non-smokers. Such a nice study, sir. I think it can have a long-lasting impact on the human society, because nowadays a lot many things are happening. When you tell somebody that you take this kind of diet or this diet is good for your health, people always argue. They say that now you will tell me that why should I eat? I think we scientists can send a silent message into the society.
Sure, sir. Sure, we'll work together, sir. We'll work together. We'll meet and we'll come at your campus.
Yes,
also
sir.
physically.
So it was actually, I tell you, there are so many things which are getting re-verified from the scientists. Like our Rishi Munis, they were not taking non-vegetarian diets. So,
exactly.
So this is being shown by research also that non-vegetarian show higher degree of DNA damage. That's a reconfirmation, I think, that has already been shared by our sages and gurus. It's really good work, sir. So
Okay,
thank you
sir.
very much.
Thank you. Thank
And
you,
we
sir.
shall meet, we shall meet personally, sir, because I think
Sure.
it's long. We have not met each other.
Exactly. आपका नाम ही सुना है, बस आपके दर्शन नहीं हुए हैं.
आपको भी मुझे कहाँ हुए, मैं भी आपसे कहाँ मिला हूँ.
जी.
So nice of you, sir. Thank you very much, sir.
Thank you, sir. Thank you. Namaskar, sir. Namaskar, everyone. Namaskar, everyone.
Thank you very much. On behalf of AIB AU MP, I would like to thank all the eminent persons who added glory to this session. Now, this is the time to close this session, and as per the schedule, we will continue with the next session. This is industrial and microbial technology with the same link.
Due to lack of time, we are not going for lunch break, and we are going for the next session. That is the last technical session. That is the industrial and microbial technology, and it shall be chaired by Dr. Mithilesh Jaiswal and co-chaired by Dr. Brajesh Varse, along with some presentations, invited talks by both of them, and also Dr. Pankaj Rai and Abhishek Mathur. So, Dr. Pallavi, you can continue with this.
Thank you very much, sir.
I would like to unshare first. I think some
Please.
issue is there.
Dr. Jaiswal, welcome and good afternoon. Namaskar. Mute है आप, आवाज नहीं आ रही आपकी. जसवाल जी, please unmute yourself.
Good afternoon, sir.
Good afternoon. कैसे हो?
बस बहुत बढ़िया, sir. Thank you so much for inviting me once again.
Once again, we are meeting, and that too on the online platform. I think online platform has become very popular nowadays, no?
Yes, sir. Yes, sir. Yes, sir. It is necessary also due to current scenario. But all we are familiar with this type of the platform.
Right.
And this is the right way to communicate with each other.
So nice, sir. I think Dr. Pallavi,
yes,
you
sir.
can proceed.
The industrial microbiology is a branch of applied microbiology in which microorganisms are used for the production of important substances, such as antibiotic, food product, enzymes, amino acid, vaccines, and fine chemicals. On behalf of MIT University, Madhya Pradesh, Gwalior, I, Dr. Pallavi Singh Chauhan, Assistant Professor AIB, heartily welcome Chairperson of this session, Dr. Mithilesh Jaiswal, Biotechnology Lab, Research and Development Division, Tropolite Food Private Limited, Gwalior. Co-chairperson of this session, Dr. Brajesh Varse, International Center for Genetic Engineering and Biotechnology, New Delhi, and to all the invitees, presenters, and attendees. Now, I would like to invite Dr. Mithilesh Jaiswal, Biotechnology Lab Research and Development Division, Tropolite Food Private Limited, Gwalior for his talk. Dr. Mithilesh Jaiswal has obtained PhD in microbiology from the University of Jabalpur, India. He is currently working on probiotic cultures and development of synbiotic product for various human and animal applications. His PhD research was based on mycoherbicidal potential of different fungi against the biological control of hazardous weed like Parthenium. His area of interest is probiotics, starter culture, and biofertilizers. He has published various papers in peer-reviewed journals. I request Dr. Mithilesh Jaiswal to kindly deliver his talk on the role of probiotic microbes in health benefits and career buildup. Please, sir.
Thank you so much, Dr. Pallavi, for nice words for me. Very good afternoon, everyone. It's a great honor for me to be here and just wants to share my what we have achieved in last 10 years in the field of probiotics, being a scientist in the company Tropolite Biosciences Private Limited, Gwalior. So I have a lot of things which I want to share with you, but due to shortage of time, I would like to introduce all these points very briefly, because we are having a very prominent speakers after me, like Dr. Brajesh Barshe, Professor Pankaj Rai, and Dr. Abhishek Mathur. Friends, as you know that my screen is visible to everyone, Dr. Pallavi?
Yes, sir, it is visible. Can you please maximize it?
That's good. So I just yeah.
Dr. Jaiswal, can you make it full screen?
So as you know that is it clear? I think it is full screen right now.
They're still in the same mode, the tiles mode.
Hello.
Is there a network issue, Dr. Jaiswal? Can you hear us?
I think so. There is some network problem.
Unmute करिए Dr. Mithilesh. I think mute हैं आप. Yes, it is visible now. Just go to slideshow. Yes, it is good. Now it's full screen, sir.
Great. Okay. Also, this technical. I just want to tell you that the interaction between the microbes and new, it's long back. We have developed a native related to the microbes, as because you know that 2 years we have seen a lot. No, so I'm here to tell you that the microbe is really the enemy for human being, but we can able to take a lot of things from the microbes. So just I'm here to tell you that how these microbes are very much helpful for our bright career in the field of the microbiology and biotechnology. So let's start. Friends, as you know that the microbes are our real friend for life, as because the microbes, if organism can able to give everything those you want, but you have to know how to ask them. That's why the different in the biotechnology and microbiology industries developed to work on the different important aspects of the microorganisms. As you know that the many of the antimicrobials we rely on today to treat the various infectious disease were first of all developed by the natural products from the microorganisms that kills or inhibit the growth of the microbes. Among all, the lactic acid bacteria are used to produce the many functional food like cheese, caffeine, and other health supplements. Not only the bacteria, but also the yeast nowadays is very important to make the bread, beer, and wine. And the acetic acid bacteria are used in a traditional manufacturing of the vinegar and many more like that. But above all the bacteria and yeast, today's are used for the various health benefits for human well-being. They are regarded as they are known as the probiotics. So let's try to understand what are the probiotics. Are good bacteria that actually live in your body. There are several different types of the definitions given by different agencies, but the most important and widely accepted definitions which is given by the World Health Organization is that the probiotics are defined as live microorganisms which when administered in adequate amount confer health benefits on the host. That is the two important aspects associated with the probiotic. That is, there is some form that is fixed, and all the probiotics is designed for some health benefits for the human as well as the animal both. Health benefits have mainly demonstrated for the specific probiotic strain of the following genera. Each and every probiotic strains are recommended from the different important health function like Lactobacillus, Bifidobacterium, Saccharomyces, Enterococcus, Streptococcus, Pediococcus, Stock and Bacillus, and many more. Recent advancement in biotechnology have made the success in the development of the different probiotic food products and as well as the different types of health diseases and risk from the pathogenic microorganisms. The three important terms apart from the probiotics, that is the prebiotic, synbiotic, and postbiotic, is directly associated with the probiotics. Try to understand the prebiotics. Prebiotics are generally, these are the dietary fibers products which may be a food for the probiotic bacteria, which not only provides the food but also support the growth and activities of the probiotic bacteria inside the gut. They are very common like fructooligosaccharides and inulin. They alone can able to provide the health benefits on the host. Second, the most important term, that is the synbiotics. Synbiotics generally a combination of the prebiotics and probiotics bacteria which provide also the health benefits better than the probiotics alone, because it always works in the combination of pre and probiotics. All the pre and probiotic supplements which are present in the market comes under the category of the synbiotics. The third and important point is the term is the postbiotics. Postbiotics are the byproducts or secondary metabolites which are secreted by the various probiotics bacteria inside the gut or at the time of the fermentations. They are very much beneficial for the health, as you know that the like organic acids and bacteriocins. By producing the bacteriocins, they can able to induce the growth of the pathogens because it has the antimicrobial property. Not only the bacteriocins, but they can also able to release the various important enzymes which helps in the digestion and various activities. So there are different types of microorganisms nowadays are available which works as a probiotic. Some are various some are common species we can see over here like Lactobacillus acidophilus, Plantrum, Lactobacillus rhamnosus, Lactobacillus brevis, and many more. Some important species of Bifidobacterium like Bifidobacterium animalis, Bifidobacterium bifidum longum, and infantis. These are comes under the vegetative groups, but the spore forming bacteria are also very, very common, particularly if you're talking about the products designed for animal health care, like the Bacillus coagulans, Bacillus clausii, Bacillus mesentericus, and Bacillus licheniformis. But these are not only specific for the animal health care, but they can also very much effective in the case of human, like Bacillus coagulans, Bacillus clausii, and Bacillus subtilis. Some other strains we can see like Streptococcus salivarius, Streptococcus thermophilus, Streptococcus faecalis, Lactococcus lactis, and many more. So nowadays, a lot of bacteria belonging to different genera are exploited for preparation of different formulation for human being as well as the animal. So as you know that all the microorganisms not considered as a probiotics, as because the probiotic bacteria have some specific or very common characteristics. That's why after those microorganisms which possess all these category, only they can consider as the probiotics. Let's find out some important characteristics like probiotic bacteria able to survive the passage through the digestive system because pH is very, very low at there, and they have to pass from that digestive system as because to ensure the maximum delivery of probiotic bacteria in the intestine, because all the important role performed by the probiotic bacteria is only in the large or as well as the small intestine. They should able to attach the intestinal epithelia and colonize over there to replace the pathogenic bacteria and survive over there for the long time for the better performance. They can able to maintain the good viability. Only the viable cells can able to perform the important function, because non-viable cells cannot able to provide any health beneficial. So they can able to utilize the nutrient and substrate from the normal diet, as because they don't require any specific nutrient for the growth. Whatever the diet taken by the body or host, they can able to take their nutrition from there. It is very important characteristic of the probiotic that should be not non-pathogenic and non-toxic. Only good bacteria or health beneficial bacteria can regarded as the probiotics. They can capable of exerting a beneficial effect on the host, so that they can consider as the probiotics. Stability of desired characteristics, whatever the characteristics they have, should be stable during the processes, storage, transportation. It is very necessary until reach to the consumer end. Probiotics also have important characteristics like anti-inflammatory, anti-mutagenic, and immunostimulatory, so that they can enhance the immune system of our body. Potency, as I'm talking, potency and viability are both correlated. So potency not less than 10 million CFU per gram for the better performance of the probiotic microorganism. It should be resistant to gastric acid and bile acid. That's why they can able to ensure their presence in the large intestine or small intestine. Antimicrobial activity against the potential pathogenic bacteria and fungi, so they can able to induce the growth of the pathogens in the gut. It should not be antibiotic resistance. It is a very, very important characteristics of the probiotic bacteria. That means it should not be resistance to any antibiotic which are available in the market. And the most last but most important point is that probiotic microorganisms should not be GMO. That means genetically modified organism should not consider as a probiotics. So you can see nowadays probiotic microorganisms are very much popular, as because people are very much aware about their health and they rely on the only health beneficial products. Probiotic today's are exploited in two segments. One is the functional food, another is the health supplements. If you're talking about the functional food ingredient, in the global market, more than 500 probiotic food products have been launched in the past few years. And the number of the probiotic foods in the market is continuously increasing due to the high demand of all these products by the consumer. The probiotic food produced by the fermentation of the fruit and vegetable, cereals and meat products are gaining attention by the consumer as well as those from the scientific world. as because they are very much health beneficial for the host. Nowadays, probiotics are commercially found in the fruit juice, milk, shore milk, oat-based product, and ice cream to meet the demand of the all type of the people, like for the children, for the adult, for the woman, everyone. So all these products are easily available by the fortified with the probiotics microorganisms in the market. For the preparation of the probiotic products, the probiotic microorganisms are artificially introduced in the food, which is available in the form of the freeze drying powder. In nutraceuticals and pharmaceuticals, both several probiotics and symbiotic supplements are available in the different forms of the various form of product for the different health benefits. So let's find out apart from the functional food, how the probiotic microorganism beneficial for the health improvement. As you know that in the last current scenario during the pandemic, everyone suffers from effect of the COVID-19 due to the low immunity. So everyone's to mitigate the risk of the novel coronavirus, people are consuming a healthy and balanced diet. Probiotic are healthy yeast and bacteria. Everyone nowadays know that aid in overall development of the digestive system that keeps the gut healthy. Probiotics are also involved in inhibiting the pathogens growth. This is the very or prime characteristic of the pathogenic probiotic bacteria by synthesizing the antimicrobial compound. They are also responsible to produce the lactase enzyme, which are responsible to ferment the lactose and prevent the human body from the lactose intolerance, modifying the gut pH. In the low pH, pathogenic microorganism cannot able to survive over there. Also in the low pH, several minerals which is not easily digested by the body can easily digest in the low pH, like calcium, challenging the binding of pathogens and stimulating immunomodulatory cells. So there are several health benefits of the probiotics. One of the most significant aspect of the probiotic is that they have proven to be safe, cheap, and capable of handling with the microbial infection. That's why the WHO regarded as the grass of all these probiotics, which are recognized as safe, to be the next most significant system when regular given antibiotic are made ineffective by antibiotic resistance. It is also called microbial interference therapy. This is very important characteristics. So probiotics and symbiotic both are very much effective to control the different infection. Nowadays, one of the important products are available in the market. I think most of the people Enterozermina, which contains only the Bacillus clausii, and it is very much effective against the diarrhea, particularly for the small infant. Able to cure the diarrhea, either it is associated with the pathogens, either it is associated with the antibiotics. It can able to cure the infant of diarrhea within 24 hours. That's why probiotics are reported to play the crucial role in the prevention of diarrhea and control over the antibiotic treatment. Several microorganisms, Saccharomyces boulardii and Bifidobacterium species, Lactobacillus reuteri, Lactobacillus rhamnosus GZ are generally for this. But one of the important microorganism, Helicobacter pylori, which for different problems like peptic ulcers, gastric cancer, and gastritis. Lactobacillus salivus is one of the most important probiotic organisms. They can able to suppress the growth of the Helicobacter pylori. It is also reported that the possible role of the probiotic microbes in different components to cure the inflammatory bowel diseases. IBS is very common, and it is reported by the several researchers that all the several probiotics are like Lactobacillus rhamnosus and Lactobacillus acidophilus. Both are very much effective against the IBS problem. Not only this, even the prebiotic also alone can able to induce the effect of the several pathogens like Salmonella infection. So if we're talking about the different health benefits of the probiotic, it will take a lot of time, because starting from the diarrhea up to the cancer, nowadays probiotics are exploited. You can see the different of the probiotic in the health problems. It can very much effective against the improvement in lactose intolerance by producing the lactase enzyme, control inflammatory bowel diseases, control of irritable bowel syndrome, avoid food allergy in infants, so many more. I'm not going to elaborate all these things over here, because we have very little time to explain. The important point is that you should know about the application of the probiotic supplements. It is very important. So I would highlight some important application over here. The viable count of the probiotic microbes and concentration of prebiotic may be affected due to several factors. As you know that in our daily life, we are come in contact of that several abiotic factors which influence this ratio of the probiotics bacteria. We can say that it creates the imbalance between the good bacteria and the bad bacteria. So it is very necessary to take the probiotic bacteria from the outsource in the form of different health supplement product like sachets, capsule, dry syrup, whatever you can want to take. Even you can take the functional food if you don't like take any health supplement. The proposed health benefits of the probiotics can only be attained when the product possesses necessary minimal viable microorganism. I have already told you in my previous slides that only the viable cells can able to provide you the health benefits, not the non-viable. So the very standard dose, if we're talking about the dose of the 100 million up to the 1 billion CFO probiotic microorganism, is very much crucial to attain the probiotic action in the human being. It is reported by the several researchers. All the probiotics work effectively. Most of the people asked me several times, so what is the time to take the probiotic? I have mentioned over here that all probiotics work effectively if taken the after meal, when the stomach acid is lowest, to ensure the maximum delivery to the small intestine where they reproduce rapidly. So it is very necessary to take the probiotic just after the meal. As I already told you that probiotics not only provide health benefits to the human being, but they also provide several important health beneficial roles in the field of the animal. Like it helps in strengthening the immunity of the animals. It also helps in the 100% feed utilization. It reduces the pathogenic bacteria in the gut, enhance the growth and body weight. It also responsible to improve the FCR, and it also overall provide the farmer to get the higher profit. So you should also know about the mandatory parameters and regulatory which are directly associated with the probiotic strain. Joint Food Authority of Organization and WHO working group on drafting a guideline for the evaluation of the probiotic in food. I'm not going in detail, just want to highlight some important things. That strain identity is an important link, that whatever the claim we are going to put on my product, the probiotic strain should be mentioned over there. Or the probiotic strains should be identified at the molecular level, like 16S RNA sequencing. It is recommended that this genotypic technique should be combined with the phenotypic test for the reconfirmation of the probiotic microorganism. Patterns generated from the fermentation of a range of sugar, like all the biochemical tests against the several sugar, should be performed before going for the molecular identification. It is also recommended by the committee that all strain be deposited in an internationally recognized culture for the reconfirmation over there. So different in vitro tests also suggested by the organization to perform before consider the microorganism as a probiotic, as because all the lactobacillus, all the rhamnosus, all the bifidobacterium are not considered as a probiotic unless until they should passes the important test in vitro test, like resistance to gastric acid, bile acid resistance, adherence to the mucus or human epithelial cells, cell line, antimicrobial activity against the potential pathogenic bacteria, ability to reduce the pathogens adhesion to the surface, bile salt hydrose activity, resistance to spermicides. So all these things in vitro tests are very much important before claiming any microbiome as a probiotics. Some regulatory, there are some recommendation also available over there for the health claim and labeling. Like currently, in most countries, only general health claim are allowed, as because for the drug medical health, you should have sufficient clinical research. The working describe on the label, like gene survival numbers of the each probiotic strain at the end of the shelf life. The suggested serving size must deliver the effective dose of the probiotic related to the health claim. Proper storage condition, corporate contact details of the consumer information. All these information should be necessary to mention on the label of the health supplements product, which is considered as the symbiotics. I'm just would like to talk about the genre of Tropilite. Tropilite Biosciences Private Limited, a Davos Group company, in which we have performed all the experiments which are related to the probiotics from lab scale up to the commercialization of the product. Tropilite Biosciences is mainly into the manufacture of starter culture for the dairy industries, as well as the probiotic cultures for all categories like vegetative cells, spore forming bacteria, and the probiotic yeast. Tropilite has a microbial culture, huge microbial culture bank of the more than 30 strains right now we have. We manufacture symbiotic products as well, which are basically a combination of the probiotics and prebiotics. Now you know. Tropilite Food Private Biosciences has expertise in manufacturing of the probiotic cultures and is specialization in the blending technique. And today we have more than 250 customers in Pan India and abroad also. You can see in the bacillus, we have more than 11 strains of the bacillus, 5 strains of the bifidobacterium, and many more, some other microorganisms which comes under the in our R&D and manufacturing activities, we focus on the isolation, improvement in stability and development of the different probiotic strains. We also focus on the standardization of upstream, downstream, and freeze drying, which is an important part of our manufacturing and mass production of these bacteria. Development of new formulation and delivery system of the pre and probiotic of new application as per demand of our customer from the pharmaceuticals and nutraceutical industries. So for the isolation and development of new probiotic strains, we have performed the isolation from the human body, dairy products, fermented fruits, isolation from mother's milk, and isolation from soil. You can see the different colonies at these pictures at the time of experiment. So after the completion of isolation, we have performed the identification of these strains. For the identification, we have classified all the activities in the 4 categories for the morphological characteristics, identification on the basis of biochemical analysis, identification on the basis of 16S RNA sequencing, and finally, we have performed the whole genome sequencing to understand also perform all the necessary or mandatory parameters suggested by the WHO, like stability test against the temperature and pH tolerance, bile and acid tolerance, antimicrobial antagonistic potential against the pathogens, non-antibiotic resistance, non-toxic and safe for the human consumption. You can see over here, this is the stability test we have performed against the Lactobacillus acidophilus. This is the pH tolerance activity we have performed. This is the gastric juice, intestinal juice, and bile salt tolerance activity we have performed. This is the antimicrobial activity and antagonistic activity we have performed against the Lactobacillus acidophilus. Similarly, we have performed all the parameters for all the 33 strains. This is the antibiotic resistance. So for the mass production of these bacteria, we have performed all these activities like media standardization, process optimization, mass production at laboratory scale before going to the pilot plant, mass production at commercial scale, and standardize the process. The most important point is the critical point for the manufacturing of the probiotic microorganism is that freeze drying, because we don't have the spray drying. Spray drying facility is only feasible for the spore forming bacteria, not for the vegetative cells. So we have only the freeze drying facility, and we perform both vegetative cells as well as the spore forming bacteria under the freeze drying, which is based on the process of the sublimation. So these are our daily routine process. We have performed the mass production under the fermentation. After the completion of the mass production, we have checked their quality analysis from our center quality control. And after the completion of all the activities, we have performed the freeze drying, make the semi biomass of the microorganism into the powder mass. And after taking that microorganism, we perform the different formulation and the form of the finished product. Product development by using the probiotic we have developed it from till now for the different customers belonging to nutraceuticals for sector. For activities for developing our continuous addition effort led to the yield and robust product. We have developed scientifically selected for the acid and bile resistance. All the probiotics which we have are non-genetically modified, and from all are considered as the probiotics which we have. So you can see in this function for our abroad customers like improve. You can see a lot of abnormalities created in the body after the COVID infections. So we have tested and for this product Improflax against these abnormalities, and people will get relief very soon by taking these products. So these are the different products we have developed in our company for the animal health application, not for all the segment, either it is poultry, for the cattle, for the aquacultures. So at the end, I would like to highlight some important point which is directly related to the business opportunity and career build-up through the probiotics for the youth and young and dynamic students. So the use of probiotics and drug supplement is widely accepted in India nowadays. Intake of the probiotic of food supplement is yet to gain the significance. So we can say that the probiotics, drug products and health supplements products, the market is very big and growing day by day. People today are more informed and prefer to take the preventive path rather than adopting a curative approach. Due to above 2 points, given this approach, the future of the probiotic industry in India, which is still at a very nascent stage, looking very much promising in coming few years. If we're talking about the Indian probiotics market in 2021, the subsequent research in December 2022 capture the following from the probiotic market in India. We can see 140 brand variants are available in the market related to the probiotic product, in which 130 probiotic supplements are available. Only 7 kombucha products and 2 probiotic cosmetics and 1 probiotic juice are available. So you can see how the health supplements products, which is directly related to the pre and probiotic combination, are very much available in the market. And if you see the average rating of all this product, it is 4.16 out of the 5 for the probiotic products. So how people rely on such products for their health benefits, you can see over here. If we talk about market size of the probiotic culture and blend, you can see the Indian probiotic market is expected to grow at a compound annual growth rate 13.56, which is very good CAGR over the forecast period to reach a market size of the 56 million in coming 2025, which is approx 6,000 more above 6,700 crore. According to the report published by the Fortune Business Insight, the global probiotic industries is expected to showcase considerable growth by reaching 94.48 billion by 2027, while exhibiting a CAGR 7.9% between the 2020 and 2027. So you can see how much big market of probiotic are available in the both domestic as well as the global market. All the market lies in 4 segments only: nutraceuticals, finished good, comes under pharmaceuticals, as well as the nutraceuticals, and animal feed supplements. I have analyzed the probiotic market from 3 divisions, and we can say that pharmaceuticals, nutraceuticals, and animal feed supplements, there are different manufacturers like PDC, traders, third-party manufacturers, and manufacturers. You can build your career in all these category if you want to work under the probiotic segment. So, a lot of people, students ask me what type of career we can see in the field of the probiotics. A lot of opportunity are available. There is, I can't able to say only some name of what you can do in the field of probiotics, but a lot of opportunity. Some I would like to suggest over here that you can become an entrepreneur as market size is growing day by day, and a lot of facility also provided by the government nowadays for the new startup program. So just create a new idea which is directly related to the probiotics, and you can start your own business. Just work for yourself. To become a scientist in a probiotic segment, as because a lots of opportunity are still available in the R&D for the new product development. You can also become a technical expert or consultant, as because those people who are working in the field of probiotic still they are suffering from some scientific evidence and knowledge for their formulation design. You can provide them information, and you can earn a lot of money. To become a market expert and business head, this is very important point, as because still now a very few people are available who can able to perform the business in the field of the probiotics, as because the market is very crucial and based on the technical knowledge. So everyone not able to perform the good in the field of the business. Only as all you people belongs to the field of science, you can easily perform very well in the field of the market expert and business head. To become the production expert, as whole production process is based on the fermentation technology, it is not easy task for everyone to perform the production. So being a scientist, being a student of the science, you can make your career in the production also. To become a quality expert, to analyze the quality and stability of the product. Quality is the very, very important and crucial part of the probiotic segment, as because there are 2 important aspects. One is the strain identification, another is the product analysis. Both are very, very important, which is only performed by the technical person. So you can also become a quality or quality expert in the field of the probiotics. You can also become a regulatory expert, as because the people still don't know much more regulatory informations. So you can earn a lot of money from different industries who are working on the probiotics. To conclude, sorry if I've taken a lot of time. There are several aspects that follow, so it is very necessary to take the probiotics from the outsource by taking the different products. As you know that probiotic and symbiotic supplements both are very much helpful for to prevent the intestinal dysbiosis. These supplements become a potential therapeutic tool to enhance the immunity, and it is very necessary nowadays to enhance your immunity to prevent your body from the different infection. The increasing demand for the immunity boosting products such as probiotics expected to the bode well for the growth of the market in the near future, so that this segment also provide a better opportunity for the youth to become an entrepreneur as both growing, continuously growing day by the day. But still lots of attention and focus are required on the scientific approach and regulatory part of this segment to ensure the safety of this product for the consumer. Thank you so much for your patience and listening me. Thank you once again.
Thank you very much, sir, for your informative talk on the role of probiotic microbes in health benefits. Thank you very much.
Thank you.
Now, I would like to invite Dr. Brajesh Barse, International Center for Genetic Engineering and Biotechnology, New Delhi, for his talk. Dr. Brajesh Barse, working as a research scientist in ICGEB, New Delhi, and associated with DBT ICGEB Center for Advanced Bioenergy Research. He is dealing with intellectual property rights in biofuel domain. He is also engaged in the license and agreement and joint venture collaborations with various chemical and biochemical companies in India and abroad. Previously, he has worked as a scientist in one of the premier institute, that is NIPER, India, where he was engaged in the research and developmental areas, specifically in the green chemistry domain, one of the rapidly growing field at the interface of chemistry and biology. He has broad range of experience in protein chemistry, downstream processing, and assessing biodiversity for identification of noble biocatalyst, screening and isolation of the microorganism, and its application for the production of fine chemicals. He has been awarded by prestigious MIUR Fellowship for Young Indian Researcher by Government of Italy in the field of nanoscience and nanotechnology. I request Dr. Brajesh Barse to kindly deliver his talk on second generation biofuel India perspective on lab to market research program. Please, sir.
Yeah, thank you, Pallavi, for the nice introduction. And I would like to thank all the organizer for this conference who has invited me. And this is really a wonderful platform to share the science and knowledge. So let me share my screen. Can you see my screen?
So it is not visible yet. Please wait for a few seconds.
The moment I'm sharing the screen, the system is hang. Let me try again. It is visible now?
Yes, sir, it is visible.
Okay. Okay, so let's start the things. Today I have chosen the topic for the India perspective on lab to market research program. So my basic idea is to choose this topic because I can understand there's a lot of students are there attending this conference, and this is very important for those students to understand and see how the India is changing, and they are making their mind from basic research to application-based research. So here I'll give you the one example of the second generation biofuel, because we are involved in the biofuel domain areas. But government has putting this effort for all the healthcare, medical, and other agriculture things and all, so that the whatever the research which is going on in this country can reach to the commercial level, and the people will get benefited of this country. The basic idea behind this different perspective of Indian science is that government is looking for the capacity building and skill development, and importantly, they are interested to have more and more startup so that the country become developing to developed nation. So that is the idea behind. So I'm just giving you the example of the second generation biofuel. So before going further, I just would like to, the screen is visible or I have to do this full screen show?
Yes, sir. Please make it full screen.
Okay, that's fine.
Yes, sir.
So as the name suggests, DBT ICGB. So DBT means Department of Biotechnology, and ICGB International Center for Genetic Engineering and Biotechnology. So there is a collaboration between DBT and ICGB, and we have established a Center of Excellence in ICGB for the advanced bioenergy research. So when I'm saying the India perspective, so here the perspective is clearly visible that Department of Biotechnology, which is the government funding agency, is like moving forward to have the joint collaboration with the research institutions and university to have a different center of excellency to get the lab research data into the market level so that things can commercialize and we can generate more and more employment, and the interest in the science will get continued. Because what we are looking in this scenario, people are moving far ahead, far from the biotechnological and life sciences. They are choosing the financial things, they are going on the investment thing, which is good, but we just wanted to keep some of the great brain into the science. So these are the programs which give the boost and motivation to the students and the young researchers and the scientists, so that they can collaborate with the government agency and the industries and come up with the new solution which can have the potential to penetrate the market. So I'm giving the brief introduction of the ICGB. What as ICGB stands for the International Center for Genetic Engineering and Biotechnology. So we are having a three different component. One is in Italy, one is in New Delhi, where I am based in, and another was in Cape Town. So ICGB Italy is our headquarter, and the mandate of the ICGB to provide a center of excellence for research and training in genetic engineering and biotechnology addressed to developing countries. So as I told you, government is now thinking in different perspective how to get this research into the market, and I will just show you the idea behind and how we are executing that idea to come up with the product, and we can move from lab to the market. So other than the field like medical, healthcare, and the agriculture, Department of Biotechnology is having like interest on promoting the R&D for the biofuel technology, because there is a need for clean energy in this country. And as you know, there is a lot of pollution problem, and the environmental hazards are there. So we have to come up with the sustainable and the green solution. So the objective of this initiative is that they can collaborate with the multiple institution so that they can build a great research program to come up with the technology. So here I am giving you the example how government had decided to exploit the potential of the various institutions so that one of the technology can be developed. So in this regard, because DBT is giving the interest on the biofuels, in this regard, what DBT has did, they have set up 4 bioenergy centers. So here is the list. You can see there's a DBT ICT center for the energy biosciences, which is based in Mumbai. One is the DBT IOC. IOC stand for the Indian Oil Corporation. One is DBT ICGB, and another is DBT Pan IIT. So I am telling you the idea behind to combine these 4 institutions to come up with the technology. So DBT ICT basically previously known as a UTCT Mumbai. So DBT ICT is having the complete skilled people who can do the process development and fermentation technology. And the DBT IOC is the industry, Indian Oil Corporation. So they have collaborated with the Indian Oil Corporation to get the this technology to scale up and the commercialize. DBT ICGB, as I told you, ICGB mandate is to do the genetic engineering. So DBT collaborated with the ICGB to provide the genetically modified organism for the biofuel process. These three organizations are like completely applied organizations. They are working on the application sides, but always whenever you are going for the application based area, there is a need to have the some of the basic research. So in that matter, they don't want to disturb this application scientist on application based research institution. So what they have come up, they have tie up with the DBT pan IIT centers, where they have collaborated with the IIT Bombay, IIT Roorkee, IIT Guwahati, IIT Kharagpur, IIT Jodhpur. So 48 scientists and professors from these all 5 IITs are doing the basic research for these 3 institutions. They are coming up with the data and the information which can be translated into the application. So these IITs are given the information to the DBT ICGB Center, IOC and ICT. ICGB Center is getting the information for all the basic engineering aspects, and IOC is getting the information for all techno-economic analysis and life cycle analysis and everything. And ICT is getting the information for all kind of process development. So these you can see it's very interestingly how they have identified the different institutions, they have exploited their potential, and they have made complete setup of different organization in a very great collaborative mode, and so that the things can be processed in a faster way, and we can come up with the results and the things. So whatever the technology or product development, these 4 bioenergy centers can forward for the scale up and demonstration at the pilot scale. And whatever the result outcome we have, we first go for the patent, because this is very important when we are going for the like you are thinking about the commercialization of the technology. You need to understand the importance of the patent, which is like very important. And we are, after filing the patent application, whatever the important results, we are also going for the publication in the international journals, and we have more than 200 international publications, including with our 4 centers. So as I am based in the DBT ICEP center, so I'm telling you, we are the like a center for advanced biology research, and we are doing the cutting edge research, and our mandate is to perform the molecular biology and biotechnology based research and serve as a platform for the synthetic biologists to work in diverse biology areas such as microbial engineering, biochemical engineering, algal engineering, and system biology. Our center basically perform the technological based research in the field of genomic, metagenomics, transcriptomic, proteomic approaches, system biology, metabolic flux analysis, metabolic modeling, engineering algae, and microbes. And whatever the innovative leads, as I told before, whatever the innovative leads we get from the center, we use to improve the technology and go for the further scale up studies. So we have the expertise in the genomics, and we have the synthetic biologists with us. We have the fermentation technologies with us, and we have a core group of system biology who can look into the proteomics and the genomics data and the transcriptomics data. So we are having a dedicated omics group to take care of all the data management. So since the talk is on the biofuel, so we have to understand why biofuel when there is already fossil fuel available in the market. Because now there is a time to change the environment, there is a need to self-sustain, and there is a need to remove this adverse impact of the environment which is causing by the fossil fuels. There is a, you can see every day there is a price hike in the petrol and diesel and oil products. We have to control this, and if we are coming up with the biofuel technology, then we will have the less dependency on the foreign countries in importing the oil. And in this case, we can save a lot of money, and which can be utilized for the other development progress project in the country. So the adverse impact on the environment due to the high emission of the carbon dioxide, because of the lot of industries are using the kerosenes and other oil products. So they are coming up this large amount of CO2, and it is going to the environment, and it is impacting very hazardously to the things. Vehicle pollution is there, air pollution is there, land pollution is there, everything is there. So because of this adverse effect in the environment, we always come across with a lot of problems. And you can see, if the climate has changed, how it will affect the human health. And as a biotechnologist, you can understand human health is importance of the human health, because very recently we seen the corona tragedy and how this human health is struggling with this. So if the climate will change, there will be a rise in temperature, there will be a more extreme weather, there will be a sea level rising and the increasing CO2 level, and which cause all this problem: air pollution, change in vector ecology, increasing allergens, water quality will get impact, food supply will get impact, environmental degradation will be there, extreme heat will be there, and with different kind of a problem and possibilities, you can see how this will affect the human health with asthma and the malaria and the respiratory disease, the cholera and the malnutrition. And there is a lot of things associated with the climate, lot of things associated with the pollution, and how we can reduce the pollution with the green energy, clean energy, and sustainable solution for this country. So this is the idea uh, of the thought of the government, that we have to have a sustainable solution for ourselves to come up with the to challenge all these things and come up with the solution, so that we can save the future population, we can save the mother earth and everything. So by keeping all these things, India certainly needs the clean energy. You are well aware how it is impacting in our day-to-day life in the winter season. A lot of smoke is coming from the Haryana and Punjab because they are burning the biomass, and then because of that, a lot of smokes and mixing with the air, and then it is creating a lot of pollutions. And as per the report, Delhi is the world most polluted city. So we have to take this as a challenge. We have to work together, and then we have to come up with a solution so that we can eliminate all these things by having the new and innovative technology. So for fighting with the pollution problem, I think biofuel is very must as a clean and in-house alternative. So the topic in my title is suggested is second generation biofuel. But before going to second generation, I just wanted to highlight what is first generation, so that you can understand why we are moving from first generation to second generation. The first generation biofuel is always made from the sucrose, starch, vegetable oils, and the grains, and which are the food product. You can see these are the common biofuel feedstocks. These are food product. And as we are the developing nation, we are very poor country. We cannot sustain if we use the food product to produce the fuel. So there is a problem. There is a food versus fuel problem in country like India. US can produce with the first generation fuel, Brazil can produce, but we can't afford to produce the first generation biofuel because of these inherent problem currently in this country. So we have, what is the solution? The solution is that we have to move for the second generation biofuel. What is second generation biofuel? In the second generation biofuel, we are using the waste biomass from the crops. We are using the non-food crops. We are using the industrial waste. And because of this waste material and the lignocellulosic, it is called lignocellulosic biomass. So we are producing the fuel by using this waste material. So it is again called waste to energy. And there is another third generation biofuel also coming in the picture, which can be produced by the algal biofuel. So we can use the algae to produce the lipid, and which can later be converted into the biodiesel. So the second generation biofuel will give you the opportunity to fulfill the requirement for the ethanol and the petrol. And the third generation biofuel will give you the opportunity to fulfill the demand of the diesel. So you can replace the diesel with the biodiesel. You can blend the second generation biofuel bioethanol with the current fossil fuels. So there is a policies made by the government of India, which is like clear-cut indication how we can go ahead with the biofuel technology. So the government of India has come up with the national biofuel policy recently in 2018. The policy aimed to increase the use of biofuel in the energy and transportation sector, and the policy aimed to utilize, develop, and promote domestic feedstock and its utilization for the production of biofuel. So they are encouraging the following application because they are reinforcing ongoing ethanol or biodiesel supplies through increasing domestic production. They are setting up second generation biorefineries. They are developing the new feedstock biofuels, development of new technology for conversion of biofuels, creating suitable environment for biofuel and its integration with the main fuels, like blending I am telling about, and the advanced technology for the generation of the biofuels. So how the biofuel can produce from the lignocellulosic biomass? Here I am giving the brief. So these are the agriculture residues like raw material, bagasse and rice husk and whatever the waste product after getting the grains from the all agriculture things. So whatever the residues are remaining, we are using it because these residues contain lignin, hemicellulose and cellulose. So because of this lignin, hemicellulose and cellulose, this agriculture waste is always called as a lignocellulose, right? So we do the pre-treatment to remove the lignin and we'll get the hemicellulose and cellulose. I am telling you because of the collaborative model, ICT Mumbai and the IOCL Faridabad is doing the pre-treatment for us and they are giving us the pre-treated biomass. What we are doing, we are developing the recombinant strain to increase the enzyme titer and then we are increasing the enzyme efficiency so that we can sacrifice or hydrolyze this biomass into the simple sugar. Once we get the simple sugar, we will go for the fermentation with the yeast strain and with the yeast strain, we can ferment and produce the biofuel molecule. This is for the second generation. For the third generation, we are also doing on even for the second generation, we are also doing the omics studies for facilitating genome engineering. We are doing the big data analysis to identify regulatory network. Then we are making the natural pathway engineering and doing all kind of system biology things so that all kind of theoretical knowledge can integrate into the platform and we can develop the heterologous host, be it algae or fungus or bacteria, anything could be, and we can produce the biofuel for the clean energy purpose. So by all this collaboration, I'm not going for the all technical details how we are doing the science and all behind this because this is completely market how we are approaching the market. So with all collaboration, our center is like ready for the commercialization. We have developed the enzyme composition for biomass hydrolysis. Here you can see this is our enzyme composition and these are the market commercial enzyme composition and we are at par, even we are great in terms of the efficiency of our enzyme. And I just wanted to tell you there because there is only one company throughout the worldwide who is developing this enzyme for this biomass hydrolysis. And we are fortunate enough that because of this great collaboration and the government support, we are almost there to compete with this giant company and we can approach to market and we can capture the market in India as well as in the abroad. So this is the beauty of the collaboration. So one has to understand how the collaboration is very important. You alone can't do any research. You need to have the collaboration because there are different brilliant minds sitting on the different institution and we have to combine all together and make one chain and then you can see the result of that particular strategy, how we are coming up with this. So we have developed and our research has captured by the various news agency, various journals. You can see that Nature India has published our research and then ex-Science Minister Harshvardhan tweeted, he is Dr. Shams Yazdani who is developing this recombinant fungal strain, which is here. And then it is highlighted by various news agencies and we have got the global recognition that we are there in developing the technology for indigenous technology for India's sustainable energy. So these are some highlight. So these are the things I am telling about the DBT initiative for the bioenergy production. Now I will just give you the brief information about the mission innovation because other than the DBT ICGB Bioenergy Center, we are also running the mission innovation program from the ICGB itself. So I'm just giving the brief introduction of the mission innovation. What is the mission innovation? Because in 2015, these three, like Obama, when he was the president, and the France president, and the our honorable Prime Minister Narendra Modi has the meeting in the Paris, and they have come up with the idea along with this Bill Gates to accelerate the clean energy revolution. And this mission innovation, in this meeting, the mission innovation name is coined by our Prime Minister, and all people agreed, and then we have initiated the mission innovation activities in India. So mission innovation is a global initiative catalyzing a decade of action and the investment in research, development and demonstration to make clean energy affordable, attractive and accessible for all. And this will accelerate progress towards the Paris Agreement goal and pathway to net zero. And ICGB was the fortunate enough to initiate this global initiative from the ICGB premises, and we are like initiated this activity since 2015, and we have completed the 5 years of this mission innovation. And what we have done, so these are the mission innovation activity. We have created the forum for the innovation leaders, and we have launched the global missions. We have built the pathway for deployment, because always there was a R&D things were going on, research and development, but there was no deployment. So we have more focus on the deployment. We worked in active partnership with the private sector and the other initiative to boost the demand for new solutions and explores the groundbreaking public-private investments. We have enhanced the confidence on government and the global level to net zero solution, and we will strengthen the global analysis of innovation progress and facilitate knowledge sharing and collaboration to support all countries to plan ambitious clean energy transition. So as I said that we have initiated the mission innovation activities in 2017, and now we have almost completed to 5 years. And then initially in the phase 1, we have the founding member of the mission innovation. We were leading in the 3 mission innovation challenges: smart grid, off grid, and sustainable biofuels. We have created the funding opportunity for the researchers, where we have made a very great platform, where what we did, we integrated the one Indian university or institution with the private investor or company, and along with one foreign participants. So these are very interesting funding opportunity announcement that which we have initiated, and we have funded almost 25 projects under this mission innovation, and we are getting very great response and results, and very good outcome of these projects. Now we are moving towards the mission innovation phase 2, where I just wanted to have some attention of the students, that they can understand how India is like moving forward. Now we are not restricting ourselves into the universities and the research institution. We are like showing our presence in the global level. So now we are the co-leads in the integrated biorefinery missions. We are the member of clean hydrogen missions. We are a core coalition member of the green powered future missions, core group member of the zero emissions shipping mission and urban transition missions. We are the support group member of carbon dioxide removal missions. We contribute to the innovation platforms. We are the member of the mission innovation steering committees and provide resources with mission innovation secretariat and collaborator module managers. So these are the MI missions, because MI missions set a global ambitions, provide political momentum, and enable governments and the private sector to work together to make sure more and more innovation happens more rapidly over the next decade. Mission brings together dynamic and delivered focus high emissions alliances between countries, corporation, investor, research institution that focus on supply, push and innovation, whilst building partnership to the build demand. So in the phase 2, we will come up with the integrated biorefineries. We will work on the clean hydrogen mission. We will work on the zero emissions shipping, green powered future, urban transition, carbon dioxide removal, net zero subsidy. So important thing is to understand from this presentation is that how the things are changing, scenario are changing. You have heard about very great presentation since yesterday, that how Indian scientists, professors are doing very good research. They are coming up with very great results. But somehow in our country, we are lacking with the collaborations. We are lacking that we cannot move our lab research data into the commercial level. We are lacking to make a private partnership. We are lacking to have a complete, because most of the universities and the institution are dependent on the government funding, but there are many possibilities and opportunities available where you can interact with the private partners and you can have the investment there. And once you have the private partnerships, easily you will go for the commercial level, and you can capture the market for these things. So here are some of the success stories for the advanced biofuel. So DBT ICGB Center has developed the enzyme composition for cellulosic biomass hydrolysis. DBT IOC Center, along with the Lenza Biotech, has developed the new technology for lipid production, and they are producing the biodiesels. Raj Industries, which is a collaborative partner of DBT ICGB, is putting up the second generation biomass to bioethanol plant technology, and they are using the DBT ICT technology to set up the plant, and they will use the ICGB enzyme to produce the biofuel. Sorry, government.
Excuse me, sir. I request you to kindly please summarize the talk as soon as possible.
Almost done. And
Yes,
government has also developed a stage of development of 18 cellulosic ethanol demonstration plants, and 4 plants are already established and rest are underway. So this is all from the my side. Thank you, and I hope you understand how interestingly science is growing, and I think more and more students will get interest to come again in the science field. Thank you.
thank you very much, sir. Thank you very much for your informative talk on the second generation biofuel. Thank you very much.
Thank you.
Now, I would like to invite Professor Dr. Pankaj Rai, Department of Biotechnology, Faculty of Bioscience, Inverters, University Bareilly, for his talk. Professor Dr. Pankaj Rai, he has completed MSc and PhD in microbiology from BHU Varanasi, presently working as an associate professor in Department of Biotechnology, Inverters, University Bareilly, UP. He has research and teaching experience of more than 8 years. During this period of 8 years, he has handled various responsibilities like PhD cell deputy coordinator, coordinator of NEC criteria 3, and overall coordinator of NIRF. His research areas focuses on bioactive compound biofuels production from organic waste using bacteria and cyanobacteria, plant growth promoting rhizobacteria, and synthesis of value added product through fermentation. I request Professor Dr. Pankaj Rai to kindly deliver his talk on substrate pretreatment technologies for biohydrogen production from biomass. Please, sir.
Thank you, Dr. Pallavi, for a nice introduction.
Thank you, sir.
A very good afternoon to the respected chairperson of the session, Dr. Michlesh Jaiswal, sir, co-chairperson, Dr. Brajesh Barse, sir, ladies and gentlemen. Before starting my talk, I would like to thank the organizing committee of this second international conference on recent advances in biotechnology and nanobiotechnology 2022, especially Professor R.S. Tomar, sir, for providing me the platform to share my research experiences with you people. Please allow me to share the slide. Is it visible?
Yes, sir, it is visible.
Okay. So in the previous talk, Dr. Barse has correctly pointed out that
Sir, I request you to make it full screen.
I have made it. Okay. So in the previous talk, Dr. Barse has correctly pointed out that there is an urgent need to shift from conventional fossil fuels to conventional and alternative energies. And this is due to the explosive population growth, rapid industrialization, resulting into high consumption of fossil fuels that are limited, and they release exhaust gases, creating health problems and global climate change. Thus, there is a need to change the current energy scenario. And this is the report from International Energy Agency USA. It states that after 2030, there will be a huge gap between the energy demand projects in the energy demand and energy production. And this huge gap can be only filled with the help of renewable energies. And these renewable energies may be obtained from solar, wind, geothermal, hydro, tidal energy, etc. But all these renewable energies depends on the geographical positions and weather conditions. And they have the limited usability like electricity generations. But biofuels don't have above limitations. They may be liquid and gaseous fuels derived from organic matter. And they may be the source of electricity, heating, and as a transport fuel. So in short, if we compare the various types of biofuels, we can find that H2 is better than other biofuels because it on combustion, it releases only water and is produced by waste materials, which will never run out as long as there will be life on this planet. So here we can see that two molecules of hydrogen, when burnt in the presence of oxygen, they produce 2 moles of H2 and 572 kilojoules of energy. So for the production of biohydrogen, we need substrate in the form of simple sugars, and using the pure sugars will increase the cost of H2 production. So to overcome this issue, H2 production biomass from biomass is imperative, and for that different kinds of biomass either it's from agricultural crop residues or from sewage or from municipal solid waste or from animal residues or obtained from industry or from obtained from forestry, is used for the production of hydrogen. And since biomass tends to be bulky and to deteriorate over time and difficult to store and handle. So pre-treatment process, yeah, we may say that the pre-treatment is necessary for the biomass before its utilization as a substrate for biohydrogen production, because pre-treatment enhances the rate of hydrolysis around 3 to 10 fold, and it destroys the matrix structure of feedstock that make it unsuitable for fermentative hydrogen production, and to overcome the recalcitrance of the biomass. And it also prevents processing problems of the substrate. Here we can see, here we see that pre-treatment technologies are of various types like physical, chemical, physiochemical, and biological. And the goal of these treatment technologies is just to reduce the complex biomass into the simpler fermentable sugars. And in this process, phenolic compounds like HMF, hydroxymethyl furfural, or you may say that ferulic acids are produced, and they may be inhibitory to the fermentative organism. Uh, Here we can see where you see that this is the biomass which is pre-treated either from the mechanical forces or using physiochemical techniques or biological techniques or chemical techniques. Then after this pre-treatment, it is anaerobically digested, or you may say the fermented, and the biogas hydrogen is produced. So in our research, we have targeted sugarcane bagasse as a substrate, or you may say as a biomass for biohydrogen production. Choosing the sugarcane bagasse is a very, you may say that it is questionable during the research, but we select this sugarcane bagasse only because of its um, uh, abundance in the India, and it is produced during the cane milling for sugarcane production. And around 1 metric ton of sugarcane produces around 280 kg of bagasse, and its composition ranges from composition is like cellulose 40 to 45%, hemicellulose like 30 to 35%, and lignin around 20 to 30%. And the cellulose and hemicellulose are the ideal substrates for the fermentative hydrogen production. So the cellulose component of the sugarcane bagasse is enzymatic hydrolyzed, and it will result into the production of sugars, and after that H2 production. And the hemicellulose component is acid hydrolyzed, and the sugar produced from that hydrolysis will be used for H2 production. For the production of hydrogen from these biomass, we use three strains. First strain, or you may say the first step is dark fermentation process, which included Enterobacter aerogenes MTCC2822, and the second step was for photo fermentation that is carried out by Rhodopseudomonas BHU01 strain. And the hydrolysis of the biomass is biologically done by the enzymes in situ enzymes produced by the Pseudomonas fimi. And the feedstock utilized was sugarcane bagasse. This is the schematic representation of H2 production from sugarcane bagasse. We hydrolyze the sugarcane bagasse with the means of chemical and enzymatic processes. The sugarcane bagasse is taken and it is acid hydrolyzed. That results into the breakdown of hemicellulose, and it comes in the liquid part. And this liquid part contains the toxic compounds like HMF and other compounds like phenols and other things. And to detoxify this liquid part, we pass all these liquid part into the amberlite X84 column, and this results into the removal of these toxic compounds, and further this liquid part is subjected to dark fermentation with the help of Enterobacter aerogenes. Now the remaining part, that is is solid part which contains cellulose, is hydrolyzed by the enzymes produced by Pseudomonas fimi in a hydrolysis reactor, and the sugars released during the hydrolysis are utilized for dark fermentation by Enterobacter aerogenes, and this results into the production of volatile fatty acids and H2. Now, the final step was to utilize the spent medium generated after dark fermentation of both the processes. The volatile fatty acids obtained from the acid hydrolysis of dark fermentation or from the biological hydrolysis of the cellulose part is further photo fermented with the help of Rhodopseudomonas BHU01 strain. This is the schematic setup for H2 production from sugarcane bagasse cellulose fraction. Here we can see that this is the hydrolysis reactor, which contains Pseudomonas creamy as a active agent, and in this sugarcane bagasse solid part is incubated for 48 hours, and the reactor is operated at optimum pH and temperature, and the sugar produced due to the hydrolysis of sugarcane bagasse is transferred to the other fermenter reactor with the help of peristaltic pump, and after this fermentation, the entropic erosions act on the sugar, reducing sugar produced, and it produces hydrogen and CO2. And this hydrogen and CO2 is further transferred to a CO2 trap, which are used in sequential manner to trap the CO2 produced, and finally the pure H2 is uh, transferred to the water displacement system, which measures the amount of H2 produced. The purity of the H2 is estimated with the help of HPLC. So this is the composition of sugarcane bagasse acid hydrolyzed for, and we during the experiment we found that the H2SO4 concentration of 2% is very much in optimum for its hydrolysis, as it produced 3.41 gram of per liter of glucose, 8.36 gram per liter of xylose, and 0.55 gram per liter of arabinose, having a total sugar concentration of 12.32, and acetic acid 1.49 gram per liter, and furfural, which is inhibitory for the fermented organism, only 0.19 gram per liter. So to observe the, or you may say that to see the effect of detoxification of sugarcane bagasse acid hydrolyzed on biohydrogen production by erosions, we conduct an experiment and used the untreated acid hydrolysate as well as the detoxified hydrolysate for the fermentation process, and we found that the untreated one produces 740 ml per liter of hydrogen with the rate of 8.8 ml per liter per hour, whereas the detoxified, which was passed through the column of amberite XAD4, produces 1000 ml per liter of hydrogen with the rate of 11.90 ml per liter per hour, which is around 1.3 fold. So we also studied the time course of reducing sugars released during the growth of Pseudomonas creamy on acid pre-treated sugarcane bagasse, and found that the consumption of reducing sugar by the actively growing Pseudomonas female cells in the hydrolysis reactors themselves, because up to the 48 hours the reducing sugar concentration was high, but after 48 hours the Pseudomonas female cells itself starts consuming the reducing sugar. So what we do, we simply put off our reactor after 48 hours and withdraw the sample, or you may say to withdraw the liquid portion for the fermentative hydrogen production. And the initially reducing sugar was reported only after 12 hours of incubation. So this is the time course of reducing sugar, acetic acid, butyric acid, and H2 production by erosions in simultaneous saccharification, filtration, and fermentation mode. And here you can see that the cumulative H2 production rate was 613 ml and production rate was 5.10 ml per liter per hour, with the production of acetic acid and butyric acid. Here we see that from time 0 to 12 hours, the sugar consumption was increasing, and after that it is start decreasing, and the same curve is followed by H2 acetic acid and butyric acid. Like H2 production increases, acetic acid production is also increased, and the butyric acid production also increases. It indicates that the consumption of sugar is directed to the production of H2 acetic acid and butyric acid. This is the time course of H2 production during photofermentation by Rhodopseudomonas GHU01 strain from spent medium obtained from the dark fermentation of sugarcane bagasse acid hydrolysate, and we found that the spent medium obtained from dark fermentation and from SSF have the capability to produce H2 in the photofermentation as well. And the cumulative H2 production was 755 ml per liter and 355 ml per liter for dark fermentation spent medium SSS and SSF spent medium respectively during 96 hours of fermentation. So we may can conclude that the optimal acid hydrolysis was by H2SO4 at 2% volume by volume at 121 degrees centigrade for 60 minutes. And bagasse acid hydrolysis has enough potential to support H2 production as it contains 12.32 gram per liter total sugar with low amount of fermentation inhibitors like acetic acid and furfural. And the bagasse cellulosic fraction following enzymatic hydrolysis by on-site cellulose from cellulose streaming can be utilized for H2 production. And also the sugar released by cellulose streaming from the acid pre-treated bagasse increased up to 48 hours, but started declining further, possibly due to its consumption by cellulose streaming cells themselves. Finally, the simultaneous saccharification, filtration, fermentation, and two-step fermentation could be effectively utilized for H2 production from the lignocellulosic biomass. And these are the publications, recent publications from our lab. And now, thank you for patiently hearing me. That's all about my talk.
Thank you very much, sir. Thank you very much, sir, for providing your deep insight on the biohydrogen production from biomass.
Thank you. Thank you.
Now, I would like to request chairperson of the session, Dr. Mithilesh Jaiswal, to kindly sum up the session.
Thank you so much, Dr. Pallavi. I just like to thank you all the presenters like Dr. Brajesh Barse and Professor Pankaj Rai from Bareilly for the wonderful presentation and a very informative lecture given by both of them. Particularly, I would like to mention Dr. Brajesh Barse for the second generation biofuel. Actually, everyone know that in the current scenario, we can't expect the fossil fuel as well as the first generation fuel for the to meet of fuel in the coming years due to the high demand of the all these things due to high for the high population. So it is very necessary to generate or focus on the research on the second generation of the fuel. Also, I would like to mention over here the point which is raised by the Dr. Brajesh Barse, that the research should be very collaborative. It should be from academic up to the industries to create some beneficial product for the consumer. So my suggestion is that, Doctor, it should also be based on the requirement and need of the industry so that they can able to form a product which is very very utilizable for the consumer. And second, Dr. Pankaj Rai given a very informative lectures that based on the biohydrogen production from the biomass. It is particularly from the sugar cane bagasse. It is one of the also very right approach to produce the biohydrogen, which is a very important biofuel. So I just also like to request Dr. Pankaj Rai to create some collaboration of all your research activities with the industries to make your research in a complete form, because a lot of demand of whatever the byproducts we have developed through the fermentation of the sugarcane bagasse, definitely it will develop in a form of very useful product. So it is I think everyone agree with this point that the research should be reach at the end point of the utilization. That is we have already discussed in my previous lecture. That means going in a right pathway, which is from the lab up to the market. That's why you are doing a very fabulous job, Dr. Pankaj Rai. So I hope in future I just wanted to see your research in a form of product through the collaboration of any industry which you like to do. And again, Yeah, sure. Again, I would like to thank Amity University Gwalior, Professor Tomar, for inviting me as a presenter, as a chairman in this session. And I'd like to congratulate once again for the grand section of the second international conference. Thank you so much, everyone, for presenting their lectures, for the particularly Dr. Pallavi for organizing, for their for her coordination. Everyone, thank you so much once again. Thank you.
Thanks.
Thank you. Thank you very much, sir. On behalf of AIB AUMP, I would like to thank all the eminent persons who added glory to this session. Now this is the time to close the session, and as per the schedule, we now move for the valedictory session with the same link after a short break.
Thank you, Dr. Mithilesh, Dr. Pankaj Rai.
Thank you, sir. Thank you.
And Dr. Barse, thank you so much for coming over here and enlightening
very
the
much.
participants with
Thank
your
you, sir,
words.
for your
Thank
invitation,
you
sir.
so much.
Thank you, sir. Thank you
We
so
shall
much.
be in touch and certainly we'll be meeting physically also, not
Yeah, sir.
on the
Sure,
phone
sir, sure,
virtual.
Sure, sure.
Okay, thank you so much.
Okay.
Thank you.
Am I audible?
Yes, Dr. Pallavi, you're audible.
A very good evening to one and all. We have been listening to the eminent speakers deliberating their ideas in the emerging areas of life sciences. All these are very good evening listening to the eminent perspective that one should adopt while discussing these issues. Ladies and gentlemen, we have with us Professor Dr. Sangeeta Shukla, Vice Chancellor and Chief Guest for the session. Honorable Vice Chancellor Lieutenant General, Pro Vice Chancellor Professor Dr. M.P. Kaushik, Dean Academics and Director AIB Professor Dr. R.S. Tomar. To make this event a blessed one, we invoke Goddess Saraswati by Saraswati Vanna for seeking hands. I request all the dignitaries for lamp lighting. Thank you, everyone. Now, I request Honorable Vice Chancellor, sir, to welcome our chief guest, Professor Dr. Sangeeta Shukla. Thank you so much. May I now request Professor Dr. Sangeeta Shukla to kindly address the audience. Professor Dr. Sangeeta Shukla is Vice Chancellor, Chaudhary Charan Singh University, Meerut. She has wide experience of research in the field of biochemical pharmacology, environmental toxicology, and reproductive biology. She has been awarded fellowship from the Wellcome Trust Indo-French Government Fellowship, UK, and many others. She has published several research papers in SCI Journal with good citation indices. She has also edited books and contributed chapters in the books. In recognition of her efforts, she held international positions as Vice President for Asian Continent of International Centers for Trace Element Study for UNESCO. France, including council member of ICTERH, she has completed approximately 20 major research projects and supervised several PhD theses and many dissertations of MPhil and MSc students. Please, ma'am, I request you to kindly address the audience.
It's good evening, eminent scientists, educators, guests from India and abroad. It's indeed a great pleasure and honor for me to be here today for this very important international event, that is second international conference on recent advances in biotechnology and nanobiology, hosted by a very prestigious university, MIT Institute of Biotechnology, Gwalior. I would like to thank Lieutenant General V.K. Sharma ji, an eminent administrator, academician, and vice chancellor of this university. Professor Kaushik, Pro Vice Chancellor and coach international event, and Rajesh Tomar, a student of our university, my student rather, and very close to my heart, organizer of this very important event. So I would like to congratulate this entire team for this very important event. And I'm told that more than 370 participants have been attending this 2-day event. For me, nanotechnology is a bright and upcoming field. And we believe with Indian brain power, nothing is impossible. I've quickly gone through the brochure and I could see that more than 24 research areas of biotechnology has been covered by this conference. For me, these technologies, they serve as a blanket in terms of various related technologies. The science of nanoscale structures, they deal with creation, investigation, and utilization of systems that are 1,000 times smaller than the components. Thus, by using these things, various fields, including microelectronics, has been benefited with biotechnology and nanobiotechnology. Biotechnology deals with metabolic processes, we all know, with microorganisms. Convergence of these two technologies has resulted in immense growth in multifaceted areas. This interdisciplinary combination has got great innovative tools. Few to mention: drug formulation and drug discovery, semiconductors, solar energy systems, pharmacological research and clinical diagnostics, supplementing immune system, detection of proteins, probing of DNA structures, tissue engineering, including tumor studies. Utility of nanotechnology to biomedical sciences implies what? It implies that we can reach the subcellular places. Subcellular spaces means we can reach the high degree of specificity. And this reaching to a target of high degree of targeted cellular tissue specific clinical implications. This is the most advantage area of these two technologies merged together. The biomedical applications of nanotechnology are the direct product of these two mergence, I can say. The challenge which we are facing is we want scientists to come up with engineers. This is the new thing, which when together with engineers working in the field of nanotechnology can create extraordinary results. If I talk of biomedical sciences, I have to say revolutionary opportunities in the fight against all kinds of cancer, cardiac and neurodegenerative disorders, infection and other diseases. These two technology has done wonders. If I talk of medical applications that offers potential development, have shown that markers, specifically epidermal growth factor receptors, combining P53 mRNA nanotherapy with immune checkpoint blockade reprograms the immune microenvironment for effective cancer therapy. This is one of the most important areas where we need to work further. Then phenylalanine gold nanoclusters are being used as sensing platform for inferring molecules. If I talk of small interfering RNAs, normally called as siRNAs, they are being used as therapeutics. And nowadays, we really need therapeutics when we have come up with all type of COVID problems. Whereas nanoparticles like nanopores, nanotubes, quantum dots, nanoshells, nanospheres, they have got structural properties where they determine the affinity and specificity of nanoparticles, which are of great significance. If I talk of nanoassemblers, what are these? These nanoassemblers, they are smaller than the cell nucleus and they fit into places and they really reach small, minus areas in the cell, and they can be used to destroy bacteria of the mouth. Recent research have says that these nanoassemblers, they destroy the bacteria in the mouth. They cause dental caries and they have got repair spots. See how they work in all medical application. If I talk of nasal sprays, they have got the nasal sprays, they are getting converted into various therapeutic agents at the moment. Nanotechnology will probably have a greater impact on the pharmacological sector if we work more hard to see that how pharmacology can be. See, this is after 2 years of COVID, where what we have learned. We need to do more research. We need to come up with more pharmacological drugs. We need to come up with technologies. We need to merge many other different things so as to protect the human kind. This is what we want to do. Everybody has got the same aim. We have to save the mankind. This is what. If I talk of nanoscale polymer capsules, I was reading yesterday, they release drugs in such a minute quantity and in the controlled rates that they're very effective in tumors. They're very effective in treating tumors versus normal cells of the body. If I talk of novel polymers and exploring specific drug polymer combinations, this technology has also done wonders. Now, the recent development in pharmaceutical sciences and medical areas has also shown nanorobots, they are operating in the human bodies and they could monitor levels of different compounds and they can record the information of the internal memory, which is so important for all of us. Quantum dots with nanodots of specific color are believed to be flexible and could offer and a very cheap and an easy way to screen a blood sample for the presence of number of various viruses in the same tissue and at the same time. So if you think that we have to combine everything together, say for example, heart, lungs, blood and sleep research and disorders, we can come up with a nanotechnology of HLBS, that is heart, lung, blood, sleep research disorders. A national institute is working in this direction. So what I will request the audience, the scientists, the eminent academicians, we need to work on how this imprinted polymers can be medically useful in clinical applications, such as controlled drug release, drug monitoring devices, biological and antibody receptor mimics. We need to work on the application of a combination of nanomedicine with biophotonics, nanoparticles as a non-viral vector. This has been demonstrated if we see the recent papers. We need to work on nano DNA technology as well. Calcium phosphate nanoparticles recently, they are present in a unique class of non-viral vectors. If we study these, they can serve as an efficient and alternative DNA carriers for targeted drug delivery of genes. See, these are the major areas we need to work more. And if we work in these areas, we can come up with many good things combining these two technologies. We can focus on nanoparticles as biomarkers, nanotechnology in the measurement of dissolved oxygen. That was very much studied during this COVID period. Application of nanotechnology, to tissue engineering, which is very important, a new area where we can work. Growth of new organs and molecular imaging. In the future, we could imagine a world where medical nano devices are routinely implanted in our body and even injected into the bloodstream to monitor health and automatically participate in the repair of systems that deviate from the normal pattern. This is what is required in the current scenario. This continued development in the fields of biomedical biotechnology, because I'm more concerned with the medical biotechnology, medical nanotechnology, because health is a serious issue at the present times. See, the youngsters, they are suffering more because we are not focusing on the right health systems. So thus we need to establish collaboration between the research groups of different fields altogether. For this, I would like to congratulate the abstracts of these conferences where people from different fields, they are participating. And this offers a wide range. It is a think tank for the young generation to think how they have to do combined research, how physics can be connected with chemistry and chemistry can be connected with biochemistry and nanotechnology. This is the right time during COVID period that entire science should come up should same platform. So the sites clean energy, defense, health, medicine, infrastructure, communication, and more areas of your tropical interest. So this conference was a boon, I think, for entire academicians during especially the period when we all are sitting at home and we are just going through some Google things, not reading the books. So this is the right time when we can generate new ideas in the youngsters. India has achieved many milestones in the area of nanoelectronics, nanomaterials, and yet to believe that we have to reach a system where Digital India, Make in India, Startup India, Skill India, and another innovative programs. We can think of such programs, we can come up with different startups, we can come up with small projects, we can come up with small ideas while combining these technologies, both the technologies. I want to raise in the relevance of our society today. This field can change the world. In this ever-changing scenario, young researchers, young scientists, they need a leadership. These leadership can generate various ideas, and ideas can only come through such conferences. So I congratulate the leaders of this conference sitting right in front of me, Lieutenant General V.K. Sharma ji, Chairperson, Professor M.P. Kaushik ji, Professor R.S. Tomar ji, and their time. So I really thank you all for inviting me here. At least I could read something. I was unable to read anything, and this was the time when I could go through some papers. And so this is what I could get. So nice calling me here. I'm grateful and thankful to you for giving me some insight about this conference. And this is a very prestigious university where they can come up with many good ideas. And I would like to request the organizers to come up with the recommendation of this conference. These recommendations should be sent to the government. We come up with a draft report. This report should be sent to the government, and it will be very helpful in future endeavors. So thank you very much for inviting me again. Jai Hind, Jai Bharat.
Thank you very much, ma'am. May I now request Honorable Vice Chancellor, Lieutenant General V.K. Sharma, Ati Vishishta Seva Madam, to kindly address the audience. I feel immense pleasure to introduce you the guiding force behind our constant efforts, Honorable Vice Chancellor Lieutenant General V.K. Sharma, Ati Vishishta Seva Medal. General V.K. Sharma is an alumnus of erstwhile King George School, Shimla. He did BTech from College of Military Engineering, Pune, MTech degree from the Pune University, and MMS from Osmania University, all in the first class with honors. He joined the National Defense Academy and passed out as a permanent commissioned army officer from the Indian Military Academy in 1972. He was granted the President's Commission in the Corps of Signal in the Indian Army. He served the Indian Army for almost 40 years and retired He also headed military delegations to France, Germany, and USA. For his meritorious service, he was awarded Ati Visheshta Seva Medal by Honorable President of India on January 26, 2011. General Sharma is an excellent academician too. Under his able guidance, Amity University, Madhya Pradesh has grown by leaps and bounds toward excellence and has been awarded as the best private university of Madhya Pradesh twice, first in year 2014 and second in year 2017. May I now request Honorable Vice Chancellor Lieutenant General V.K. Sharma for addressing the audience. Please, sir.
Good afternoon, everyone. Professor Dr. Sangeeta Shukla, now the Vice Chancellor of Chaudhry Charan Singh University, Meerut. As far as Professor Shukla is concerned, I must say she is a technocrat and also an excellent administrator. I will use that word, 2 tenures simultaneously, consecutively in Jivaji University, and then to be selected to do another tenure of Vice Chancellor. Position in the field of biotechnology is well known, and what she spoke today about the nuances of biotechnology, nanobiotechnology is something which is quite revealing, and to that extent, all the researchers who are listening today must take lessons from whatever she has spoken, and I'm sure the outcome will be very positive. As far as I'm concerned, I would like to dwell upon the other side of nanobiotechnology. Any technology which comes, it has a flip side. I remember when I was young in school, at that time, the plastics came. And before that, we had glass bottles, glasses, steel, heavy metals, packaging in tins, etc. Once the plastics came, the whole world was gaga, saying that here's a revolution, so light, usable, they can be thrown away, much cheaper. And today, we are talking about banning the plastics, right? Similarly, the water purifiers came. I remember in our time, we had a water purifier, which the first one I had purchased was which had candles. The water used to go through those candles, which were porous in nature. The objectionable material was caught inside the candles, then the candles had to be rechanged after 6 months. And then came the technology of ROs. Now we say RO technology is no good because it takes out the bicarbonates and the magnesium to such an extent that the water, if the level becomes too low of these minerals, then that water, if taken, it starts absorbing those items from your body and weakens your body. Now some other system has to be found. Similarly, The term nanotechnology was first fielded by Richard Feynman in 1959. This term was first used in 1959. So that means the technology is very young, younger than my age, I would say. And then the first time that the nanotechnology, actually in 1959, Richard Feynman had said in his talk, he just referred to it that there is plenty of room at the bottom. He did not mention about nanotechnology as such, but it was in 1974 by Noria Taniguchi who used the word nanotechnology for the first time. So the first time the word was ever used was in 1974, right? So you can say how old nanotechnology is, but yet the advantages, and it is human tendency, you know, everything, we are positive minded people. Everybody is taught to be positive in outlook. You are supposed to see this glass as half full and not as half empty, because that is human nature. And rightly so, unless you see positivity in everything, how will we advance? But at the same time, we must look at this negative effects also, which in our exuberant to get maximum mileage out of positivity, sometimes we tend to neglect, for which later on we have to pay the price when it is already too late. Some of the disadvantages of nanoparticles are that they are so small. A nanoparticle is a billionth of a meter, and they can interact at the molecular level with our genes. They can interact with our RBCs, white blood corpuscles, which are there, with the RNA of the plants, DNA of the human beings and animals, and they can permanently change those in the manner that we do not know. While there are drug delivery systems which is being used by nanoparticles, but that nanoparticle which goes inside, deliver the drug, what happens to that nanoparticle thereafter? Do we have a system to remove it from our body? Have we thought about it? It has done its job, but it stays there in the system. Nanoparticles being used for waste cleaning, waste management, for cleaning of water, for cleaning of sewages, for cleaning of water tanks is well known. They are being used, and for removing the heavy metals from the pollutant, which are the very big pollutants of the industrial waste, which is very much required, because once the heavy metal particles, they go inside the ground, they are getting absorbed by the plants and being eaten by us, causing damage to our bodies. Researches have been done on this. How do we remove nano-used solutions have yet to be found. Similarly, the nanoparticles being used for cosmetics, for sun creams to prevent the ultraviolet rays, they contain nanoparticles. Through our skin, the nanoparticles can easily penetrate the skin and go inside our bloodstream. Not only that, the blood-brain barrier which is there, it is thought that they can penetrate that and go inside and damage our brains. It is possible. But unfortunately, because of so much money having been invested by the pharmaceutical companies, so much of money which is at stake in doing research to see the positive effects, not much heed is being paid at this stage towards these effects. Probably when we realize that the damage has been passed on from a generation to generation, it may be too late. Therefore, I would urge the researchers to take on research in such areas also as to how to remove the nanoparticles from any system where they have been employed once their utility is over. Because on its own, they are not self-destructive destructive in nature. Smaller the size, more difficult it becomes to destroy that system. Bigger size can be destroyed. And especially, it is known that if they go inside the lungs, they get most affected. Leave aside man-made nanoparticles. even the dust pollutants, once they go inside the lungs, the coal workers who are working in coal mines, inhaling very small particles of coal, which are nanoparticles, very minute particles of sand going inside us, the MSP levels which are there, all that is causing tremendous damage to our system, that is the breathing system. And they are being used in packaging. They are being used in preservation of food. So all that, surely some of them are going inside our system. And these, even Ayurveda says that the coarse material can remain in the body, but finer the particles, even maida is considered bad as far as Ayurveda is concerned. And what to talk about nanoparticles? But unknowingly, they are going into our system. And what will be the effect of those will only be known after some time. And when the damage is being felt, at that time we start the remedies to that. For the betterment of the humanity, that we think about the health issues caused by nanopollution. There is a plastic pollution. When it was thought that the space is free, but today there is space pollution. Now there is of nanopollution. Before it becomes a reality and it becomes a challenging factor, I think the scientists and technicians, technocrats must look at this issue, and I would advise the researchers to work in this field also, which is an area of not concern today, but certainly it may be an area of high concern tomorrow. So early breakthrough in such areas like removal of nanoparticles after their utilization is over, what percentage you can remove from the system, whether it is water system or it is purification system, or it is waste management system, or it is drug delivery system, or it is cosmetics, whatever be, wherever they are being used. After they have done the job, they must be removed. I think very little study and work is going on in that. That was something which I thought I will speak today. As far as the conference is concerned, I am very happy to state that we have had more than about 370 paper presentations, which were done by the researchers very enthusiastically over parallel sessions which were created, and a number of excellent speakers. I would mention some of them, like Professor Dr. C.P. Joshi, Professor and Chair, Department of Biological Sciences, who was the chief guest during the inaugural function from Michigan Technological University, Professor Jagar Ott, and then, of course, parallel sessions, Dr. Rajendra Prasad from MIT University, Haryana, one of the best scientists that the MIT have got, Dr. Manoj Prasad, Dr. Goel, Dr. Kaas Jain, Dr. Rajiv Prakash, Dr. Vasily Photopoulos, Dr. V.P. Sharma from CSIR, Dr. Aparna Khanna from MIT University, Mumbai, Dr. Priya Darshini, and of course, today in the morning, we had Dr. Raghuveer Singh Tomar, Dr. Gitanjali Yadav, Dr. Ramkumar Mishra, Dr. Gopalji Gopal, and session chairs, Dr. Nalini Shrivastava, Dr. Ravi Kant Singh, Jaish Harse, and of course now our most respected chief guest, Dr. Sangeeta Shukla. I am thankful to all of them, and some of the names I might have missed, for having shown light and having effectively contributed towards the international conference, which is very apt today and which is the need of the hour. In fact, the suggestion given by Professor Dr. Shukla regarding making the recommendations and sending them to the government for implementation is apt, and I'm sure we will try and do that. And I'm sure the researchers who are listening to from the which was released yesterday morning, and I wish this conference all the very best, Kaushik, and to Professor Dr. R.S. Tomar and his team, who have effectively worked so hard to put the things together and to make this international conference a great success. So thank you very much and Jai Hind.
Thank you very much, sir. Now moving to the session for prize distribution with the permission of respected chief guest, Honorable Vice Chancellor, Pro Vice Chancellor, and Director AIB. First of all, I would like to request our respected chief guest for announcing the best presentation.
Announce the full name, Meenakshi Dubey, their affiliations.
The award goes to Miss Meenakshi Dubey.
On behalf of Meenakshi Dubey, our research scholar is receiving this prize. Now, I would like to request Honorable Vice Chancellor to announce the next winner.
The next award goes to Mrs. Rashri Datta, Agriculture and Ecological Research Unit from Indian Statistical Institute, Kolkata, for her best presentation of research paper titled Evaluating the Potential Role of Plant Natural Products from Cleome with urease inhibiting properties on
The next. The winner is Miss Rashri. Thank you very much, sir. Next, I would like to request our Pro Vice Chancellor to kindly announce the next winner.
So next goes to Miss Biotechnology Institute of for a paper lateral flow essay for differential detection of elapid venom from other big four in the recent advances in today. So Ms. Dipali Rawat.
On behalf of Dipali Rawat, Pratibha is receiving this award.
Thank you. Congrats.
Thank you very much, sir. Next, I would like to request Director AIB to kindly announce the consolation prizes.
Good afternoon, everyone. Since we had 14 parallel sessions, so we have selected 10 out of them as a consolation prizes. First is Mr. Mohammad Fazil. He is from Sri Sakti Institute of Engineering and Technology, Coimbatore. He has been giving this prize for his paper entitled. Second prize goes to, not second prize, he's been giving this prize for a paper entitled, actually, second consolation prize. It goes to Ms. Priti Ji from Krishnamal College for Women, Coimbatore. Bee stress from Anabina species. Next goes to Ms. Sasibi Saini from Devi Ahilyabai, Khandwa Road, Taxila campus, Indore, for her work on effect of essential micronutrient zinc on growth, nitrogen assimilation enzymes, and various system-related parameters in Phaseolus vulgaris. Next goes to Mr. Patel Vishal School of Mechanical and Chemical Engineering from Galgotia University, on his work improved iodine and methylene blue number through functionalization of Jacaranda mimosafolia. Next prize goes to Miss Pooja Rajput. She is from Department of PG Studies and Research in Biotechnology from Karnataka. Her work is on study on the modulation of nicotine-induced gemesticide-resistant signals by amylin-loaded zinc oxide nanoparticles. And other prize goes to Miss Anjan Hazara from Agriculture and Ecological Research Unit, Indian Statistical Institute, Kolkata, for her work on expressional dynamics and differential splicing of WRKY family genes along physiological cues in tea plant. Last two. First is Mr. Vipul Gupta from Karaganda Medical University, Karagandla, Kazakhstan, for winning the Constitutional Prize. This international prize goes to this particular boy. He has worked on role of impairment in T cell maturation and development of rheumatoid arthritis. And it's the last one that goes to Mr. Karuganti Sukumar from Department of Microbiology, Center of Excellence in Life Sciences, Bhattidasan University, Tiruchirappalli, on his work on a detailed study on the silicate solubilizing bacillus equiphanesis isolated from radiosphere of maize. So congratulations to all the 10 Constitutional Prize winners. Thank you very much.
Thank you very much, sir. May I now request Professor Dr. R.S. Tomar, the convener of the conference, to kindly give the concluding remarks.
So now we have reached to the moment when we are going to conclude the 2 days international conference in 2022. And on this occasion of in 2022, I would like to inform the August gathering that this particular conference has set a milestone by bringing so many presentations that we have never seen, sir. And I think this is the advantage of online sort of working, that those who are invited or those who have given their abstract, they are presented. Except 2 from invited talks, there were 27 invited, 1 the guest of honor's address. Out of those 29, only 2 dropped out and 27 presented in the invited talks especially. Now I'm talking about the parallel sessions, sir. In parallel session, there were 5 sessions on day 1 and 9 sessions on day 2. So total 14 parallel sessions were there, in which there were 317 presentations and all the participants presented over there. So these are those selected 3, those who had highest marks with their performance and our assessment. So on the first day, sir, conference was inaugurated online by lamp lighting ceremony, followed by Saraswati Vandana in the presence of Chief Guest Professor Dr. C.P. Joshi, Professor and Chair, Department of Biological Science, Michigan Technology University, USA. Guest of honor addressed by Professor Dr. Jargot, Professor Emeritus and Head of Lab, Rockefeller University, New York, USA, along with Pro Vice Chancellor, AUMP, Professor Dr. M.P. Kaushik and myself. The inaugural session started with the welcome address by Pro Vice Chancellor, AUMP, Professor Dr. M.P. Kaushik, followed by the keynote address by the chief guest, Professor Dr. C.P. Joshi, on the topic plant cell wall biotechnology for improving bioenergy production. Afterwards, guest of honor Professor Dr. Jargot delivered his talk on gene linkage mapping and interaction of genes. Inaugural session ended with the release of abstract book of the 2nd international conference by dignitaries on the dais online. There were 3 main technical sessions on different themes on day 1, and 15 speakers from reputed international and national academia, they delivered their talk. Researchers from India and abroad, such as ICR, NBIM, MAU, Pratsula Engineering, Tiruvallur, GLA Mathura, Indian Statistical Institute, Kolkata, RKDA Bhopal, Bundelkhand University, Jhansi, Jivaji University, Gwalior, Rajmata Bajaji Scindia Agriculture University, Gwalior, Government KRG PG College, Gwalior, CMAP, Lucknow, IACR Bhopal, GR Medical College, Gwalior, Government SMS Model Science College, Sri Krishna University, Chhatarpur, Tropolite Foods Private Limited, Gwalior, CSIR NBRI, Lucknow, ITM University, VISM, Government Girls College, VIT University Bhopal, Bhatidasa University Tamil Nadu, and Tropical Forest Institute Jabalpur, National Institute of Animal Biotechnology Hyderabad, and Medical University Kazakhstan, North Central Hospital Brown, New York, USA, they participated in the conference. After inaugural session, the first technical session of the conference on molecular biology and plant biotechnology started with the invited talk of Professor Dr. Rajendra Prasad from MIT Institute of Biotechnology, MIT University, Haryana, and he spoke on the topic One Gene, One Protein, and One Life. The session was chaired by himself and co-chaired by myself. The second invited lecture in this session was delivered by Dr. Manoj Prasad, National Institute of Plant Genome Research, that is from NIPGR, and his topic was SW5A, the Trojan horse against tall CNDB infection in tomato. Followed by next invited talk by Dr. Sairendra Goyal from Department of Botany, University of Delhi, on the topic an integrated approach using genomics and breeding for a traditional Indian oil seed crop safflower, and Dr. Vikas Jain from Department of Biological Sciences, ISCR Bhopal. He spoke on deciphering the physiological relevance and molecular properties of mycobacteriophage D29 Holen. After lunch break, second technical session on nanobiotechnology and biosensor started, and session was chaired by Dr. Rajiv Prakash from School of Material Science, BHU, Varanasi, and co-chaired by Dr. Basilis Fotolis from Department of Agriculture Sciences, Biotechnology and Food Science, Cyprus University of Technology, Lemnos, Cyprus. Dr. Rajiv Prakash delivered an invited talk on the topic New Generation Biosensors based on nanomaterials, functionalized transducers. Afterwards, another invited talk was delivered by Dr. Basilis Fotolis on the topic Potential Use of Nanomaterials Towards Sustainable Agriculture. Followed by his talk, Professor Dr. B.P. Sharma from CSIR IITR Lucknow. He spoke on toxicology, recent advances and strategies for sustainable and regulatory aspects. In this continuation, another invited talk was delivered by Professor Dr. Aparna Khanna from MIT Institute of Biotechnology, MIT University Mumbai, on the topic Design of Novel Nanomaterials for Efficient Stem Cell Tracking. Followed by her lecture, another lecture was given by Dr. Seema Nara from Department of Biotechnology, Motilal Nehru Institute of Technology, Allahabad, Uttar Pradesh, on the topic aptamer as detection agents in vitro diagnostics. The last invited talk of the session was delivered by Dr. Subhendu Ranjan from Department of Biotechnology, CSJM University, Kanpur, Uttar Pradesh, on the topic electrospinning and electrospraying for active compound encapsulation. Session was ended with the parallel discussions and panel discussions between resource persons and participants. Afterwards, another technical session on applied biotechnology was started, which was chaired by Dr. Rajesh Tiwari from MIT University, Lucknow, and co-chaired by Dr. Alok Patel, Biochemical Process Engineering, Division of Chemical Engineering, Department of Civil, Environment and Natural Resources, Luleå University of Technology, Sweden. Professor Dr. Rajesh Tiwari delivered his talk on the topic Harnessing Science Technology and Innovation for Societal Benefits. After the lecture, Dr. Alok Patel delivered a lecture on the topic Bioprocessing of Volatile Fatty Acids as Building Blocks for Lipogenesis in Oligenic Microbes for the production of nutraceuticals to reduce dependency on animal sources. Followed by another invited lecture by Dr. Jyoti Pradarshini from GRMC Gwalior on applications of nanotechnology in healthcare. The technical session ended with the invited talk by Dr. Sonia Johari from Department of Life Sciences, IT University Gwalior. She spoke on therapeutic approach of watermelon, rent, biosynthesis, and characterization of selenium nanoparticles. On day one, there were total 5 parallel sessions for the oral presentations of participants from India and abroad. Parallel sessions were conducted online on Google Meet platform, where distinguished scientists and researchers as chairperson, co-chairperson in different sessions, in which a total number of 105 presentations were done on day one. On day 2 of the international conference, session 4, that is the main technical session on Zoom platform, was started on the theme of omics. It started with the invited talk by Professor Dr. Raghbir Singh Tomar from Department of Biological Sciences, IISCR Bhopal, on the topic Dissecting the Role of Epigenetics in Regulation of Cellular Homeostasis Pathway under Stress Conditions. The session was chaired by Professor Dr. Raghbir Singh Tomar and co-chaired by Dr. Gitanjali Yadav of Computational Biology Laboratory, NIPGR, Delhi. The second invited talk in the session was delivered by herself on spatio-temporal gene regulatory networks of algal biophysical CCM for food security.
followed by invited talk of Dr. Samir Srivastava from Department of Biotechnology, Motilal Nehru Institute of Technology, Allahabad, on the topic transcriptomic profiling of early onset CRCs. The session was concluded after panel discussion between presenters and participants. Now the technical session number 5 on animal and medical biotechnology started with the invited talk by Dr. Ram Kumar Mishra, Department of Biological Sciences, IISR Bhopal again, and he spoke on getting insight on cancer ex vivo laboratory approaches to delineate the menace. And the session was chaired by himself and co-chaired by Dr. Pankaj Suman from National Institute of Animal Biotechnology, Hyderabad, Telangana. Second invited talk was delivered by Dr. Pankaj Suman himself on the topic field applicable after sensor for detection of residual antibiotics in animal food products, followed by other invited talk by Gopal Ji Gopal, CG Bharta Institute of Biotechnology, Transdia University, Gujarat, on the topic role of recombinant DNA. Started with the invited talk of Professor Nalini Srivastava, School of Studies in Biotechnology, Biochemistry, University Gwalior, on the topic organophosphate pesticides from neurotoxicity to cell death. The session was chaired by herself, that is Dr. Nalini Srivastava, and co-chaired by myself. Session included one more invited talk by Dr. Ravi Kant Singh from MIT Institute of Biotechnology, MIT University Chhattisgarh, Raipur, on the topic DNA damage as a biomarker for exposure, the COMET assay perspective. After lunch, the last session, that is technical session number 7, was conducted on industrial and microbial technology. That started with the invited talk by Dr. Mithilesh Jaiswal. He is from Biotech Lab of Research and Development Division, Propolite Private Limited, Gwalior. He spoke on the topic role of probiotics microbes in the health benefits and career buildup. The session was chaired by Dr. Mithilesh Jaiswal and co-chaired by Dr. Brajesh Barse, International Center for Genetic Engineering and Biotechnology, New Delhi. Further, Dr. Barse delivered a talk on the topic second generation biofuel, India perspective on lab to market research program. In this continuation, one more invited talk was delivered by Dr. Pankaj Rai from Department of Biotechnology, Faculty of Biosciences, University Bareilly, and Dr. Rai deliberated on the topic substrate pre-treatment technologies for biohydrogen production from biomass. And this was the last invited talk of this technical session and the last talk of the conference in the technical sessions. So on day 2, there were nine parallel sessions for the oral presentations of participants from India and abroad. Parallel sessions were conducted online on Google Meet platform with distinguished scientists and researchers, a chairperson and co-chairperson, and different sessions in which total 212 presentations were conducted. So, sir, we conducted 14 parallel sessions on Google Meet and main session on Zoom platform for 2 days continuously. So that's all about my concluding remarks on Intranet 2022. I truly appreciate and thank you all for your gift of time and patient hearing. Thank you all.
Thank you very much, sir. May I now request Professor Dr. Vikas Srivastava, Organizing Secretary of the conference, to give vote of thanks.
Good evening, everyone. On behalf of Organizing Committee of Second International Conference on Recent Advances in Biotechnology 2020, I, Professor Vikas Srivastava, really feel privileged to propose official vote of thanks. First of all, I am thankful to Professor Dr. Sangeeta Shukla, Honorable Vice Chancellor, Chaudhary Charan Singh, University Meerut, Chief Guest of the valedictory session, for accepting our invitation and enlightening us with words of wisdom. Thank you, madam. I am also grateful to Chief Guest of inaugural session, Professor C.P. Joshi, Professor and Chair, Department of Biological Sciences, Michigan Technology University, USA, and Guest of Honor, Professor Jargot, Professor Emeritus and Head Statistical Lab, valuable address. I am also grateful to all the invited speakers of international conference who spared their valuable time from their busy schedule to share their knowledge and research experiences with us. I am also thankful to all the chairpersons of all the parallel sessions who have spared their valuable time and evaluated the oral presentations. I extend my heartfelt thanks to our Honorable Vice Chancellor, Sir Lieutenant General V.K. Sharma, Aviation, for providing constant support and motivation. I express my sincere thanks to Pro Vice Chancellor, Sir Professor Dr. M.P. Kaushik, for his guidance and valuable suggestions. I am also thankful to Director Administration, Colonel Sethi, for administrative support. I would like to thank all the directors, HOIs, registrar for gracing the occasion virtually with their presence. I am also thankful to Team ASCO for providing media coverage and to provide the support to make the valedictory session live. I am also grateful to IT team of Gwalior campus and central team of Noida for providing continuous and hassle-free IT support throughout the event. I would also take this opportunity to thank all the participants from universities and institutions of different countries for their participation to make this event success. I am also thankful to our students who have supported the conference at various levels. I am grateful to the convener of the conference, our director, sir, Director AIB and Dean Academics, Professor Tomar, sir, for his continuous guidance and motivation and to provide the constant support to keep the hard work. I am also thankful to Team AIB, Professor Dr. R.K. Mishra, Dr. Raghavan Saxena, Dr. Anurag Jyoti, Dr. Neha Sharma, Dr. Manish Kumar, Dr. Pallavi Singh, Dr. Pushka Udawat, Dr. Suren Singh Parihar, Ms. Soni Bhadoria, and Dharamveer for supporting the event. Once again, thank you all. Thank you very much.
Thank you, everyone, for making this event a blessed one. Thank you so much.
Thank you so much.